There was a good few seconds yesterday when we were testing it.
There we go. It does take it.
Because like I'm gonna confuse this after
Yes.
Excellent connection. That's what we like to see.
The other ones I'll use one of my accounts.
Is that right?
That's fine.
Yeah. Really just for the backup.
I mean
it's a
that's
bit boring.
not really put on a small
It's all
call
different
from
accounts, but it's just as a backup record, really.
Yeah,
I know.
mutually, give you one
You're
mail.
right to
Yeah.
the other rooms.
Okay, yeah, fair enough.
That's fine. Thanks, mate. Really appreciate it. Good work.
Right. It should be alright.
But
I am
leave
thinking.
this.
Yeah, I'm thinking I'm going to sign out your account. That shouldn't impact the stream.
Sign out on here.
Yeah, I think because basically I'm taking this laptop around to basically
Test it.
test it out, and yeah.
Well, actually, I've got it on my phone, right? Surely I can monitor it on my phone.
I guess so, actually, yeah. It should be interconnected. Signing out on here, it shouldn't do anything. But we'll find out shortly.
Your videos?
You don't have health?
No.
Yeah. Quite bad. Also, you're right, I sent an email from your email address. Basically, I need to send a YouTube link to Charlie from VMix.
Yeah, I just remember
So he's
that.
basically monitoring it all simultaneously.
It does the background music. Just send it to like the manual stream or whatever. Because it's gonna be on YouTube. No, we're not sending the background music. Because then the streamer is streaming it.
Oh, one thing. I don't know if anyone else is in this. Background music.
Where's your...
Yeah.
Have you put any signatures out?
What was that? Was that that one?
Coming from one of the laptops?
Yeah.
Yeah, that one.
Somebody
Yeah.
was watching the YouTube video.
Yeah, that's right. And the chairs are just down there.
Yeah, to be fair,
Biotech enhancements, isn't it?
Biotech Ventures.
there was definitely silence.
It shouldn't make any difference. Really,
It's all for your portfolio, Hannah.
I can't remember why it would.
You get a stand.
That's upstairs. Good.
Also,
Right now.
see the card on top?
Yeah.
Causing problems.
Maybe it's just me because I'm not
That's what you feel thing, mate. Not signed in.
Well, I put it on digital. I think.
The light will only come on when you start it.
Oh, okay. Because I was wondering why that was.
Yeah,
Because
yeah.
I'll leave you to run around, mate. Thank
I've
you.
the lights always on the.
Yeah, some of them they do
Good.
come on straight away.
Yeah.
But with the countdown touch, they don't. I don't know why.
Oh, okay. Oh, so that's different to the usual.
Yeah.
Okay.
You're right.
I'll
Just
still
confirm,
confuse- it's like
auditorium,
okay,
we're going to
okay,
both stages to begin with, aren't we?
Yeah.
Just for
We're
that,
not
because there's
doing
nothing
anything
else on.
Yeah, exactly.
Can I?
We're just going around testing the other
I just don't
auditoriums.
want the cable
Tested
to,
already.
Great.
But
cable on the
we'll
other
go.
way.
Just
Oh yeah, because there was
we're actually already
there
live
was
at
no
this.
vlogs on.
yeah.
Back here.
Have you met the AV tonight?
Forgot about it.
Yeah.
So I should have
Who,
introduced myself.
who should I introduce Hannah to?
So we've got Ed and Awara in this room as well. We've got Michael Murray's not here at the moment. He's on camera, but video sound.
Okay.
Nice
So
to meet
this
you
is
guys.
Hannah.
I'm Hannah.
Hannah, nice to meet
Stage
you. I'm
manager
doing
in the
video.
room. So she'll be liaising with the speakers and the compares and yourselves.
Great, and we've got quite a lot of panels and Q&A, so are you going to be running around with the mic as well?
It'll
Not
probably
me,
be
but-
the mic runner.
Oh, we've got a mic runner actually.
Yeah.
Full-time. Great.
Yeah, well, not quite full time. So they're sharing that job with being like the room
Right.
monitor. So like
Oh, okay.
telling people if it's full, but then they'll come in for
Or
Q&A.
I'll do it.
Or
Yeah.
Hannah will do it.
Okay, great. Amazing.
Tom.
Perfect.
We do. Two.
In here.
Two handhelds.
Really?
Yeah, it should be. Every room has two handhelds.
Oh my god, it's so quiet here.
I just saw Q&A on the schedule.
So auditorium
Yeah.
happy, cameras happy. I spoke to them, told you that.
Is that?
They
I don't think they're
themselves, they're all happy. So that should be all okay. Fine.
Just knowing that.
They're still just like everything's out. So I just let them know. So no, you're like, just let me get to them.
Yeah.
But yeah, it should all be. Everything's all
I'm quietly confident. I mean, James was here yesterday setting it up.
Yeah.
Oh, I've just put the cold air
Oh,
over
lovely.
here. Sorry.
Nice.
Overheat everyone. Bring it.
We're not starting till 10, are we? Nice.
Nice.
You alright?
Yeah.
Well,
It's a fucking nightmare through there.
at least, because I didn't build it, I'm just glad that everything's
Yeah, it went okay.
everything
So I
looks
hate that
okay.
when you turn up in the morning. It's like, oh, we only finished half of it.
Yeah,
Go,
and you're
okay.
like, okay, I've now got an hour to finish the room.
Yeah. How have you guys been? Been alright?
Super busy. Like, it's just been like 6 weeks of non-stop stuff.
Yeah.
But I can't complain.
Because you do a mixture of like cameras, video editing, right?
Yeah. So this is what I was doing. This is what I was doing yesterday. So these are my cameras and
Oh,
lights
nice.
and everything. We're doing something for a pharmaceutical company. So we had some
Oh,
1200Ds, and then we had
Fair off, a few camera setups there.
so we had 4 cameras on it: FS7, FX6, 2 A7S3s.
Nice.
Because that was the closest I could get to matching cameras.
Yeah.
Yeah, it was simple. Oh yeah, and we had a 600D just still
Nice.
as the back. So we had 60Xs as the backlights.
Oh, right,
But-
wicked.
It was nice because I could charge out all the kit on it. So it ended up being
Is it all your kit then?
most of it, yeah,
Wow.
most of it. So it ended up being a juicy day rate, which is why I phoned up. I think it was Martin who talked to me and asked if I was like, oh, "Is there any chance I could just do today?"
Oh,
they were
because
quite-
were
they
you
were-
meant to do the rig day?
Yeah.
Oh, sorry.
They-
Sorry.
They very graciously allowed me to do it,
Yeah.
which is good because it just means
Yeah.
if I do 2 or 3 of those a month, they make my month sort of thing. Whereas with this sort of job, you have to do like
Oh, quite a few. Yeah.
to make over a week, don't you?
These are just the nice. These are just the nice fillers, aren't they? When you like, if you have a slow month or something like that, you're like, oh, perfect. Okay. Like,
Yeah, exactly.
never say no to it and that.
So that was.
Oh, you found?
Yeah, I remember
That was
Let's
fun.
Star Wars.
Yeah, that was
I've
cool.
just been doing loads of stuff like that.
Are you doing the edit on that? Or are you outsourcing it? Or what?
No, I won't edit that. I don't tend to do that much editing, to be honest. Unless I've shot it. I mainly do colour grading now.
Oh,
That's
okay,
the main
nice.
thing I do.
What?
However, I did agree to edit one thing I shot for Imperial Hotels Group.
Mhm.
They're
Yeah,
based
yeah.
around Russell Square.
Mhm.
Got like 6 hotels.
Like
We're
the
doing a
other
load of internal and external videos for them.
Oh,
And
nice.
Rick
Yeah,
Brett
Oh,
agreed doing the edit. I should have passed it on to somebody else because they're like
Revision.
every day. Yeah, but the thing is, they're just building a bigger build, and I keep telling them that. They're like, look, just do it. They're going to be phoning me. They said at 8:30 today. And I'm like, I can't do anything until I get back this evening at 8 o'clock. It's just like, so I've literally.
Have they got a fast edit, like a quick turnaround for this then, or are they just on it?
No, so I allocated 4 days to edit for them, and they agreed to basically that. We got them to approve the edit after the 4 days. Everything was good. They didn't show it up their level of senior management, and you know what it's like. Everyone wants to have their 2 cents to validate their job.
I do a lot of edit for things like that.
Yeah.
And you're like, right, so you want me to basically do 6 different people's visions in an edit. It's like, just pick one. Just
Exactly.
one person.
Exactly.
But like you said, everyone's got to be like, oh, well, my job's important, so I've got to say that it's this. Like, come on, man.
Like,
You don't even care.
I spent an entire day doing a custom animation in After Effects, and then somebody turned around and went, no, we now just want video underneath it. It's like, you do realise that you paid somebody to do that for a whole day, and now you've just taken it out. It doesn't bother me, but it's like,
Yeah, because it's still money, but it's just
It's just
stupid,
like-
isn't it?
you could be so much more efficient.
Mm.
Just be more cohesive with your statements. Speak as a team, as a unit, not each individual person having their 2 cents.
Yeah.
I'm shooting another 2 more films for them, and
Dreading it.
well, we've got most of the footage, so
It's just pick up days, is it?
it's one more set of interviews that we need to do, and a little bit of B-roll, and that's
Yeah.
it. And then, but they've already eaten. They were allocated 9 days in total for 3 films to edit. They've already used now 4.5, nearly 5 days. So then they're only going to have 2 days editing per the other 2, and I'm half tempted just to walk away from it. But then I don't want to annoy my client. 'Cause
Yeah.
I do quite a lot with him. We do like TV commercials for like. Have you heard the shampoo brand Wash and Go?
Yes, yeah.
Yeah, so we do stuff with them.
Yeah, that's awesome.
Yeah, a bit of Frankie and Benny's, but they're all social stuff for them.
Yeah. So do you mainly work by yourself, or do you ever like get like other operators and stuff in?
Yeah, so I do most of the time. So that shoot yesterday, I brought on 3 other operators.
Yeah.
It all depends. Do you have your own kit?
Yes. So I've only got a Sony FS5.
Yeah.
It's like the little big brother to the
Yeah.
FS7, and a Panasonic S5 Mark II, but obviously it's not really in the Sony range.
Yeah.
Get a Canon.
Get a Canon. I've got all my own lighting and stuff.
What lights do you have?
I've got a set of dados, and then I've got 2 like a big.
Are they the LED or?
No, they're some
They're the
tungsten.
tungsten.
Yeah.
So they're 150. The 150 watt ones.
Yes, yeah.
They're good.
Yeah, I can't. I keep thinking about changing them. But I'm like, at the moment, I'm like, they're still working fine for me for what I need them for and stuff.
Yeah.
And then I've just got 2 like Pixel Pro lights, like an LED, like a nice LED panel, and then like a big I put like a big softbox on.
Yeah. Have you seen the new iFootage?
If you didn't ask about the Q&A, it would have just been.
What's it called? It's like an angel light, I think they call it, or an angle light.
Oh no, I haven't. No.
It's as accurate as a sky panel, apparently. This is what one of my mates that's a gaffer, and it's 300 quid.
What?
And it's a 1x1 panel.
Oh Jesus. Okay.
It literally came
I
out.
assume it's like Chinese, right?
Yeah.
They're just the way they can copy the technology is insane, isn't it?
Yeah, it came out like a couple of weeks ago because one of my friends, he presents the Pro AV YouTube channel. I don't know if you've seen it.
Yeah, I know Pro AV,
They do a load of things. So, and he basically said this is the one of the best lights I've seen for the price.
Holy shit, 300 quid. The thing is, it's actually worth taking a gamble on that as well, wasn't it?
Yeah, so I was so 330 on Wex. I'm tempted to buy 3 of them, put them in a hard case, and then you've got like a
full lighting setup.
yeah, because they're fully RGB. They're full RGB.
Wow, what's the same usually? Oh right, yeah.
Yes. You should watch some of the reviews of them, but I'm thinking.
Can you just run up to the top? I'll just take a picture of the.
I've got it runs off Velox as well.
Oh wow, bloody hell.
I've got a set of like LED Go ones.
Yeah.
I've got 3 of them, and they're great. But
How you doing, mate? You alright?
Hello. So I think in the first talk we have like 3 people on here.
let me check
So we need, and they ask also for a table with water. Is it possible to organize a little table?
Yeah, I'll have a word with Sarah when she gets back. She's the room manager, so she can sort that out. But we have 20-minute talk, fireside chat Q&A with Claire and Steve. And then after that, we have a panel with a Q&A with Claire, Arthur, Neil, Elliot, and Luke.
Okay. So what are you planning to do with the 3 extra chairs?
So that will be the floor manager will speak. I imagine she'll have a plan. We're in charge of the AV, so the video and the sound.
Okay.
No problem.
Yeah, so you've got the, you say LED Go's?
Yes, they're really good. So like if I'm just doing like, but to be honest, I don't use them anymore because now if I'm doing like a talking head, I've got 300X, so I just use that through a light dome, and then I tend to. I've got these Chinese, they're called Wii Light that I tend to. They're pretty good. They're 400.
Like the nan lights sort of things.
Yeah. So it's the equivalent to like a 650 watt tungsten, and I'll just use that as the backlight, and that tends to be what I use now for talking heads.
Nice.
But I'm thinking if I get those iFootage ones, I can get a set of 3 in a hard case, chuck it in the back of the car, and then you've got a really useful 3.9, and they can run off Velox. So I just chuck.
Do they have like SFX built into them at all?
I think so. Yeah.
300 quid. That's crazy.
I think they might even.
I paid more than that for. I paid like £700 for my my light panel. It's ridiculous.
Yeah. No. One of my friends has brought 2 of them, so I'm having to play with them on Thursday because I teach at a uni.
Oh,
so
nice. What uni?
Staffordshire University. So
Nice.
that's where I'm originally from.
Right. Okay.
So I still go up there every week because I enjoy it and I get to see my family. Otherwise, I would never get time to go up there.
Wow. That's a nice thing to do as well.
Yeah, I enjoy it, and also you get to play around with new kit.
Yeah. Are they a pretty good uni? Like, do they get a lot of kit in?
Yeah. So we try and get the best kit we can within the budget.
Of course.
So at the moment we're rocking. So we don't have any ARRI stuff, which I'd love us to get, but we don't have it because if we like to, because every student produces a short. It's not like you're working in a crew of 10 where you could get, you know, a 35 and it only goes out 3 times. These cameras are being used all the time, so they've
got to make sure they have enough to cover all. Like, how many students is it to a course?
So it depends on the year group. It can be as high as 120 and as low as sort of 40.
So have they got to have 120 cameras available?
Yeah. So if say for example, we have 25 FS5s. We've got probably close to 30 C100s. They hardly ever go out there, just first year cameras. We've got 10 Blackmagic 4Ks, 10 Blackmagic 6Ks, 10 FX6s, 20 C70s, 5 Red Komodos.
Bloody hell.
And then we've got like, because we also have a broadcasting journalism course, we've got loads of like all-in-ones as well, but I don't really have anything to do with them.
Yeah. Oh, mate.
But yeah.
Wow. That's awesome.
Yeah.
Well, mate, I've maybe send you my CV over in case there's anything you need. I've got like a show reel and everything.
The main thing is just website, what kit you got, and
show
reel.
Perfect. I've got a kit list and everything.
I don't really need a CV, but just
cool.
whatever you've shot, and then
perfect.
Yeah.
I'll take your email. So.
Do you have Instagram?
Yes. Yeah.
Just drop it me on Instagram. I'm not that formal.
Wicked.
I'm terrible at using it, like
Yeah,
in terms of posting.
Yeah,
but
me too.
I speak to people on it, so I'm just _Edstone.
Top one.
Yeah, that's me.
Oh, nice.
Do we know anyone that
Sachin?
Yeah, he's a gaffer.
Yeah. Nice guy. I used to work with him when he used to do like wedding films back in the day.
Oh, okay.
Yeah, a long time ago.
Nice.
Oh, no. Starving. Oh man, I was so lucky. I was walking up, the prep was open. I was like, yes, getting a breakfast in there quickly.
Yeah.
But what are you floating around?
Yeah.
So I suppose you've got to be ready for 9, haven't you? Really?
Yeah. Sort of being made for the next half an hour. Just kind of.
No, because your being since kind of stressful.
If you could just, if you could just kind of, as you are walking around, just is everyone okay?
Yeah.
You guys okay? Are you all set? When they start first, I think they're called in there. The streams are both live next door.
Yeah.
So I think we're pretty ready. I just want to go and help Ben. I think
Yeah,
we're
that's fine.
struggling down there.
Yeah, that's fine.
If I can leave you here.
Yeah.
as
Do you
well.
hand with anything? Because we're pretty much all set.
I'd rather you guys be here in case people rock up. I think.
Cool.
This is the bigger job, and I really, really like the organising of it.
Okay, great.
Whereas then,
Yeah.
it's just really boring because Ben, as he's new, he just doesn't get it. He left last night. None of those rooms are taken down out there at all. Didn't even find us. I told him about 20 times. Sorry, that we've got spare speaker stands in the storeroom. Yeah, speakers on the floor.
Okay, so
Came this morning,
when did he start?
3 months ago.
Yeah.
Oh, so he should be starting to get a grip of things.
Should know how to get a screen on the wall and everything.
Yeah.
And not necessarily him doing it, but if he sees it, tell someone, you know, that's not right.
Yeah. Okay.
Anyway, I'm just going to go and give him a hand.
Yeah.
But yeah, just keep my eyes and ears up.
Yeah.
Oh, Hannah, somebody came in and was asking about chairs. I think they've got
Yeah.
5 sides.
I mean, it's minus 30, but the thing is, hopefully, so I think we'll push it a little more.
And then we'll send the curve on it and then it's the maximum.
Cool.
They have time. As long as everyone's actually.
Thank you, mate.
Yeah. Not starting for a while, is it? We've got time to just quickly fix it up.
Yeah, of course, mate. Yeah, I think 10 o'clock for starting. Thank you, mate.
We fixed it. Well, but it didn't break free. It's like it locked. On what? In there?
Nigel's room.
Oh, right. Yeah,
That's
no,
why
it's
it's
one. Yeah.
the NDIs went all funny and everything, but we basically he's sort of using it now.
The laptop froze up, but it wasn't like frozen.
Cheers, Hannah.
Yeah. So hard to
I mean, that's the
carry
sort of thing.
Well, if
we're
you
gonna do
need a
all
hand,
day.
because it's just one slide I've got to change, so I can always help you pull on and off.
Thank you.
Okay. Oh yeah, so those are the only two though. Yeah, we don't have any more. So let's log into our own account, start doing our own account. We're not doing it. But the main record is OBS, and then obviously your backup. Yeah, I've got a timer set on my yeah.
Headphones to work on that beam.
Yeah, that's standard,
Might just
mate,
leave it out.
honestly. Yeah, that's what I would do.
Yeah.
Genuinely. If you desperately need to reference something, just have it come out the screen. But,
I don't think there's speakers of any kind.
as long as you can see it meter in.
That's alright.
Yeah.
Cool. I'll
Right.
go to the main room in a few minutes.
Wicked. Yeah, so all the breakouts start at 10 o'clock.
10 o'clock.
So in theory, if you have a look when you get back, just ping us a message to let me know whether you become a breakout or not.
Right.
Because it might be that your schedule ends at 10 o'clock.
Yeah.
Right.
How are you doing? You good?
Good, thanks. I'm fine, thanks.
Good.
Good. I just wanted to check with you about water. I would put them on your table and hand them to the students when they come and get my stuff. Some of them won't want to.
Your screens are all set and ready.
Yeah,
The subtitles, captions are running directly through YouTube, not the Googly thing that they were on before, so they will be, I think, exactly as what you pictured. If there's any problems, ping us a message.
Okay.
If you want us to change what's going on those screens,
Yeah.
if you've got a plan, let us know. If not,
Got no
just
plan.
okay, send me a message then.
What's full is what we want to stream.
Okay,
Whatever's
right.
full.
So
and then auditorium to start with.
that's already set and that's
Yeah.
literally live now.
Perfect.
So yeah,
So when they start
that's going.
speaking, you'll switch into that. At the
Um,
moment, is it holding slide?
So at the moment, it is just whatever is going on in there. We'll get it swapped to a holding slide. That's fine. What time do you have people coming in? Like just to the
now.
area? Oh, okay.
8:15.
We'll do that now.
Registration opens.
Okay.
Thank you.
All right.
No.
No. So they're speaking out loud. Okay.
Yeah. Yeah. We decided not to because it's too much.
yeah.
Like logistically,
Because
and
a
not because we took off coffee tables because it was too much really crazy stuff. So we brought coffee tables to make it more complicated.
Yeah,
If you're having a glass on the floor,
that looks
all right.
Yeah.
But glass on the floor looks like it's a bit dodgy, and someone's got to clean them up and stuff. Okay. Cool. Thanks. I would put those out, Hannah, on your table so you don't forget. I would just put like 15 or 10 out or something, so that it's also simple to know. Are you all right? You got to do it.
And those three are
Yeah, exactly.
not Heidi, you'll be with Hannah, who's not Hannah. You have Nikki?
Oh, hi. How are you?
Oh, you've got them all. Right. Okay, I'm going to take the others to meet their stage managers. Sorry.
Lucy, I have just asked Sam. Lucy. Lucy.
How many chairs?
I have just asked Sam for the number of chairs.
I think it's
So we should get that in place then.
No, I also asked these guys to count how
Oh,
many
okay.
chairs there are.
That'll be fine.
Okay, so breaking barriers.
You don't know until you've got the review.
We're not doing cuts. I've just pitched them for now. It's fine.
We didn't want to use them though. It was a strategic decision. We took them out because it's messy, because we're not having coffee tables.
Hannah, do you know if
What number did you guys say?
88.
88.
Yes.
Thank you.
Only on this part.
Okay. You got 88. What on that side?
Just a second. Altogether.
167 you got. Okay, cool. Let's see if we can. That's what they reckon. So I did. Okay, so don't have equal numbers. Nine times 6. That's 80. Okay, so 134. And then we've got shorter rows. So 134.
Yeah, so I thought I'm that two screens in Italian. One on the lower one on the upper. That's one just on the down.
I got 157.
Yeah.
167. This is going to be fun. I've done many conferences before, so. No, not.
There is quite a few Q&As.
Oh, okay. Already known about.
Yeah. So with the first one, we have 2 chairs on
And it's more sort of photographs and things of, like cancer cells and stuff like that, but incredible paintings. So, right, so this is where I need to come.
This is where you need to come.
Yep.
This is your stage manager.
Hello.
You need to come and mic up here.
Okay, that's
Yeah.
fine. No worries.
Great.
I'm speaking at 4:30, so it's just coming to find
Oh,
out where I
yeah.
need to be. Hi,
Hi.
Susan. Nice to meet you. Likewise. I'm the runner. Very good. You guys are super organised. I'm very impressed. Huge venue as well. I'm like, wow.
Yeah.
Yeah. Okay, good. I will have another look at my slides and remind myself of what I put in there. It's quite quiet, like 8 minutes is not long.
You can do it. You can do it.
Brilliant.
I'll be smiling.
Yeah, you can do it if you've got a clock or something, so I can see your 1 minute left.
There's a little bit of stuff that might be happening.
Brilliant. Excellent. All right, see you later.
Okay, so yeah, there's 2, then there's 5. So people can run chairs.
Oh, hold on. So there's a 10:50 session and there's a. Huh? Is this one session, or are there 3 sessions?
Yeah, 3. So this one is your fireside chat. These 2 are
People are coming more. So good. People moving in and out.
I mean,
To reset it all.
I would imagine. Say.
Okay.
So each is one. So there'll be 2 kind of lines.
One for them and one for the audience.
Yes.
Yeah.
No, sorry, both audiences now have.
Oh, okay.
So then I'll give you one of the
Yes, yes.
Morning.
Good morning.
I'm looking for the audio-visual team because I'm a speaker. I just need to have a quick look at the slides.
Okay. Is no one there? I'll go.
Is it maybe I'm just too early?
They have been in here.
They were just here, so
Oh, okay.
There you go.
Oh, hi.
Hello.
Hi, one of the speakers.
Oh, nice to meet you.
What's
I'm
your
Ed.
name?
Ed. I'm in charge of I'm doing the
Ed,
video
could I
stuff.
check? I need to make 2 small changes to my slides.
Yeah.
I meant to say
Yes.
actually, you can come at 3:15.
Okay, 3:15.
Yeah, because there's a break between 3 and 3:30,
Perfect.
so you don't need to be
Oh,
here.
okay,
So
3:15.
early.
So actually, get mic'd up here. Or-
Mic'd up right here with these. Yeah.
With
So
Ed?
come here
Okay,
and
okay.
they'll get you mic'd.
Right.
What time are you speaking?
I think Arndt Chatil is so
Sorry.
here. That slide. You see that?
Uh-huh.
Second last.
Aunt, yes.
And I
Okay.
just is it PowerPoint? Maybe I
It
can
is
just.
PowerPoint. If you come here, I'll get it up. for
Happy
you.
to identify myself, so I'm not changing anybody else's slides. isn't it?
I trust you. I trust you. Right.
Just need to remove some sensitive numbers. That's it.
That is fine. Let's get it open. Cool. So this is these are the slides I have for you.
Yes. Do you mind if I just go right here?
Starter.
You can supervise me that I'm messing anything up. So. So.
Do you want that
Is
gone?
there a cursor? Okay.
Yeah.
Here. Good. That's one. Can I check if this is working? This is the only real.
This will be the clicker for you.
Okay.
Yeah, so just have it. Practice. So green go forward. Yeah.
This is what I want to see.
Yes. So this is what we'll put on the clicker.
Did you, just a question,
Yes.
because sorry for the inconvenience.
No, not at all.
Actually, you did not change. I sent over to Lara.
Can I introduce you to Isla? Isla is one of your speakers this afternoon.
This is the version that was
You're in the
handed to
Biotech
me.
Spotlight
So when I
session,
came this
aren't
morning,
you, at
these
3:30?
were already loaded
So,
on.
this
So,
is Hannah's the stage manager, and
It's
then
not much
she'll
change.
make sure you get mic'd up.
Do you want to, you can put it on this memory stick, you've got access to it,
2:45?
and then
No.
This is what they told me. 3:30 is a talk. 2:45
Can we do it from
No,
my
it's
iPhone? I can send
fine.
it.
It'll be 3:15.
3:15
Or
or
3:30.
3:30.
Because I have it
3 o'clock would be fine if you wanted to come and double check.
If
your
you don't
slides
mind,
and
because
things.
No,
Or
that's
if you
fine.
wanted to
Time,
check
do we?
slides, you
Yes,
can
we
do
do.
that
Let
in
me
Oh,
the
that's
morning.
fantastic.
With
It's
these
good I'm
slides
checking.
in there.
That's
Yes.
great. I'll do it now.
Do it now. That would be better.
But maybe it's
Yeah.
coming.
3:15, something
You always
like
sent
that.
it yesterday
Thank you.
morning, but no worries.
We've got water. We took coffee tables off just because there is quite a lot to move on and off, but we have got spares if we really want one. So I will grab you. Can you liaise with Hannah and get a ring to go and get a coffee table from the atrium? We've got all the spares in the atrium. We have them.
Hannah is your
Oh, okay. Yes. I could just use some. I could just use this. I'm allowed to write in this. I'll go. Okay. So. Okay. Okay. So, between 3:15 and 3:30, you'll be mic'd up later, and no chance to speak with their meeting or anything like that. So yeah, if you come back at 3:15.
You're finding it confusing.
Is there anything? Yeah, obviously you can come earlier if you need to speak to certain slides or anything like that. Maybe from 3 o'clock.
Name, how are you?
Oh, hello,
Lovely to
Jason.
see you. How are you?
Nice to see you.
Great.
How are you?
I think you're in here later, aren't you?
Yeah, I'm here,
Yeah.
so that's why I came to be familiar with.
Good. Yeah, see buttons to press. So how are you feeling about your 8 minutes of glory?
I think it. I think it should be fine.
Well,
I tried to fit the slides within 8 minutes. So good.
well,
I can trade you 1 minute for good price.
yeah, well, it's probably going quite. Yeah, both probably. Good. Yeah. So I emailed Peter yesterday just to say
I've
it's
seen
a
that,
shame
yeah.
if he can't make it. But
Yeah,
it's-
he's down. He was keen to come, but
it's a long way to come if he's down in the southwest.
Okay.
So yeah, I'll actually be introducing him.
okay.
So I've got seven companies, 10 minutes each. So I'll be trying to be key timekeeper.
So we are doing
And I'm going to try to come up to Sheffield. You're having your opening.
Yes, I need to send you.
They're outside.
So just book save the date, the 10th of July.
Yeah.
Are you
I just wanted
No,
to
please,
see my
please, go.
slides. if that's okay.
Oh, yes.
Sure. See how warped they are.
That's fine.
And Biotech, that's the first one at Biotech.
I would say put them in. Yeah.
Massive queue.
Green's good. Red's good.
From what walk of life, or is it a secret?
It is.
It's a secret.
It is a secret of the world. So
Okay.
it's a very small group of 5 people that the industry who knows.
Okay. Yeah. Well, if the more VIP the VIP
This is what
is, then
But we do have,
Everyone else will see.
Cool.
Great.
Coming as well.
Thank you so much.
well, in my mind, you're the VIP.
Oh no.
Because you're the person who's leading this, and it's yeah, that's what it's all about.
Thank you. I, it's if we can do something to impactful on patients.
Yeah. Have you got any people lined up for batches?
No, we do work with currently we do work with 2 academics at the moment.
So in process development, analytics development, so that when GMP comes online,
Even the outline for
Okay, I've found it.
So you want and
what production scale are you using for
Let me send
No, I
you.
Oh, so you've already got some 50-litre bioreactors,
no, we do have 50
That's fine.
and they're up and running.
Well, they are fully set up now.
Okay.
The 2 liter has been
I think it would be fantastic.
Yeah. So it's process development scale.
So it's exciting. It's a lot of work. It's exciting.
It's 50 litres.
We recently checked the head of GP. She came from a biotech company. She's very good. So
Okay.
they really insist on her.
What's her name?
Holly Garth.
Holly Garlic.
She worked in Manchester.
Well, it's probably a pretty large file to
Oh, is it? Has it left your outbox?
It's exciting. And then, obviously, by platform biotraceable. So
Yeah.
the lead programs is probably too late for GMC to manufacture for both. Actually.
Yeah.
Platform bio, but it's pipeline, second, third product, pipeline
Yeah.
product.
Yeah.
Yeah.
Any moment.
Well, it's attractive for investors, that principle that you've got reach through to
Just on one huge
facilities.
worries. I've got some prints to do there. There's never been any changes.
Great. Is this
You know how it is.
It's the same as the digital version.
Oh, okay.
Well, there's no way
I just
around
thought-
that. Because apart from anything, you've got to purchase the materials.
Thank you.
Just like with the bill of materials for
There we
GMP
go.
and getting it all in. It's the supply chain
Yes,
that has to be foreseen. If you have enough process standardization,
Is it both?
throughput,
So
then you
well,
can-
I just need.
Oh, one's a PDF and one's a
Yeah.
PowerPoint. So let's download the
shelf, but
so that we can make the changes we need to make.
So
Great.
looking forward, I mean, for you.
And we can delete
Yeah.
I was about to.
this whole part.
No, I already opted in. I said, I want to go to Sheffield. I want to see the moon. I want to see this opening. It's going to be cool.
Oh, that's would be great.
Yeah. Good.
It will be a big event, so I forward to it.
I was actually looking at your deck the other day. So
I was advised to come early.
previous
so before you
XB102 Choice Astronomy.
Yes, no, it's definitely the best time
slightly
because if you
shorter
try and do it
version
in the middle of the show, it can be quite
shorter version.
quite
Stressful.
tricky. Yes.
Was that already available?
If I enable editing.
Is that one of the constructs that they
Okay.
screened
Oh, it's on your
3-4 years ago? Because there's the sort of there's exactly the same cassette that was in 102, and then there's a slightly 500-600 base pair shorter version.
I think the short version is the one that went already to patients. No.
Okay.
It had two 2A elements in it as opposed to a fusion plate. It looks slightly different, maybe.
So we had information from actually pushing us as well with deficiency.
Yeah.
So we have been in discussion about design, which constructs, and we thought that we can test both the shorter version and the one with
Slightly longer. Okay.
So the shorter version is perfect. So it's the packaging for AVDs.
Yeah.
It's no problem.
Oh, you've already shown that.
So
Or theoretically, the size limit should be.
Yeah.
Okay.
So the second one is at the limit. I mean, it's under the packaging capacity.
Yeah.
So it's within the packaging capacity.
You can still have drop off in tighter or quality if you're going towards the limit.
Yeah. So we will work around that. I mean, we are
Okay.
playing with.
That's it. Should we?
Let's
I'll let
save
you
that.
save it.
Shorter
Yeah.
Yeah.
I'm going to put it on this memory stick as well, just so you
will allow us to.
have a copy of it.
Well, it's fantastic that you're working with it on a live project, and
So
I
you're happy with
reintroduced Peter to
Happy with it, yes.
to Roger Barker.
Great.
Thank you very much, Ed.
No,
We'll
thank
see you
you.
later.
Who
Eh?
did
We'll be
you
there
later.
3:15 or something.
Yes. See you
I
later.
need to talk to him. We haven't yet. So I know Roger very well because he is one of our partners
doesn't
on
sound
Arc.
relevant
to you.
It's more camera.
He
said he was happy.
So I need to.
Yeah.
As long as you can see the projector, that's all I care about.
It's to the MRC Santa Fe itself. But I need to talk. I need to talk to him about the.
Yeah, I'm not sure if he's signed up
not
to tonight. Some of our
This afternoon.
SAB chairs are here.
Yeah.
Like, I don't know if
I don't know if he's around.
But you might be.
Yeah, if he's around here, I may.
Literally, this morning, it's just on.
All right. Well, I'm going to check out the podium.
Okay. All right.
See you later.
So, thank you.
No,
See you
problem.
later.
Yeah, very close. You do? I thought you might. And you're a charity event? Yeah. Yeah, because it's now. It's a self-funded. Yeah, charity. Yeah, not donation. Like huge amount of investment. Well, we have Life Up Ventures, which most people on the stage will be from. So Claire.
Oh, I'm looking forward to
Claire
it.
is one of the composers. She's head of Life Up Ventures. So yeah,
Good stuff. I'm going to find a bin.
yeah, there should be.
Do
one.
you need me to take anything? I'm good, thank you. Okay, good, thanks.
Nice. Next recycle.
I don't think power the other issues. But
yeah, they are. They're kind of awkward. Careful, but the best way.
I was going to say, have you learned a technique?
Yeah,
Yes, she has. What do you do at Biotech?
Marketing.
Oh,
Yeah.
digital or everything?
Both. Mainly digital, but a bit both. So all the launch and stuff.
Well, I think you've done a beautiful job.
I didn't design it though.
You commission the people to do that, right?
We've got head of brand, she's got design as well. So
Yeah, when I was at the NHS Leadership Academy, I was in charge of the marketing team. So, but learned a huge amount from the team, and ended up working with designers for absolutely help. Yeah, so they're marketing. I like the creativity. Do learning and development. It's a bit of my background and people engagement stuff. Yeah, so it was a bit out of my comfort zone. And then I sort of sat down there with the team and I said, do you know what? It's all human behaviour. It's all human behaviour. What do we need to do to nudge people to do?
yeah, it is. Yeah, it really is.
Yeah. So once I kind of got that click, we just went, right, and then we did some great stuff. So I was very proud. Under my watch, we drove our socials from 75 fellowship of 75,000 to 225,000 in 18 months. The girl does the content. I do the content, you make it pretty and push it out.
Yeah, absolutely. Where are you from?
I originate from Australia, but I've been there for nearly 4 years.
Really? Where else?
Far North Queensland, little town called Ingham.
Really?
Oh, are they?
Yeah.
But it's not.
Oh, as soon as I speak to someone from Australia, and I think I sound more Australian now because I've had a friend over staying for 5 days.
Okay.
So you know, calm down a bit, yeah.
I don't mean even when I grew up.
You go, oh, you're Australian, are you? I'm not. Hilarious. So, when I put my case into the local
All going good?
So far, they're just about to start.
Nice. Hello.
Who their stage manager is? I believe over here. Oh no.
Hi, I'm Michael. I'm just working on campus.
Hi.
No, are you safe now? Oh yeah. You're right.
Hi.
The runner.
Okay.
So that's one thing.
Oh sure, yeah, yeah.
Right. Okay, thank you. So that was for
Paolo's out, man. But Paolo, the chairs are quite heavy, so I said I would help with them because there's 3 and we need to get them in.
These are the white ones, aren't they?
Yeah, there's 3 outside and we need to
I'm
bring
getting
them
some,
up.
but it's
So,
really tiny.
That will be at 10:40.
Yeah.
Okay, that's fine. Thank you very much. And then again,
So we just the rest of it. Yeah. So the rest of it is just taken.
It's like minus 35.
2 here, 5 here, and then.
so I've got a
Yeah, added plus 2 for
Yeah, that's right.
coffee table.
maybe the same.
I'm sure it's probably.
So that's what you need,
Yeah.
and I'll bring it in. And then that's the morning.
Yeah.
And
Yeah, so if you're doing that, you're getting like 35.
19 past 2, something.
I think it's on the settings because they got the same issue yesterday.
And one chair and one coffee table.
Even when I was sending things
Yeah.
to
So
those
we'll
people,
move
I would send them out.
Okay, mate.
Okay. Again, just let me know if you want to keep I'll be around.
Where did you get the tea from? Did you get it from the cafe?
Yeah. So literally just as you go out there and
Yeah.
where you want, yes.
It's expensive though.
No, they do a free. They're doing free teaser coffee. So you know when you're just on the phone. If you walk through there, you'll see there's like a station of pastries and teas and stuff there.
Oh,
Yeah, mate. I was gonna say to you, but I assume you're on the phone with the client.
I was, yeah.
How did it go?
No, but I just
Yeah, fine. He's it's one of those where his client's been
no, I just didn't want to start backing it in before.
Mauritius. The project was supposed to be done last week. I've obviously taken on other work because I only scheduled in those 4 days for the edit. And it's just like they're like, we want the changes now. And it's like,
come on.
Well, what do you expect? You know, you were allocated time.
Yeah, you've
gone over that time.
That's not going to happen.
If anything, we should be charging you extra. I don't know if you want to.
That's fine.
Right, I'm going to go and grab a tea. Do you want a coffee or anything? Tea? Just like breakfast. Do you take any sugar?
Ladies and gentlemen.
Do you want not enough sugar? Just a bit of milk? Or back? Right, I'll be back in a few minutes then.
So it's both of these here.
So you see, it's like this thing.
Thank you.
Hello. Do you know whether there might be any XLRs? Because the mini jack PC one was cabled.
I don't know if there's any. Yeah, there usually is a cable from somewhere. Hey, you're right.
How's it going?
Yeah, I just don't know where it is.
Was it early start?
Were you here yesterday?
Yeah.
Do you know where the cable trunk box is just for XLR?
My one is at the back.
Okay, yeah, you might.
You got two at the back. Spare.
How you doing? All right?
Isn't it?
Yeah.
So
I can't remember your name.
Michael.
We
did a few things at the NEC
Yeah.
and loads of little bits and that.
Yeah.
How you been, mate? You all right?
Yeah, I'm all right. Just cracking on.
Yeah. Are you au fait at all with the Atmos show?
No.
Okay, perfect.
What's happened?
It's just an audio thing. So for this one here, we're just getting low audio levels coming through. But
what's it chucking out on the desk? Is it?
Desk is fine.
Yeah. No, I don't know. What about this man here? He looks like he's quite smart.
What's that?
we're just having it's a minor issue.
The audio level's not quite high enough. Shall I get out?
Oh yeah. How much? So what are you pumping into the camera? Also, we want to do that to make sure it's recording. Otherwise, recording.
Right, turn it on.
I would say then on here it will be what you're pumping out of the camera.
Yeah, that's it.
So then you can increase it. That just increases that.
Headphones.
Okay. Let me have a look.
This does increase the dB, but you can see we're already at max.
Yeah. Sorry. Let me just adjust the camera and see if that actually does much.
Hiya. Hello. How's it going?
Yeah, good. I'm your first speaker on, so I found the right room, which is always good to know.
Great.
Before we, so I'm plenty early. It's good. I'm on at 10:30. So I'm Steve Bates at 10:30.
Steve Bates, yes. And you're going to be with Claire
Turlow. So I'll come and find you 10 minutes before, and I know where I am going now.
Great.
That's good.
Just to check, we've only got one slide for you, which is just.
That's good. We're fireside chatting, so you don't have slides in a fireside chat.
Exactly. I was just making sure you're happy with that.
If you come back, we'll get you mic'd up. And,
Cool. No, I know where I am, and it's always good to.
Hi, are you one of the speakers in
I
this
am,
room?
yeah.
Hi, I'm Hannah.
Hi,
Stage
Hannah.
manager. What's
Good
your
to see
name?
you. I'm Steve Bates.
Steve. Okay, so you're in the first
Yeah, I'm up
section.
first.
Okay.
So I found the right spot, which is good.
Yeah.
So I'll come back at 10:15 to be mic'd up and break all that stuff. We're on to 10:50. Is somebody going to give us a time out or a time down? Are you still there and give us a quick time for you to shut up?
The agent, yes, I actually have
That'll be
both
running.
it left and
Excellent.
left.
That'll be running down the extra.
Okay, so I'll be at the back here.
But
And there is a
it's
time.
actually going to fit to the camera, and they will just kind of connect the eye. Why I also sending an extra fee to this question.
I'm going to wait for this. Then you can sync them up. Haven't jammed the time code.
Let's just make sure.
What you could do.
Anymore?
Which this is you're not. It doesn't
But
like
you
use
use the mic.
I'll be waving, yeah.
And yeah, it's got one on the internal. So this is his
Yes, so
internal,
there'll be.
and then that's his external. It's two channels. So it's stereo. So you're going to be. So if you start sending something,
And then it's finishing with one next week to add more chairs.
we'll start seeing it.
Great,
Cool.
thank you.
No worries. See you in a bit.
I'm sending the book. I'll send the camera, and I'll send the.
But what is it you're sending?
Pink noise.
I'm not seeing it, but you plugged in. So channel 2 from your camera. That's what we're getting from the desk, isn't it?
Yeah, input 2 on here, yeah.
We're getting
So if
those now.
I drop that all the way down, is that affecting it? I'll put that right down to like 1 from both of them. But that should.
But we're getting quite a lot now from your internal mic.
Okay. I was just going to go and see if someone knows where the camera box went, because I just want some headphones.
Yeah, you should have it.
Yeah, no, they should be in the camera box. Yeah, so why is there no slide capture?
I assume you should be sending some in here and it was lined up correctly. Is there an XR going into this?
Yes, at the back there, yeah. Is that coming from the desk?
Yeah. Could you just speak into a mic? Oh, I wrote that.
Did you?
Record or die. We've got to shake it off.
I don't
Yeah,
channel
plug
2,
it.
that's definitely.
This one, send it out there.
That must have been on there for literally 3 months.
So it's signal passing through that. So
Yeah.
then I don't know how much.
It should be picking that up. What's that going into?
That is going into the back, into channel 1 in.
One in.
Of recorder channel 2.
So that should be coming up here.
And then the other side is recorder channel 1, again into channel 1. I'm just going to go and see if I can find some headphones, because this would be a lot easier.
Oh, there we go. Yeah, because we were line. That makes sense.
I was start to say it's not something that you have to do like post or pre.
No. We were just
So
being
now
an idiot.
it's like here.
Gone.
We
Fixed it. Okay, what was it?
line input.
Yeah.
What did you change?
You go to analog, and then you can change if you want phantom, line, whatever.
Oh, perfect. Okay, brilliant.
Maybe it's worth to tell to the other guys as well.
Yeah, just make sure. Okay.
Do you want to just send a bit more? Just want to check that.
I want to check with the mic.
Basically worked with
Oh, yeah, yeah.
scientists was doing everything that created his work and
Testing 1, 2, 8, 8. 1, 2, 2, 2, 2. Testing 1, 2.
mean, I don't know which audio they'll use, but this isn't being monitored, so I wouldn't recommend using this.
No. So what's happened is I'm taking away the edit from this OSB.
And this with the lab here.
So these are just.
Are you doing the edit?
These are just fail-safe backup.
Cool. Cool. Testing feed. Nice.
So no, I don't do it.
Hello. Testing feed. Testing feed. Yeah. Testing. 1, 2, 3.
Yeah, that's great. These should hopefully just be an absolute backup of the backup.
Yeah, because I'm not monitoring. You're monitoring that, so hopefully.
Yeah. And then when we go,
you're good there,
we don't need to stop. We're just going to leave these continuous.
Yeah, I'm just going to leave it throughout all the sessions.
Yes,
obviously
please.
break the breaks. And you do on that side.
Yes, and I'll be on the camera. Monitor in there as well. Nice one. Thanks, guys.
There we go. Happy days.
How's it all going? I'm streaming out there, all right?
Totally with it.
Yeah, basically, if the stream all goes well, that makes my
Yeah, I think it's worth telling to all the other room as well because in the West Argonne yesterday.
Oh, they all having the same problem?
I think so, yeah.
Right, okay, let me go and check the others. The auditoriums, they were all right when they went, so I'll just go and check.
Auditorium, I believe so, but I'm not sure if it's one of the next one. I don't know.
Okay, I'll go and check. Thank you.
Please.
Just busy. Not doing anything interesting though. Just cool. Way interesting. What about you? What have you been up to?
Like January when I was with you, February, March as well. Now it's still busy, but nothing huge. And I got like few cancellations.
Oh really?
So it's a big up and down.
Yeah, I haven't really been getting much events work to be honest.
You know, I don't know music, so it's kind of balanced.
So the young
Like in the May, it's
so
still.
in my day,
Yeah,
And I got like 5 days cancelled.
I missed your
that's not nice.
And now they're saying it's
For example, those
really happy to do exchanges.
Yeah,
and there's
I'm
another girl,
so
Isabel,
far
she
in
just spent
May,
3 months
I've only
traveling
got
that age. She said
1, 2,
she'd
3,
love to
4,
get
5,
a year-long visa. She said, I worked there, it was really easy.
I've only got 8 days into May, but I am going on
I
holiday.
think if you're happy to do hospitality.
I got like one. I can just my day off.
Well, same here as
Like 1, 2, 3, 7.
yeah. It's good. It's not really what I do.
Yeah, but it's not what I'm used to.
Yeah.
I'm used to like 2 days off.
Yeah.
And then we still like this, like today I'm working here, and then as soon as we finish it, like 7:30, so we out.
Because I just thought
Staying longer.
Where you? Oh, you got another job on?
Yeah, do some club in Central London. Yeah.
Oh my god. You're gonna be knackered.
Yeah, quite a lot. But that's the thing. I have to squeeze in stuff.
Yeah. But at least you get double pay.
Can't pay a little bit less.
so if someone tries to sit there and say, I need your name, please.
But it's fun. And
250.
yeah. And also,
Alex will
it's not
say,
stressful.
this might be your turn.
You're having fun. Are you allowed to drink and stuff at the club?
We were used to.
Yeah.
You can still drink. Now it's a bit. It's those posh mates and that. Depend on neighborhood to be
Yes.
honest.
Yeah.
But yeah, it's not but just gonna be the stress to run it, then I just want to open up then. Yeah, it's working out.
Yeah. All good.
Thanks, guys.
No problem. I mean, these are basically little two shows
I'd say
smashed
if there's
together.
more
Yeah.
shows than one.
I've got a little, well, it's not little, I've got a sumo, an animal sumo.
No,
so
no,
I
no,
use
no,
them
one.
all the time.
Sorry.
Can you record anything lower than ProRes LT on those?
No, but I don't ever use it for recording. I just use it for, so like here, we were using it. So we had two. This was one of my brother. No, my friend had another one, so he brought his on, and then I brought mine on.
They're amazing, aren't they?
It's just nice to have a nice big display.
Yeah.
To be honest.
I did a short film a little while back, and the guy who was doing first AC focus on it, he was using that.
Was he?
He was like, "This is insane."
Wow, that's pretty.
Yeah, it's a pretty nice monitor for focus on,
Yeah.
right?
Yeah. Jesus.
Push your legs, still a bit close. Push your legs. That's good, stay there. Have a look.
Yeah, nice.
Make sure you look comfortable from the back.
Are you happy with where the countdown timer is, Hannah?
Yeah, they already adjusted it.
Oh, have they?
Here, he was doing the first one. I think Claire should sit there.
Okay.
And I think that one could move over to touch as well. And because I was just thinking, if we bring 5, you don't want to have to rearrange these. So yeah, if there's one, this one goes back a little bit, then there's that one looks out, and then you can arc it a bit. I have to say, if it was me and I was, doing presenting, I would be doing that as a chair. Yeah, I would be too. I'm sure that the experienced scientists who know what they're doing. Do you know what? I didn't count that chair. So 166, that could be 167. It's 166. Okay, that, it's in this space. Because that's not just for anyone. Yeah. It's an access requirement for. I think you'll see it. But I'll let you know when we get to 160. Yeah.
What food is that one?
We don't expect really. The morning session, we hope we've got people staying.
I might go for a quick smoke before we start. Because we're not starting until 10:30, are we?
Hopefully they would come in.
Yeah.
Just looking at time.
So we've got an hour, haven't we? So I'll be back in like 15.
Depending on what I can see how the first one is, if we really interconnected. The CEO room, we might have a little
Sorry, mate.
the space is starting to heat.
No, you can stay there.
So what's happening?
That is just doing the ventilation system.
I
If you can hand it in and hand it out, you will make money.
Yeah,
So that's what
yeah.
we're trying to do.
Yeah, and you heard a lot a year ago or so, constantly the government was talking about the UK as a science superpower, and there is a lot, we touched on this in earlier panels, there is a lot of evidence that we definitely are. If you look at what's going on in the fundamentals here in the UK, what's your view on why we can be so competitive?
So my take is, if you look at what it is that's going to make the next period of time, it's a fundamental understanding of biology that's changing from it being when I was at school, you chopped up rats and drew pictures, to it becoming more like chemistry. Now it's physics and maths because it's bits and bytes. So biology is fundamentally changing, and the best place in Europe to do that is the Crick. Well, this is my shorthand for it. Loads of that going on in the Crick Institute. The other thing that's changing is AI and machine learning and the developments there. If you look at where Alphabet have chosen to put their global HQ of that's in Google DeepMind, that's in London, that's not in California. Their expression of that in a company is Isomorphic Labs, that's in London, in King's Cross, not in California, not in Boston. If you've got the fundamental understanding of what's going on inside cells and the new ways of doing biology with the fundamental understanding of AI, I think you've got both of those in London. That's why I'm so confident. Both of those come together in King's Cross Station. If you go halfway between the two, that's platform 9 3/4, and we all know that's where magical journeys start.
Okay. So 9 3/4. Next time you're in King's Cross, you'll know exactly where to look for Steve. He'll be there.
But it's true. It's what it's competitive advantage.
Yeah, it is. Absolutely, it is. Yeah. I mean, we see so many companies coming up that are tech enabled.
And the reason I say this is I used to work for Genzyme, which was a big US-based rare disease company back in the day, and Kendall Square was right for the time then because it was a cheap space in Boston between the universities and some of the money people. I think that the competitive advantage is now slightly different, and the connectivity of London, the ability to do those two things together. It's quite hard to put that together in the States in one place because many of them are in California, some of them are in Boston. We're actually better at being an ecosystem that can connect better as long as we connect that ecosystem fast.
Yeah, and are you finding that the membership of the BIA is changing as a result of this shift?
Yeah, I think, we've sort of tried to get the term tech bio up as an example of that. I think maybe in a decade, all I'll have done is take 3 letters from the front of a word and moved it to the back of a word from biotech to tech bio. But if it's branding that changes the work, and I like your new colours, thank you.
That's great.
For the green's gone, and I see that the orange is in. I think that can be important because it resets the mind and it enables people to see that there is new possibilities and a new way of doing this. The companies that will be built and the bits that will be bought by existing companies, by the J&J Innovations, by the AZs, by the Lilly's, all of whom are active here and will buy whether there's a recession or not because they need pipeline and new revenues, is some of this stuff. And I think that's where they're getting their innovation from. And we know that over half of the new marketed drugs that go through the FDA come out of the biotech ecosystem, not from the global pharmaceutical companies who are fantastic at distributing, and you can't do without them. We have 1 minute left.
Yeah, so Q&A, we've got time for a few questions. Does anyone have any questions for Steve? Yes, we've got one here.
Ciao.
On the left.
How long do you think it'll take the city to forget Neil Woodford and start investing in private again?
I think that the city will understand that the companies that Neil Woodford are in would have made them money today if they were still in them, like Immunicor. I think the animal spirits of wanting to make money is the key thing there. And if you look at the companies that are being that were invested in at the time, some of them are being very successful. You wouldn't expect all biotech companies to be successful, but there's enough in the portfolio from the olden days that people are now thinking, shucks, I wish that had stayed going, because if it had kept going, it would be making money today.
Any other questions? No? Well, thank you very much, Steve. Thanks for
Thanks,
joining
Beth.
us.
close as possible together to the light.
All right, I think we're ready to go. Thanks for bearing with us. You'll be relieved to know that it's my last panel, so you don't need to listen to me anymore. But very excited. We have such an exciting panel here today. We've got some really senior VCs from funds all over the world that have come in to join us today, and so we're very grateful. I just want to start with a very quick introduction from each of you and maybe a little bit about your fund.
Yeah. So pleased to meet you. I'm happy to be here. Thank you, Claire. Arthur Franken is my name. I'm general partner at Gilde Healthcare. Gilde Healthcare is now actively investing Fund 6, 740 million across Europe and the US, both in biotech, medtech, and health tech. Yeah, let's leave it at that.
Same, thanks. Very grateful to be here. Luc Dochet, managing partner at Droya. We're a therapeutics-only fund focused on genetic disease and oncology, so 2 specialist areas. And we have about, today 480 million under management.
Hi, L.A. Charles. I'm a venture partner at SR1. SR1 was first founded in 1985 as part of GSK, but now we're an independent fund. We spun out in 2020 with approximately 1.5 billion assets under management. We're on our second fund.
Great. Hi there, Neil White. I'm in the dark because I'm still in California time. I'm from a new fund called Yosemite, or Yosemite as you call it here. It's a spin-out fund from the health team at Emerson Collective, which is a family office. We focus on oncology, mainly in therapeutics and some digital health, and about $1 billion under management.
Great, thank you very much. So we've got lots of topics to go through today. I think, there'll be people in the audience who are scientists, clinicians, maybe thinking about what are the options, could I build a biotech company, do I have something that might be of interest to VCs? And I think it's important to sort of talk about the venture landscape and to recognize that actually all funds can be slightly different, and some of you have different areas of focus and different strategies. What would be your advice to people looking to raise money from VCs when they think about how to approach funds and how to look at different fund strategies and compare them?
Yeah, I can give it a go.
Yeah.
When I joined Gilde in 2001, we were really looking at an early biotech ecosystem in Europe. We ended up investing in the Series A of Uplinks. That was my first deal I was involved with. But now we're living in a complete matured European biotech ecosystem. So that has implications for young entrepreneurs, for people in the audience trying to navigate the routes. The good news is there's much more capital available. The other news is it's not bad news. You need to come to VCs with a much more, I would say, prepared and educated pitch. The field has matured. And I think Europe is now in a kind of acceleration mode in nurturing European biotechs. When I started with Uplinks, great outcome, but it took way too long from an investor's perspective to where it ended up. And I think the way we at Gilde have evolved our thinking about early stage opportunities, really Series A's, is how to get close to the speed with which we see biotechs evolve on the other side of the Atlantic. And we help with that. We make all kind of advisors available, but that's really central, I would say, Claire, to thinking about how to start up a company and do it fast with all the help you get from investors.
Mm-hmm. The only thing I would add is, of course, do your homework. So as we discussed, all funds are kind of the same and different. Yeah. So we're a therapeutics only fund. So if you want to do diagnostics or something else, don't come to me. that's probably a waste of your time and preparing your pitch. So, prepare yourself. Check, who's investing into what, what kind of deals do they do, what do they like. You have companies focused on single assets. So if you want to build a large platform play, probably not the right address to go to neither. So I would say do your homework, and second advice would be don't be shy. We're very approachable. You can get a no, that's okay. Most people get a no, but you can learn a lot from the no's. I was on the other side for probably more than 20 years. I definitely got much more no's than yeses, but ultimately it's only the yeses that counts. So don't be shy and do your homework would be the two simple sentences.
And Neil, your fund is a slightly different structure, doesn't it?
Yeah, I guess I'd say, there's varying degrees of involvement that investors take in supporting entrepreneurs and academics in translating their science. Our fund, we have started to do more of it, but company building and new co-creation. So we'll actually assist the academic in liaising with their tech transfer office, maybe even supporting a sponsored research agreement to do and replicate certain studies in the context of their own lab before licensing technology. So yeah, seed investors will get very involved, and then there's, Series A, B crossover funds. But I'd say typically our lens in the therapeutic side will look for things that have a dose-dependent efficacy and some clarity around the mechanism of action. That's kind of usually where we can plug in. But I guess for the earlier stage stuff, part of our model, as you alluded to, is around unearthing ideas that otherwise wouldn't have seen the light of day. In the US, we have the NIH and your R01 grants, and typically they, support incremental advancements of the science where, a researcher is already well known for something in the field. The currency in academia is publications, and you'll get a grant for something that you can easily execute on. And the exclusion criteria for the R01s is that if the science is too audacious, it won't get supported effectively. So we give these sort of philanthropic 200 to 300K grants to support this audacious science and see if it lends itself to the next stages of translation. And actually, that is kind of where our fund started, with the philanthropic side. And then it was only as some of the research came out and looked interesting that we then started to make investments. And actually, our very first grant, it was looking at T-cell exhaustion, and we found some targets you can kind of, or pathways you can overexpress, and that ended up becoming a cell therapy company, Lyell Immunopharma, and then it went public and basically returned many multiples of all the grants we'll ever get. So yeah, there's possibility, and there's different stages you can plug in with investors.
And Elliot, when you meet potential new founders, so people still working in labs and academia, sometimes we hear tech transfer offices saying, oh, they're not quite ready to speak to investors. They're not ready. What do you say to that?
I think I may be a little bit of an outlier, which emphasizes a point of diversity. So in doing your homework, which is we're very happy at SR1 to go early to folks. In fact, in some cases, I've read some publications. I've called up the academic person involved with that publication and just asked, are you interested in doing something here? My preference is sometimes to work with these founders and understand what the vision is for the company, where the potential is, and work with them together to go build something. So with tech transfer offices, they've been great in facilitating things, and in some cases work to put together the company. But really, we've spent, all of us here, our careers in this area and trying to understand what will make a robust company and how to get these ideas translated into medicines. And so I think including us as part of the process is a very good way to do it.
Mm-hmm. Do you agree?
Yeah, I agree. I would like to be clear to this audience. We are very busy. We have a huge deal flow. So if you really pitch immediately your, let's say, university spin-out or your TTO opportunity to us, the likelihood you will get full attention is not that high. But there are other ways to engage with the Guild of Healthcare. For example, we have a sizable team, and we take part of jury member panels and things like early TTO competitions and things like that. We always find it important to be there, and that is taking also a large chunk of our time. But really to pitch straight out of university to us the likelihood to get a lot of attention is low. But really think about other ways. I mean, my presence here today, right, is also a testimony of that. I don't expect today to find here immediately an investment opportunity to work on for the next two months and to structure a Series A. Perhaps I will. That would be a great outcome. But being here, talking to us, and for us, it's always a journey. Engaging with the company. Most of our portfolio companies we have today, I think, weren't immediately a fit, and we have been monitoring them over time and coaching them. For us at Gilde, it's very important, no matter how early you are, what therapeutic modality you're working on, to be able to talk about a target product profile, a product market combination. Have that in mind. If there is a physician founder involved, usually that is the case, but very often we lack that piece of the story.
Yeah. But for the really early research that people are maybe just wondering whether they think they might have something that's commercially interesting, Luke, what would your approach be for people like that?
Obviously for us, we're also a specialist investor, so we do oncology or kind of genetic disease, which means, we have teams going out a little bit like Elliot here, reading the publications, going to the specialist conferences, we do visit the posters, and then we start discussions, and then you quickly kind of feel whether somebody has a little bit the urge to maybe go one step further, and my team is kind of a bit trained to kind of listen to that and have those discussions. Obviously, as a specialist, it's easier because, you're networked, you're focused on one specific area, and it directly becomes clear. And a lot of these discussions often go nowhere or are the beginning of building a relationship. And so from having a first discussion at a poster to, maybe creating a company together, sometimes that many steps in between, and that takes a long time to get there. But I think, again, this fits into what we said at the beginning. It depends from fund to fund. So for us, it's more likely. We also do a lot of new co-creation. We have our own labs. So we have about 15 people in our own lab space in Leuven. So we even sometimes just ask you to reproduce, or give us the opportunity to reproduce some of your data in our labs, just to be triple sure that, there is something, there is a beginning, and then we get involved. And I think, and then the other thing is, of course, are you truly the entrepreneur that wants to go there? And there's nothing wrong in just passing on your ID and being involved as a scientific founder, but not by definition having to go in biotech. That's, again, a different mindset. So I think there's very, very different ways how to get there, but it always starts, just communicate. If you have an ID, you're very excited about an ID, don't stay with it. If you just stay with it and you just sit at yourself and you just try to publish, that's okay, that's a good first step, but it's not gonna get a drug to patients. And so if you're truly excited about that and you want to contribute to that, just talk to us.
And maybe just to add to that point, I mean, I guess we have access to, a lot of academics through our grants work and relationships we've cultivated, but I think to your point about, staying close to the literature, I mean, a lot of us are already under CDA with a lot of these labs and universities, and we're getting sent pre-print manuscripts and really, ahead of the publications, because by that point it's too late and it's going to be too competitive to, wrestle with these guys to get into the deal, right? So, I think it's never too early to engage with some of the seed, Series A, NewCo kind of company building funds and cultivate those relationships over time.
Yeah. I mean, I think it's hard sometimes for people maybe you're in a university that hasn't had high number of successfully commercialized companies, or maybe you don't have very robust tech transfer office. So we often get people saying this, I don't even know how to go about approaching people like you. I mean, other than, as you said, it's very difficult because you have very high deal flow. You have a lot of people coming to approach you. What would your advice be to somebody who's thinking, I don't even know where to start?
I think the tech transfer offices do have to play a role there, or other organizations, where you get your funding from. Even as an academic, I think getting your funding is also every time again a pitch process, whether it's a grant or something else. So I would say it's not that different. So don't be shy. Just reach out, try to talk to people, think it through a little bit.
Keep networking.
Just keep networking, and this is why it's great to see this big show up today.
Yeah.
I think it's a good sign that people are interested to kind of try to do that, to bridge that gap, and to go in towards the translation thing. And I think always, again, my advice is always look at it also from the patient point of view. The likelihood that your ID turns into a drug, there's more likelihood that it will fail than it will succeed. Making a drug is not easy. But if it's positive, make sure it has, a big impact on patients. And if you can show me that what you work on can be truly game and life-changing for a patient, I'll listen.
Yeah, I think people need to demonstrate a little entrepreneurial spirit, I think, when approaching this, and not think about just one specific channel to get your ideas heard. I think that's something that we look for in individuals too, with founders, academic founders, just their ability to go out there, identify the right groups to speak to, and also just recognize that when we build new companies, it requires a lot of effort, and we need to focus. And so that may mean we don't have the capacity even to look at several ideas at once. Now, we've got a great team of venture partners and investment professionals within the firm that allow us to prosecute several deals at a time, but we're not going to look at 100 ideas and move on 50. We're going to look at 100 ideas and maybe move on 3.
Don't you look for second-time CEOs, first-time round, every time again?
We do. we do everything to try and maximize the opportunities out there. And so your point is leveraging our networks. And we have a really strong network. We're both in Europe as well as the U.S. And with that long history, both from individuals as well as the firm, we know a lot of folks. And so if we see a great idea, we're very quickly able to match those ideas with the appropriate individuals and prosecute more things. So 3 is not the number; it's larger than 3. But I think you should set your expectations that it's typically venture capitalists look at thousands of opportunity in a given year and do a handful. So
I think
the hit rate is pretty low.
The only other point I was making, nobody gets born as a second-time successful entrepreneur. Everybody has to do it the first time. And so I think I'm also looking at it in the past. It was very hard. You're too young, you're too inexperienced. But I think I'm really past that. And I think a lot of my colleague VCs are past that because we just need to grow more talent. We need to give people an opportunity. So this entrepreneurial spirit, the passion, the willingness to take some risk yourself, but to really go for it is there. And more I give people the chance to be a first-time CEO. And we try to advise them, we try to coach them, because the amount of talent that goes around and does it a second and a third and a fourth time, if we just count on those people, we're not going to do a lot of companies anymore. So I think that's the other thing. Just don't be shy. We're willing to give people chances as well.
We like first-time CEOs actually, as long as they reflect the right features. Look, I got to know you when you were CBO at Prosenza, right?
Yep.
And your CEO was Hanshi Khan. And Hanshi Khan is still in the sector. He's still helping young entrepreneurs. So if young entrepreneurs, for example, in this audience, I'm sure that in the UK you have like 5-fold more of those people than probably in the Netherlands or Benelux. But people who've done it before, talk to them, who are kind of independent but know the VCs, open up, start networking. If you come in with like, I'm a young entrepreneur, I'm a physician scientist who wants to start this company and I'm being coached by, former CEO, then that resonates already. I mean, that really helps to get the attention of a busy VC. And that is, I think, Elliot, what you also mentioned, the entrepreneurial spirit, network, and be willing to be coached. Yeah.
I'll add, also on the philanthropic side, to Luke's point around, celebrating diversity of views and ideas, we'll also redact the applications, like remove the applicant's name and elements of their CV, so we're really focusing on the brilliance of the idea itself. And I guess the other point I wanted to make on the investing side is that, it's a continuum in terms of the degree to which the academic wants to be a founder and involved in the company, where you can be very hands-off and still have equity and economics into the company. And I'll say we have about 65 oncology companies, and maybe 80% of them are therapeutics. Our most successful therapeutics companies, it's actually one CEO. He's a CEO of two companies, both very good. Well, I mean, speaking about my own portfolio, I'd have to say they're very good, but it's Dragonfly Therapeutics and also Skyhawk Therapeutics. But this is Bill Haney. He's come from a real estate background, not a biotech scientist in any stretch of the imagination, but a brilliant individual, and has been able to, I guess, speak to the idea and ignite interest in investors, and also think strategically about the target product profile, addressable market opportunity, and this sort of thing. So yeah, I think individuals and CEOs come in all shapes and flavors, and there's no real pattern recognition as to what will make a winning one.
Well, it's a good thing as far as diversity goes. As you said, I really like that approach of redacting some of that sensitive information. We're sitting here in London, and I think a lot's been said already this morning about what the UK has to offer in terms of scientific resources, the ability that the institutions we have, the sort of knowledge base we have. Does it matter to any of you where a company is based?
To be honest, for me, no. We're a global investor, so I think we go where the best company is. I think also, we do see it being a bit more of a virtual world where, location is just a thing. We talk to anybody. I got more companies in the US than I have in Europe. That's more driven by, I think, still people, entrepreneurial kind of spirit, and a little bit talent. People that have done it, that are willing to do it, willing to take more risk there. But other than that, location is kind of completely agnostic for us. I've run a company here in the UK, kept my family in Belgium, was commuting, to Stevenage. I couldn't find enough arguments to convince my family to move to Stevenage, which I know is hard, but nevertheless, that's where I failed.
Yeah.
But I think, we're getting there. It's really, yeah, to me, it doesn't really matter. And even there, not the whole team has to be on site every day again, because if you want to build a strong team with the right skill sets. So I have companies where, I have my CMO living in Switzerland with the right skill set, the company, being based in Leiden, where the lab is, where we have a strong operational team. But the CEO actually lives in the US. And so, they meet quite often, but it works. And that was something that was unthinkable. How many times I was asked in the past, if you want to be in this company, you got to move to Boston.
Yeah.
Yeah, I'd agree. So I was running NeuroBio a few years ago. This is a company that was technology based out of Oxford University, and everything was right in Oxford, which was nice. The current company, the technology comes from Memorial Sloan Kettering and Fred Hutch, so New York and Seattle. We have our labs in Boston, and we've got a number of people, including myself, here in London. I'd say it means a few more late nights to hit the West Coast. But there's some advantages, I think, in being able to be in different geographies. But most importantly, we're just interested in finding the best people and the best science to make medicines. And that really transcends any kind of location. And we've got a good footprint at SR1. We're spread out from Europe all the way to the West Coast that allows us to do it as well.
But I do have to say that London has a pro within Gilda. I mean, that for us represents an ecosystem within Europe of, a local talent pool. So for us, if we see an opportunity coming out of London and another close competitor located in Athens, in Greece, then probably it will be a topic of discussion. We prefer the one in London, probably, because it's just, it looks like it has access to a better network. So, location is important, but I also agree with Luke and Elliot. We're living in times where there's more flexibility around that. Yeah.
I wonder if sometimes people confuse the issue of the location of a company with the ability of that company to raise money in the US. And what you're saying is that's not at all the case because you're all global investors, right? So,
and I can speak from experience. You know, in 2013 we did the first Nasdaq IPO for a European-based company with Prosenza. So we were kind of laughed out of the room when we talked about it. You know, we had only operations in Leiden, and so we were doing our primary listing on Nasdaq. I wish I could have taken a patent on that one, because after us, many followed suit. So where your physical presence wasn't important. It did mean we had to travel a lot. We had to be available and a lot of late nights, because you got to be up and you got to be available to the investors, you know, in their time zone. But it was not a limitation. I think this was a start of something that we see now, that, you know, you don't have to be based in the U.S. to, you know, to raise money from U.S. investors, certainly not from the public markets. On the other end, of course, you know, it shows that you have to be quite differentiated and be unique to convince them, because if they find something equally in Boston next door, you know, you have a tough time. It's the same as London, Athens. I think for a European company, you got to show your competitive advantage over the U.S. one if you want to attract mainly U.S. investors. But other than that, I don't see an issue with it, to be honest.
I'd agree. So I was a biotech investor based in the Bay Area until I moved here in 2012. I'd say in 2012, it was a big wake-up call to see the differences between the two geographies. But now it's great. There's lots of capital. There's plenty of U.S. investors that look overseas for investment opportunities too. I think companies here, including the ones that I work with, still need to think very heavily about the U.S. because there's roughly 10 times as much capital there and some really great people that you can access to. But certainly there's a strong appetite from a number of good groups that are based in the U.S. Who will make investments over here.
And I see that as the role of a Gilde Healthcare, although we focus Biotech on Europe. From the moment we do have an office in Boston, but that team focuses on MedTech and HealthTech predominantly. We might go there one day, one time with a local deal maker, but for today we're sourcing European biotech opportunities. From day one we get involved, we see whether there's a need to think transatlantic and global immediately. And if there's a need, they see it already emerging in our due diligence process. Like we have all the tools, the networks to pick up competitors everywhere. And from the day we are involved, it's important for any young biotech company to look at global competition and also the global opportunity to tap talent out of whatever pool. So it's our role as an investor.
I guess the only point to add is around, you know, to really ensure the future financeability of your company. It is, I mean, not all investors are agnostic to geography like we might appear to be. So it's, the earlier you can in your syndicate, it's useful to have investors perhaps in the US or in Europe that can then help socialize your technology and, you know, maybe lead in the next round of financing. So, I mean, the earlier you can protect for that, I guess, the better, obviously.
I guess the reason I think this is a really important point, and I take your point that not all investors are as flexible, but is that, you know, very often here we hear a lot of talk around, well, in order for companies to grow, they need to leave the UK because they need to go to the US. That's where the people are. That's where the money is. And I think what I'm hearing is that actually, if that company is doing something which is globally competitive, you can make that work regardless of whether they're in London or Manchester or wherever.
I think it also depends a little bit on the stage, but certainly in the beginning, you know, being close to the people that really know the underlying science and the biology is and remains key. So I rather have the company close to a couple of its academic founders and keep them involved rather than move them for, you know, with all respect for everybody, for a manager, you know, to the US. And I think it's such a complex business that we're in, you know, things will happen along the way. You know, you will do your preclinical work, you will see crazy things happen which you didn't anticipate, and then, you know, having to understand and having the ability to go back to the scientific founders, you know, get their deep insights, I think is important. So most of the companies we invest in, at least a lot of the R&D operations are still very close to the mothership, I would say, whether that's the founding academic institute or you know, an individual that can depend. But I think that's to me more important.
That's the most important thing.
Yeah.
Neil, you agree?
Yeah, just to say, I think historically, there's been this gap between, late stage investment in the UK and kind of early innovation, the gap in the, available managerial talent, large pharma kind of BD type folks, but I think now you're kind of seeing that dissipate around modalities where you've had great British successes, like in the T-cell engager space with Immunocore, et cetera, and now like we're seeing a bunch of BiTEs companies in the UK that are popping up with ex-founders, et cetera. And I think now also with the Ascend gene and cell therapy incubator, sort of multi-asset company from Monograph Capital. So good examples where we're kind of fleshing out that next tier of sort of managerial capacity for some of these innovations.
Yeah, absolutely. Yeah. So, I mean, you touched on the actual act of translation. Obviously, we're here talking about translational science, and I think because we focus at LifeAct so early in that translational journey, we often feel very far away from the patient, especially on the grant side of the work that we do. We're funding very early preclinical work just coming out of academia. And as a VC, you need to make sure that what you're funding gets to a patient, because that's how you make money, or you make money along the translational journey. I wonder if you can talk a little bit about some of the things that you found in dealing with preclinical translation. What are some of the big issues that you deal with as investors, and how do you protect yourself and your financial exposure in that such a risky area?
Yeah, it's a very good question, and I'm actually now looking at two regenerative medicine approaches and super cool biology. All experts we involve are super excited, trying to translate that into a clinical development plan with timelines and patient-relevant outcomes. Something you regenerate a tissue that is damaged in a disease context. That's very hard. While when we're doing our job, talking to pharma, we're a VC, what are you looking for us to de-risk, for you to acquire? Then they basically say disease modification, right? Disease modification. Symptom relief, okay, then nice, right? If you're first in class or something, but disease modification. And translating preclinical regenerative medicine approaches into white spaces, pioneering clinical development spaces, I find very difficult as an investor to understand. So the way to get me engaged is to acknowledge the challenge and kind of, bring me along that journey. And that is speak the same language. And as long as you acknowledge that and take perhaps smaller steps rather than jump straight into a phase 2, that's a way to engage investors like me in that journey. But the translational challenge is definitely there, particularly in those things.
I would look at it, I try to not stack the challenges on top of each other. So if you have and a novel therapeutic modality and a novel, biology to understand and novel endpoints to be developed, that might become a bit challenging. Yeah. So if that would be the case, I try to then look at it, how do I limit this? And so if I have the option, it doesn't sound really sexy to still go for a small molecule or an antibody, over gene therapy or gene editing, which is the new thing, I might kind of go for that if I can do it, but at least I understand what happens there. I know how to upscale. I know how to deal with the CMC. If it's in a novel biology, of course, or with novel endpoints, then you try to engage the clinical experts as soon as possible. So even when I was still the CEO, I always hired my CMO literally 2.5 years, 3 years before we would be in the clinic to kind of get the clinical perspective, to think about regulatory strategy, early on. Whereas in the past, a lot of my investors in those, they say, look, why would you spend money on a CMO now? And I think so it's for me really trying to uncouple the different things and don't stack them on top of each other and try to kind of focus on one problem at a time. And so if it's too complex, maybe, I'll stay away. So if it's too much, which sounds probably very appealing, sometimes that scares me.
Yeah.
But that's just a personal view.
Yeah, as investors, we're trying to manage several different types of risks. So there's the technical risk, which I'll talk about a little bit more, but there's also financing risk, commercial risk, execution risk, which is the management team, development strategy risk as well. So many different things to think about, but just as we think about the pure technologies, I agree with Luke. Taking on what I call compounded technical risk, where 5 different things have to work before you even have a chance of making a medicine. It's a challenging way to go, but sometimes you can mitigate these through strategies. So for example, if you have a really robust technology that would have application to some new targets, you can still get there and get to those, but it may be the case when building the plan together, we would focus with the team on thinking about first demonstrating the power of the technology, maybe with some targets that are well established in the industry that may have clinical validation, and then later thinking about those new and interesting targets.
Yeah, I think that's the divide between risk associated with a platform opportunity that can lend itself to different products, et cetera, or investing in a single asset or a single program. I think in the latter case, and we have a lot of single assets coming out of our grant-supported work, we invested in a company called Cullinan Oncology based out of Boston, and then they'll basically act as a sort of multi-asset holdco with lots of subsidiaries, each with different sort of orthogonal risk profiles, different mechanisms of action. It's focused on small molecules and biologics, but actually the way we license technologies into these subcos is first, the licensing terms are then dependent on our ability to replicate the data from the academic lab in a third party or by a third party CRO. So I think on the single asset kind of opportunity profile, we'll really make sure to mitigate these translational risks by replicating the studies. But I think on the technology platform side, then it's really a sense of, do you have the relevant models that can sort of substantiate, the translatability of the technology? I mean, we've spent a lot of time in platforms around, allogeneic cell therapy and hypoimmune cell therapies, but then it comes down to, do we even have the right kind of syngeneic models or NHPs? How representative are they of these sort of technologies? So I think from the platform risk point, it's good to sort of, appreciate the amount of capital, the intensity that's required to get these things validated and into clinic and to a point where they're appealing to investors. Yeah.
But this is also building experience on the therapeutic modality. I think Arthur will smile when I say monkeys and azos don't get along. It's certainly something where, I worked a lot on antisense oligonucleotides and just, you still have to do the large animal studies, but if you kind of take the learnings from an antibody or a small molecule in an antisense and, try to translate the findings in monkeys, to be honest, there's never an azo going to get to market. But it's really understanding the modality, building the experience, making sure you have access to that. So also understanding what modality, how to read into that is kind of very important. And so I think it's really, this is also where some of our experience can be helpful for a company to listen to that. Because
absolutely, yeah.
I've seen programs that would have been shut down that ultimately led to great drugs. That really help patients, if you would not have had the right team around it to really understand these preclinical findings. And I think we've and the same goes in clinical studies. It's always a question, is it the drug that fails the trial, or is it the trial that fails the drug? And I think there also we kind of made too many mistakes by using the old paradigms of pharmaceutical development, which has to be large numbers, the p-value is the only thing that counts. If you just make it over, it's great. If you make it under, you're out, which is, if you think about it logically, is doesn't make any sense. So I think this is also something we're advancing in, we're learning a lot, but that we have to keep doing. It's really understanding this. Everybody is different. I come out of a more rare disease background, and there's already not a lot of patients, so the large studies are not possible. But also, if you really look at it, the interpatient variability is huge. I worked in Duchenne, and if you look in Duchenne, a 10-year-old with Duchenne, they're not all the same. Trust me. Their background, everything is so different. So if you then have to do a study with a limited number of patients with a lot of variability, and you apply the old standards to that, it's wrong. So I think also there, our learnings from what we've seen, helping the companies with that is very important. Just to give you a couple of these translational challenges that we face, where we learn a lot, and that we have to kind of work very closely together with the company so that not too many things go wrong.
And on this particular example of rare diseases, it's just one example of a translational tool. Phase 0 studies, patient registry studies, patient history studies are for an investor very interesting because you kind of follow the course of the disease. They might be established already. If not, it's worthwhile seriously considering, because that will tell you much more about the heterogeneity and set you up for a good phase 1, 2. So that's just one example of a good translational tool.
So there's so many elements to risk mitigation. You talked about actual scientific risk, there's operational risk, there's sort of financing risk, and so as VCs you have to balance all of those things, and you also have your own investors that you're trying to keep happy, and just as a field the attrition rates are quite high. So you quite often see people coming up with new structures, trying to de-risk, whether that's sort of a bridge bio type structure or other structures where you're trying to get more people at sort of top co level, and then you mentioned putting assets in sort of SPVs or other vehicles. Do you think that that's really could be the solution, or is it just a different way of trying to spread financial risk? And actually, at the end of the day, you still really have to deliver the science.
Yeah, we've got a good case in point now working through where, serial entrepreneur, many successes, companies well know, and what's becoming in vogue now in the U.S. is founders really realizing their worth, and it's a great thing at the same time, right? We want to reward people for their innovation and their ideas, and flipping the conversation where they're not risking losing their equity into subsequent financing rounds into the IPO. So putting in terms that non-dilutively sort of protect their ownership. And what we're trying to do to mitigate that is thinking through the other ask from the founder, in that, I want this large amount of capital up front. And actually, I like some of the arguments where, one of the ways we mitigate this finance risk, or how the capital is deployed, is to put very detailed tranched structures in place where, the team has to meet certain milestones before they unlock the next tranche of capital. But I think what that often does is it really hinders creativity, where, you're sort of drip feed piecemeal the capital available to the researchers, and everything is sort of shaped to meet this next milestone. But actually what we found is that sometimes you'll have discovery along the way, and new areas of science are opened up that actually you should be double clicking on allocating resources towards. So I think it's a matter of rewarding and incentivizing teams the right way, retaining the talent, and making sure that they're sufficiently capitalized to unearth and to prosecute these ideas.
I think there's a lot of innovation going on in different places to try and do better. So, a second ago we were talking about translation, and there's some really nice innovations in terms of clinical trial design with adaptive trials and things like this. From a finance perspective, I think there's also some innovation. We're playing with some new structures at SR1, thinking about implementing in the near term. But fundamentally, we're all judged by the clinical data we create and whether we can create medicines. And so I think there's really no hiding from that risk. We don't want to hide from it. This is our business, is to understand, try to optimize our chances for success. But in the end, if we're not making medicines, we're not successful.
Try to fail early if you fail. I mean, that's something that resonates with me, that it can be done via tranching. So for us, we have a different way of looking at risk when we want to invest in a single product opportunity. Then we have a very exit-centric approach. Then we are convinced that this asset somewhere along its journey will be acquired. That's the outcome of our due diligence. But we also have a strong preference to invest in companies. And then the companies need to have all kind of opportunities to pivot when and if needed. So when we start leading rounds for company financing, Series A, we we tried to raise significantly larger rounds than a couple of years before, pre-syndicating, trying to skip a Series B and bring ideally 1 or 2 US investors already in, really to turbo boost that company, but also enable it to deal with failure.
Yeah, you do. I mean, that is something that I think the UK struggled with, is just sort of drip feeding that funding in, and as a result, not being able to pivot if something does go wrong.
But what Elliot said is at the end of the day the most important. We're out there to make medicines, and that's what kind of makes or breaks, whether we're a successful fund or whether we're a successful company, yes or no. I had the luxury and the luck to work closely with Henry Termeer, who was the late CEO of Genzyme, and he always said, if it's good for the patients, it's ultimately good for the company, and then it's also going to be good for shareholders. So if we can help patients, we'll find out a way to structure it in such a way that it turns a success. I've never known, a company that made a drug not to be financially successful.
And even in the case of ultra rare disease,
look at Ultragenics, and they made a business model out of it. It works, and so it's a different model. But then it also boils down to the impact. We had a huge discussion in Belgium about Zolgensma. Zolgensma was the SMA drug, terrible disease, and so it wasn't a fair discussion. It was a public discussion. But parents with a kid, and I understand, I'm a parent, I would do anything for my kid, went to the media, we can't get access to the drug. But one, the drug wasn't approved, and there was a production issue. So it wasn't about the drug being, costing $2 million, but that's what they used. But then there was a public upheaval. How can anybody ask, $2 million for a drug? And then I went back and I actually spoke to a journalist. I said, just do the maths. The cost to let that kid die to the Belgian system was about, $2 million in terms of they were spending about 6 months of their 2 years that they live, in the emergency room. The other months, they needed special care, they needed physical therapy, a lot of, medication, symptom relief medication. So the total cost to let the kid die was $2 million versus $2 million, for a drug, and that was $2 million because the cost of production was still so high. So I think it's also important to show that to the world. But it shows that if you can help a patient, there is a model to be made, even in these ultra-rare cases.
Yeah, and Neil, I know you do a lot of work in oncology. I mean, pediatric oncology, you heard this morning, is a big area for us. What do you think about the dynamics there?
Yeah, I think similar, we look at oncology broadly, and I mean, on the importance of pivoting. Everyone's pivoting to autoimmune now, given recent in cell therapy. But in indications like pediatric ones, but also in things like pancreatic cancer and brain tumors, et cetera, I mean, these are like the highest hanging fruit. And the thinking is that, in pediatrics, for instance, and also GBM, et cetera, this, immunologically cold environments, if you can find ways to modulate it, then you'll have, much broader market opportunity in other indications or different patient groups. But it's obviously a challenging space. I mean, a lot of investors, they see these things and they're totally risk averse. And I think that's kind of where we have a tool agnostic approach to how we can deploy capital, whether it's, philanthropically supporting some of this work, getting it to a stage where it can be, licensed into a company, or, I mean, there's also success stories in the pediatric oncology kind of focused area. This Day One Pharmaceuticals is a similar kind of multi-asset structure we talked about. It's gone public and also focused on pediatrics. So, yeah, I think it's finding the broader applications for your technology beyond pediatrics is one thing, but obviously there's unique challenges, and we just need to find the mechanisms to plug into to work towards them.
I must also add a sense of reality here. I mean, we have LPs, and they want to see big returns for our investments. So we always try to be smart about where M&A is gravitating towards. And M&A is not only big pharma, but we do pay attention to big pharma M&A. There's now, with the maturing European ecosystem, also mid-size and also biotech M&A. But when you look at pharma M&A, right now the trend is, and that's just the reality, due to the IRA, Inflation Reduction Act, and the length of exclusivity now, 2026 and beyond, trying to fill the gaps of Keytruda. it's big markets, big opportunities. It's not small markets anymore. So that's the pharma's thinking. And all the M&A that you see is phase 2 plus data. And then they're not shy of giving you $2 billion upfront, $3 billion upfront for that. But that's the reality right now. So that, I think, is also important to mention here.
Yeah, so I was on the buy side for some of these bigger companies, and what you're saying is absolutely true. There are trends. If you're thinking about starting a new company, you're pretty early in the process, so you have to try and forecast years out, which is usually pretty difficult. The way I think about the markets when you're first starting a company is, are you addressing a fundamental unmet medical need, and will you provide a big benefit to patients? And that's great because that's what I want to do in my career. And I think if you use that as a starting point, understanding what other drugs are in development, you usually get to a place where you can find a good-sized market that's very investable for both your partners within the firm as well as your LPs.
And also there's regulatory mechanisms to be aware of around, pediatric designations and priority review vouchers, et cetera. And these things can be worth hundreds of millions of dollars. So, at the outset, having the view as to how you can get there to warrant, receiving one of these vouchers, et cetera, or orphan designations, that'll be the way to set you up for success for some of these harder to address market opportunities.
Thank you, yeah. We've got a few minutes left, and we've got such a great panel, so I'm sure some people have some questions. Please do raise your hand if you'd like to ask anything that's been on your mind about VCs. There we go, one.
You all spoke about entrepreneurialism, and can I just ask you perhaps to reflect on what you've observed as international investors between entrepreneurialism in the US versus the UK and Europe?
Good question.
Yeah, I think my first comment would be, there's more drive in the US to do it a second time. I think that to me is a big difference. If I see people in the UK, I see people in Europe, they had a big success and they say now I'm done with it and I'll be an advisor or I'll be an investor, something like that. That's how they turn to. Whereas in the US, I've seen many people, who had huge successes, who made enormous amounts of money, put drugs on the market, and I see them find the energy to kind of do it again and do it again and do it again. And I'm sometimes really impressed by those people. But that's to me a cultural difference that, for us one time, we're like, yes, we've done it and we're done. And there I see people kind of, we've done it once, let's do it again, let's do it again, let's do it again. So that's to me a huge cultural difference that I noticed. But
yeah, I think there's a big cultural difference between the two. There's also an experiential difference too. And so, having been in both markets, I just sort of reflect on a time where I approached somebody at Stanford and you said, well, that's great. How soon can you give me a term sheet? Because I'm getting 3 on Monday and, here's my IP. I've got this all lined up. And they're very knowledgeable about how the industry aspects works despite being in academia, whereas over here, I've found folks that have equally great science and they're completely new to the system. So look, that's I think that's a generalization, and clearly I found great people in the US too that were brand new to this, and it's really fun to work with those folks. So what I would suggest is just stay true to who you are and what you know, and if you've got an appetite to learn, you can get there.
And what I'm observing, and I think it's really promising for the life sciences ecosystem in the UK, is the willingness of investors to look at some first-time founders or people that have just come straight out of their graduate studies. I think there's a good program at Cambridge, a bioenterprise sort of master's program, and Oxford Sciences Enterprises, they are also putting a lot of first-time CEOs in places. This Theolytics is one company. There's also this Charm Therapeutics here, a new young founder, and that was something you see a lot more of in the US relative to the UK proportionally. But I think that's something that's changing, and we're very excited about backing.
Any other questions? We can take another question. Yes, right there.
Can you say a bit about your current perspective on platform versus specific program?
Yeah, I can give it a go. We're seeing a lot of growth in a variety of therapeutic modalities. There's no shortage of that. We started out, building our track record, investing in the first generation, kind of second generation antibodies and things like that. Those models worked back then. They were able to strike a lot of pharma licensing deals, technology access licensing deals. I would say today we have a different look at it. The respective technology platform must have a very clear reason to win, some kind of unfair competitive advantage with a clear patient application. And that's the new way of looking at it from our point of view.
Yeah, I think in this financing environment, where interest rates have gone up and capital is generally moving away from the biotech sector, it's definitely moved towards an asset-centric environment, and people discover it's just very expensive to fully fledge a platform technology. In cancer, just briefly, I mean, people will start with.
I think we're at the change.
There you go.
It's not a reflection on what you're saying. But Judith Edition will.
I mean,
yeah.
that's
okay, all right.
Whatever.
We're definitely at time's up, but thank you so much, everyone, for joining us. Thank you. the rest, yeah.
Right, can you hear me? Yep. Is the mic working? I don't think the mic's working. Is my mic working? Hello, hello, hello. Is mic working? Good. Right. So, good morning, everyone. Thank you for joining our next session. As you can see, this is probably the most popular track, and today we're taking you through the full story of the policy agenda through the lens of the BIA. We've heard from a number of esteemed international investors, and now the moment you've been waiting for, when we actually hear where the rubber hits the road, the CEOs and the founders of the brave challenges they take on, and actually just to get a bit more understanding of that story, what it takes to actually put yourself out there, start up a company, raise money, manage a team, work towards an exciting exit, and really, really very delighted to have Hakeem, Natalie, and Mike join us this morning. My name is Imran Hamid. I'm one of the senior investors at LifeArc Ventures. It's a pleasure to have you all here. And Hakeem, Natalie, Mike, if you'd like to introduce yourselves and tell us a little bit more about what you're doing.
Can we start?
Yep, yes, please.
So hi, everyone. I'm Hakim. I'm CEO and co-founder of a company called Closed Loop Medicine. And we as a company are solving a 500-year-old problem that was originally coined by Paracelsus, the father of toxicology, which is only the dose makes the poison. Everything we put into our body can either be helpful or harmful, and that is defined by the dose. So we're using software, and specifically software as a medical device, to optimize the dose of a medicine to an individual to maximize the therapeutic benefit that that patient receives.
Right. Hello, everybody. I'm Natalie Mount, and great pleasure to be here today. So I'm currently a venture partner at Abingworth, which is a venture life science investing fund. But I guess in this context of this panel, I'm more talking about my experiences as a CEO. And I was founder CEO of a company called Adaptate Biotherapeutics. We were a gamma delta cell engager company. I took that company from founding through to a successful acquisition by Takeda Pharmaceuticals in 2022. So I've been through that life cycle, and as I think one of the previous panel members was saying, people who were in the room then was saying, well, people do that, and then in the UK and Europe they never do it again. Well, I'm afraid I'm one of those people who can't get enough. So now coming round for another round with some new stealth mode ideas for new companies.
Good morning, my name's Mike Westby. My background, after doing a postdoc at St George's, I went into the pharma industry as a virologist, worked at Roche and then at Pfizer, where I met Natalie and some of my other ex-colleagues in the room. And then when Pfizer exited research in Sandwich, in Kent, 12 years ago, I then sort of went into the biotech sector, where I've been ever since. So I've set up 4 companies. So in respect to some of the previous speakers about they go for first-time CEOs, well, I'm guess living proof that you can go again and get some money. The current company that I co-founded with our CSO, Paul, who I've just seen come in the room, it's called RQ Biotech. We were formed out of the COVID pandemic when the BIA brought together a group of scientists developing monoclonal antibodies for vulnerable patients. We did our first licensing deal with AZ, which gave us a large upfront and enabled us to build out our company. So we're now working in 3 different disease areas, and we have a lab based at White Space in London.
Great, thank you all. So it'd be interesting to reflect on the discussion we had previously with the investors talking about how they invest. And given that you're all CEOs and founders of companies and had extensive experience in that, I'd love to understand what was that decision point that resonated with you personally to start up a company, and what was your experience thereafter in terms of fundraising? How much of
what they said previously rings true, and perhaps what are the experiences you had that might challenge people's thinking on how easy it is to fundraise?
So I'll kick off. So we weren't a spin-out, we're a start-up, so it didn't come out of an academic lab. And a couple of us had got together and identified an opportunity for bringing different modalities of care, software, drug, and device together. And I'd always wanted to start a company and was looking for the right team to get around me because I knew I couldn't do it alone. And I'll never forget, I was chatting with someone who many of you in the room may know, Dr. Andy Richards, who helped us found the company way back when. And it was at a London Stock Exchange event. And I was sort of talking to him about the frustrations I had for bringing this idea to reality. And he said something that has always stuck with me. He said, Hakim, you sound frustrated. I said, yeah, I am. He said, are you frustrated enough to start a company? And that was that moment of, actually, yes, I am. And that's where we went away and we incorporated and started moving that company forward. And then gone on a journey of getting seed investment and follow-on investment from funds like LifeArc to building out the company to where it is today. But I think there are so many steps on that journey from just deciding to start and then deciding to actually really throw yourself into the company. We were very fortunate in that the other co-founders I built the company with all had other roles that allowed us an element of flexibility to start building the idea outside the companies that we were already working in, to a point where we made a decision where we had to then jump in. It got to the point where if you were going to commit, you had to commit with your whole self, not at the hours before 7 a.m. or in the evenings. You really had to jump in. And so those are the two really defining moments, the moment to decide to do it and then that moment afterwards to jump in. Now, I know many people starting won't have that same privilege of being able to make the choose and build up the evidence to make yourself confident to jump in. But those are the 2 points that allowed me to start the journey.
Thank you. Natalie, your experiences, how do they match to that?
So a bit different. So when we started our Gamma Delta companies, actually we had 2 Gamma Delta companies, Gamma Delta Therapeutics, which is a cell therapy company, and Adaptate Biotherapeutics, which was the engager antibody-based company, but both of them target on gamma delta cells. So they're in some ways a bit more traditional starting in terms of this was spin out of science that had been started at King's Crick, funded by CR UK. So very much came from an academic founders. And then I was recruited as part of the founding management team to actually take that technology forward and grow what became the 2 companies. So our kind of journey for raising investment into that is, I think anyone who's been there and done it will tell you fundraising is really hard. And I think at the end of the day you look back and people who have done it talk about how much they raised and how amazing it was and how pleased they are. But that journey is a really tough journey. And I think when you go into it the first time, when you're excited about your idea and you're all full of all your plans and amazing things, then the first person says, no, we're not interested. That's really, that's something you've got to develop the resilience to and take the learnings from it, because, people don't invest in things because it's not the right time for them. It's not the right fit with their portfolio. They do think they've got things you think they need you to improve on. But you've got to take those learnings and not think that means that because one person said no, that your idea isn't good and isn't going to go on to be successful. So I think that to me was a really important part of the journey. Everybody has to do that. Nobody went out and fundraised and got yes from everybody they met. That's never happened. Everybody has that, and I think you learn from that and you improve your pitch and you go from there. So our fundraising journey at those companies took an unusual turn as well, because Abingworth had seed invested in the companies, but then during our pitches we met with Takeda, who then became really interested in the company and decided that instead of making just a straight investment, they actually wanted to invest in what was a build-to-buy model. So they put equity investment into the company, but also a lot of non-dilutive investment into the company in exchange for the option to purchase the company at a future date. So that kind of changed our whole dynamic and the whole kind of path of our future, which years down the line did lead to a successful acquisition and a great exit for both companies. but meant really then, I guess, over the next 5 years, we worked extremely closely with the Takeda organisation. So a very different, I guess, funding path in the end from a traditional starting point to what became, for us was a great journey and a great outcome in the end.
Thank you. And Mike, your experiences, how do they compare to perhaps what Hakeem and Natalie have gone through?
Yeah, a lot of similarities. I guess one of the things that I learned was you can get money from lots of different places and none of it is free. So people talk about non-dilutive funding, but that sort of holds you to doing exactly what you say on the grant for very good reasons. But definitely looking at the range of investment, understanding that different investment has different goals. I think we heard in the last session really well some very different views. There was a lot of agreement, but also those investors invest for different reasons and in different ways. And spending time understanding what those investors are investing in and why they've got to meet, they have to meet particular goals for their fund. And so they will have a time when they've got to exit. So someone investing in you when they've got quite a short time to exit really will want you to achieve your near-term goals because they've got to get out. Whereas a fund that's just starting up will have a longer lifetime. When I started, I actually went to a family office and I didn't even know they existed when I left Pfizer. Pfizer just had a big pot of money. It pumped into research every year and you got on and did your goals. And then I went into Mayfair, into a private office where my pitches were to the rich and famous sometimes. And at one point you'd go to a VC with a wonderful presentation and you'd be pitching, you'd be asked all sorts of technical questions. And then I remember one instance, I went with my chair at the time to meet with another family office, and the conversation I had all my slides ready, I had my narrative, as the Americans say, was absolutely nailed down at that point because I'd been told no from about 40 people already, so I knew exactly what to say. We went in the room, we sat down, the person on the other side of the table looked at my chair, said, "So, have you invested in this?" And he said, yes, I've put some of my money in. And she said, "Oh, great, I think we will. Drinks, anyone?" And I was like, so confused. I mean, literally did not know what was going on. Had a drink. I can't remember what it was. Got in the lift, came down, and I said, "Clive, what just happened?" And he said, "We just got £1 million." And it was so wild. But what I learned quite early was you'll find a lot of investors out there that will invest in you or your idea, and all they care about is the person. Yeah. And you heard the VCs talk about we want to be near the founders, so they might not want them to manage the company, but it's a really important part of investing that we all do in all of our in all our personal lives. You believe in a story, you hope it's credible, but it's very much driven by the person that tells you that story. So I think my learning was just don't take it personally because you will be turned down. And one of the things I learned with VCs that I think came up in the last session is quite often they will come back if they like the idea, but it's not right now. Or they'll come because somebody else is going to lead the round and then they'll come back. So it's really important that when you tell them this is what we're doing, if you see them again and have done something and tell them, that has a huge impact on them because, again, that builds trust. So the one thing I've learned is investors are super smart. They do remember you more often than not, and they will remember what you told them. And if you haven't done what you've told them, then they'll be quite quick to pick up on it. But the flip side is if you have, you'll be that further along the sort of trust journey with them. And then when the time's right, they'll already have a bit of experience from you.
Yeah, I think it's a really important point that on that previous panel talking about learning from the no's, because there are the no's which it's not a fit. You're doing a therapeutics, they're a digital health fund, or vice versa. There's not a fit there. But then there's the no's which are the not right nows. Yeah. and looking at seeing and watching your development. So we had a no from one fund, but it twice, but then they did invest eventually because of exactly what you've just said, which was we said, we're going to do the following few things. Can we come back and tell you when we've done them? And we went back and told them when we weren't even raising money to say, we said we're going to do this, we've now done it, now we're going to do this, I'll come back when we've done that. And they can start to follow a story, and that element of de-risking, that when you say you're going to do something, you do it, or there's a reason for the pivot. And so one of the big learnings is not only to really understand what the no means, to ignore it sometimes. And there have been funds that have said no, and I've written back to them and said, with the greatest respect, I think you're wrong, and I'm going to come back and prove why. And I've done that, and then got the follow-on meetings and got the investment. But you've got to understand and not, as you say, take the no personally. It's the nuance of the no, and to keep going through and listen to it again and keep learning. As you said, when you'd had 40 of those, your narrative was nailed down.
Is there something or a type of phenotype where someone who's out there constantly hearing no still bounces back and springs back? What inside you kind of keeps you going? Because a lot of people would just kind of give up after the 10th, 15th, 20th no. What's the tip or trick for perhaps a young scientist or a budding entrepreneur out there to sort of hold on to that keeps you going? How do you maintain that motivation?
So I think you have to listen to the feedback. I mean, say there may be good reasons. There usually are. These people, investors are not. They're pretty smart people. They've seen a lot of companies. They've got a lot of experience. So I think that particularly if you're at an early stage with a company, one of the great things about being early stage is that you can actually pivot. If you really think the feedback, you've got to change something about what you do. You can either change the way you message it, which may be actually what you're doing wrong, or it may be that actually fundamentally, you need to change, what you were planning to do. So I think, listening to that feedback's, really important, because then I think you're not, if you're going out every day doing the same thing, the same presentation, the same people getting the same message, then you might be quite determined to keep going, but actually at some point you've got to think no. And I think after every presentation, we would sit down and go, okay, well that worked, well that didn't work, so let's amend how we're explaining things. Let's think about that question. Let's build on it. So I think you kind of have to see it as you need to go in with your eyes open. This is going to happen, and make sure that you kind of you've got a plan. You're evolving as well. You're learning as well. And that helps you to keep going.
So I guess rather than seeing it as a binary outcome, look at it as a process to refine and your narrative and your strategy and the problem.
I think no's as well are not the worst actually. I think the maybes are much worse. A no, you can, if people are honest enough to tell you why it's a no, you can learn if you're willing to listen. All the time that you're trying to juggle balls and it's a maybe, particularly when you've got your first investment and you're looking for a follow-on investment, it's exhausting going round, catching up and following up with people. So I think people who will give you time and will then say, do you know what, no, it's not, it really isn't. It isn't the worst thing. It's trying to turn a into a yes that's exhausting, because all the time that it's not a no, you've got this horrible thing. One of the things we may come on to talk about is most of the time you're just running out of money. It doesn't matter whether you raise 80 million, 8 million or 800,000. Time is your enemy, and you're running out of money. And so you've got to have this reasonable expectation. So you don't want nothing, but you really want to find those people who will properly engage with you as quickly as possible. And so no's just mean, right, that's off the list, I can focus on that. But I think, as Natalie said, you do need to learn. And I think the other thing that's out there is spending time. If you want to run the company rather than, say, be the lead scientist, if you're going to be the entrepreneur that's going to run the company, it's definitely worth the time speaking to other people in the field, reading. I mean, the web now is just amazing at what people post, like VCs, like some of the US VCs will do their sort of view of the year, and in an hour you can see. So we're in infectious disease, which 3 years ago was quite a big field, but at the moment there's only 3% of US VC money invested in infectious disease, only 3 years after the world's biggest pandemic in decades. So that I could look at that and immediately feel defeated. But actually for me, that's a big help, because I know 97% of no's, I don't need to go and waste their time, and I can focus on the people who actually are staying in, and that tells me they're resilient because they're bucking the trend. They'll have very specific requirements. I can go and find out what they are because people post, they tell you what they're looking at. So I think as well as listening, sort of spending time to really get into the minds and the environment that's out there to try and work out an investment plan is probably time well spent.
So we talked a lot about fundraising, clearly that's very important, but this is a really long journey, and no doubt there's a number of different challenges and unexpected issues you've come across. I wonder whether, in retrospect, there are any areas that came to you as a bit of a surprise or you weren't necessarily expecting that gave you those big challenges. I don't know, Mike, just continuing on from that sort of fundraising story, was there anything after that comes to mind?
Yeah, then I thought I'd be doing science, and then I realised I needed to speak to lawyers. I mean, I think that there's a huge amount that you're going to do that won't be, I mean, in the room there's probably way more, loads of experience, but it won't come as a surprise to those that have done this already that what got you to get the money in, you then, if you're going to build a company, or even if you're just going to develop an asset, if it's come out of academia is so successful because of collaboration. If you look at the reason we have such a big impact in the UK, it's because we collaborate worldwide, not just inside the UK. As soon as you've got money, or you've got a company, or you're developing an asset, you can't have collaboration. You've got to have agreements between parties about who's going to own what. So a CRO will do work for you in exchange for money. You own the data. But what if you need to collaborate with another university to do a piece of work? Well, that university, quite rightly, that's funded by a grant, often by grants, will want to own some of the intellectual property. And so really structuring what it is that you own and how you're going to interact with these people means you spend a lot of time with lawyers. And a good thing of being invested in a family office was you can use that back office, and some of the VCs and some of the tech transfer offices, I imagine, you'd be able to access some of that. But a big learning for me was the amount of time spent thinking about the science is a fraction of the amount of time of setting a company up.
And Hakeem, sort of going to that next step of then having to manage a team, recruit people, no doubt there must be some challenges in that. Any reflections of things that you were surprised by?
So one of the things in doing an early stage startup where we're sort of market making at the same time. So one of the things that we're doing is trying to bring two different regulated worlds of software as a medical device alongside a therapeutic, put the two together as a single prescription combination product, which hasn't been done before. And so I repeatedly shared this with the team and say, we're doing something that hasn't been done by people that have never done it. And that is a challenge, especially when in a leadership role, you've actually been out of the company financing and spending all your time talking to investors. And you get the money in, you complete, the money's in the bank, and you come back to the team, you go, what has happened? Because you thought that everything was going to plan, and actually the amount of repetition needed to ensure that the team is still working towards that same goal and hasn't started splintering, going in different areas, I underestimated that. And so the amount of time now, we've just recently closed some financing, and I've got a window of time before we go out again. And the first thing I'm doing is getting back inside the company and working out what is actually happening, because financing is a distraction, especially if you're stuck with lots of maybes. And so that was a big learning, that the amount of time that it can take you out of the company to get back in and sort of work with the team and bring everyone back together, and then you go off again. And so that was something I didn't really expect. And linked with that, I think what you were saying, Mike, is you've got your own internal team, but also it's the strength of the external team. It's the strength of the legal support you have around you. It's the HR support you have around you, which actually you don't necessarily think about when you're starting the company. It's the external counsel that you bring along with you on the journey.
And lastly, sort of, we've talked a lot about the fundraising and running a company, but you've obviously had two very notable exits with both Gamma Delta and Adaptate. Were there any surprises or things that happened during that you hadn't necessarily expected to go through?
Yeah, I think as with Mike Hakeem was saying, it's such an intense thing being the CEO. It really does become, it's an extension of your family, your personal life. Everything is about the company, the team you built, and my companies have been relatively small companies. Certainly, we're talking about 20, 30 people, so you know everybody, and you're working together very, very intensely. So, and I think you can become, and we often rightfully are really focused on getting to the exit, because you want to see your medicine progress, you want to see success in terms of how you've defined that for the company, and when an exit comes, that's obviously building up to an exit is a very incredibly intense time. And I guess during that process, me personally, I guess I hadn't really thought about anything other than what was getting to the exit, delivering that, getting everyone where they needed to be, seeing the our portfolio become then part of Takeda Oncology portfolio. And you don't really think about what's going to happen that next day, next start of, and I clearly remember that, finishing. Everybody was kind of settled in the new part of the company, obviously, as CEO, and my job there was finished, and I was done. And I remember that first morning when it happened that both my children had left home, both gone to university, and that morning my husband left for work, and he closed the door, and I was like, I've done it. And I was like, oh, right, I haven't actually thought about this at all, because it's like, the phone's not ringing, I've given my laptop back, it's like, this is it, we're done. So, it was, and I think, we won't talk about that, and I guess, you realise actually, how much of you went into the company, and, and I think, then obviously, there were lots of opportunities, lots of exciting things, but I think it's also, real pleasure to be able to look back and say, I really enjoyed that. I actually miss that. I don't feel, like, oh, thank God that's all over. Actually, you miss the people, you miss the excitement. It is a roller coaster ride, but I guess I would also say, enjoy the journey. The exit is a thing, but enjoy the journey as well, because when you get to the exit, everything changes. And Takeda at AACR last week talking about my portfolio, and that was good, and I was very proud, and I was like, but I was making notes of things, so they could points for improvement. It is. I think you have to give that up, obviously, at an exit. But I would say, enjoy the journey too. It is. It's great fun.
And what's that dynamic like when, presumably, there's some people that continue to work for Takeda, and you've extracted yourself moving forward? How does that feel? Is there a sense of you sort of miss that team and feel like you owe them more, or is it a case of actually you're happy to see them now sort of at Takeda, and that you feel your job is done there?
Yeah, and I think really, I guess, fundamentally we do this because we want to make new medicines for patients, and that's the thing that I'm you're nervous about when the company becomes acquired. Is there going to be some reason why actually these things don't go on to where you want them to get to, and of course, you have no influence over that you have no inside track anymore to know what's happening, and it is very odd being there. But I think, at the end of the day, we do this because we want to make new medicines, and I think, seeing things progress, it is very odd. It's very odd to hear someone else talking about, your work and your company, and you're not part of it, and they start showing data you haven't seen before, and, it is, it's odd, but also, I think, you feel very, very proud, and I think that everybody in the company has been on that journey, and they've all learned such a lot. I mean, it's been, everyone, you've seen how people you've hired, as new scientists, first time in industry, have actually really developed through that as well. And I think that's, one of the great things you take on is, you've grown your network. You can see the next generation also learning, how to work in these companies, and the therapies are going on. So, I think you have to take the positives from it, despite feeling a bit left out.
And, one of the other dynamics that we've not touched on yet is what it's like to have investors and board members directing your team, your company, getting involved. And this is an opportunity to dish the dirt, if you will. I don't know whether, Mike, you've got any observations about what it's like to work with boards, both good and bad.
I've got a few. So I think, the boards are funny because for me they were a new entity because I'd worked in big pharma, and there was clear lines of sort of authority. You knew how decisions were made, and I guess governance was present in my experience in pharma, but it was sort of, I guess, slightly different. So, or at least it felt different to me. So I think understanding the board, when you've raised money, then you get a shareholders agreement, And in that shareholders agreement, it will lay out how you're going to be able to run the company. But, fundamentally, the investors, most of the time, are there to govern you, not to manage you. So, the leadership team's job is to execute on the plan to grow the company. And very often you're thinking about an exit, but actually you're trying to grow the company because, as we've seen in the last few years, if you're driving towards an IPO and then suddenly all the IPO market collapses, then you've got to change your plan. So within the investment, there will be an agreement about who's going to be on the board, how you can appoint directors, how you won't appoint directors. But all directors in the UK have what's called a fiduciary duty, and it took me about 8 years to be able to say that word, and I'm still learning sort of what it means. But essentially, whether they are there because they represent the investors, they are on the board, according to UK law, to basically ensure that the company is run properly, and that we do things in the interest of the company, not the interest of the fund, which is not to say that there aren't sort of areas that the directors who are either investors or independents will want to sort of take the company. And I think that's really important to realise that you do have a duty to the board to do what you say you're going to do, to report, to spend the money appropriately, but also, they're looking to you for direction because they're not running the company. And sometimes it might feel like it if you've got if you've got quite a active investor director in there. So I guess I would sort of say that trying to have a relationship with the board members outside of board meetings, don't bring the board a surprise and then expect that it's going to land well. People need time to sort of think it through. So that's one learning. And then I'd also say, if possible, if you can get an independent director on board, that's sometimes quite useful just as somebody who's not got another angle. They're there because they've been invited on. They've got an incentivization usually around options, but they're really there to help you as the leadership do the best for the company. So I think, board meetings, obviously, there's loads of, stories, board meetings go well, go badly, you leave and go get frustrated because suddenly the member on the board wants to take it in the wrong direction. But usually if you can have those discussions outside the board, that will greatly sort of help you in the board meeting.
And Natalie, building on that, are there any things now that you wish you had known in your sort of previous roles about those board dynamics, or things you discovered that may be relevant to people in the audience going down that path?
Yeah, and I think I think what Mike said about your job as management really is to lead the company, and you know this company inside out. You're living this kind of every day. So I think it's really about learning how you can get the board members to help you. In the end of the day, you are in this together. You've got to understand, they're different, different needs. And I definitely agree, Mike. You need to try and build that relationship and build that trust. But also, I think, it's important that actually you're giving the board, the direction. Yeah. They may not agree with you, but actually, you do know your company inside and out, and actually you can help them to help you that way. I think also, one thing boards can do and do, which I know can often come as a surprise the first time, is actually they may be thinking about something completely different to what you're thinking about, and they can come out with something, that is, you think, well, that's not what we're doing. This is the plan, and all of a sudden...
Is that the day after they've made the investment?
Exactly, yes. Actually, what we really meant was... So I think, those kind of surprises, you definitely try not to surprise your board. Sometimes your board surprises you with things that actually, I've been thinking, actually, maybe you should do this or we should try that. And I think, those you have to kind of learn how to kind of manage that, how to, give the right consideration, but also kind of take things, kind of in a calm direction, those meetings too.
And Hakeem, closed loop medicine is a very different business. It's very much on the frontier of tech bio, as you said, doing something that's never been done before by people who are very much learning how to do that all. When we think about your board and the composition of that board, I think you've worked quite hard to get a really creative mix of people. Tell us more about how you've done that, and perhaps what are the things you were trying to solve for, and how you sort of solved for it.
I think that's quite true. I think when the journey we've had, I think in terms of board structure and role, was very much led around investor directors to begin with. And we've now got into a position where we actually are bringing now independents in who are the right type of independent to help alongside the governance of the directors in terms of their network. So we've sought people who are explicitly not in the UK, not connected very well to us so that they can expand our network. People who have done elements of what we are trying to do before so that they can challenge. And we get some really healthy challenge from the independents of why is it taking that long? Go away, halve it. And you go back to the team and go, is it possible? Actually it is. And you go back and you can improve. So we're now shaping our board to be much more international, more independence, with bringing in facets of the puzzle pieces that can help challenge us along the way, because we can't find anyone who's done it before because it hasn't been done. But that balance and shift from investor to director majority to more of a balance between independents and investor directors is actually, you can see it in the board meetings now. You can see the dynamics changing, you can see the conversation getting better. And that doesn't happen overnight. You really have to work at it. And for many companies sort of spinning out and starting up, you will have less and less control over what's in the shareholders agreement because it will be dictated to you until you get to a certain point where you can start to actually having meaningful conversations because you've built up the trust with the investors. Say, you know what, when we write the SSA next time, how about we do this and how about we do that? And that then gets to a really interesting point where you're starting to build the board for the company rather than the other way around.
Rather than protecting the investors'
Exactly.
interests. Yeah.
Exactly.
And this is a bit of a leading question, but do you see that there is a difference between how UK and or European investors influence at the board level versus perhaps US investors? Or US board members, I should say.
Yeah, I think what I've seen from some of the US board member, or the board member we have, and another one we'll be bringing in shortly, is their view on timescales and vision. Is sort of, just do get a sense, and it came out from the last panel, of just a slightly more pioneering than what we see normally. And I think one of the challenges, if I look back on the funding history, we've gone through angel funding, seed funding, and then brought on LifeArc and others in a larger round a couple of years ago, is that the smaller aliquots of funding meant that the milestone you wanted to hit wasn't quite in line with the path you were taking. It might take you a little bit off. And so then when you do go back to an investor and they say, why have you done that? It's like, because I didn't have enough to do what I wanted to do. And the zigzag is fine from a sort of a sailing analogy. You've got to get to your waypoint, but you can't go too far away. Whereas some of the stories you hear from entrepreneurs, say on the West Coast, is they just come out of stealth with 200 million and go straight forward. And that's not something we see here in the UK, and it's something we're not going to pretend we're going to get it overnight, but I think there is that difference in working out what are the key milestones, what are you going to fund, and it's that perception of sort of investing in the vision as much as the asset.
I mean, there's a lot of luck and serendipity in what we're all doing, both as investors and founders and CEOs, and often when we're going through that journey, you can go through what feels like catastrophic moments, where actually when you get sort of further down the line, you realise that was potentially quite fortunate. I don't know whether you've had experiences like that, Natalie or Mike.
So I guess when you're running the company, I guess you never know whether what you're doing is a mistake or not. I think that's, and even in hindsight, so you don't know because you didn't do you didn't take the other path. So I think, it can be, you have to make hard decisions for sure. And I can remember, hard decisions that we've had to make on, making changes in the team or, changing things in the portfolio and stopping doing some things in favour of doing others. And I say, you don't know whether that's, you can't really say whether that was the right call or the wrong call. I think as CEO, you have to be, think about it very carefully, take advice, but then you need to make a decision and bring the company behind it, and then make a success out of what you've decided to do. So, I think some of those are the very hard moments when there really isn't clearly a right or a wrong decision, but you have to decide for yourself what to do and take the action and then after that your job is then to get on behind it and make it work at that point.
Yeah, I think the experiment or the event that you don't want is, again, it's not, if it's a clear cut, it's never as bad as the one where, as Natalie was saying, it's not clear cut and you've got to make a call. So even if an experiment flatlines and you go, I just really needed that data, you can make a decision on the back of that. It might not be the decision you wanted to make, but it's actually quite easy and everyone will support you because the data is the data if you've done the experiment well. But I think, it is an absolute sort of roller coaster when, for me, coming out of pharma, you had projects and the site eventually needed to do a certain number of successful projects, but there were teams, dedicated teams, all basically within the same building trying to do the best. And you knew that, a bit like in a VC portfolio, as long as some go really well, then the fund is all right. When it's just your biotech, and I think we heard a question earlier about, platforms, a lot of people start out with something that could be a platform, whether gamma delta T cells or the last company I was in, Centuri, you've got this ability, and that instantly builds a bit of ability to be a bit more resilient. But as we were just saying, you don't in this country get the quanta of capital to explore multiple paths. And so, and at the moment there is a massive, I think because of the M&A and the way the field is, there's a massive push on products, as we've heard. And that doesn't intrinsically build you much resilience as a small biotech. So I think, if that's not for you, if you want the certainty of knowing you've got a job until a large event like Pfizer's decides that it doesn't, it's going to make a change, then biotech is much more uncertain. But then at the same time, there's much fewer politics. I mean, even a bad board, you've only got to meet once every couple of months. Literally, it's not that bad, but you do get to sort of experience the highs and the lows really firsthand. So a great bit of data coming out of the lab. I remember just before we did the deal with AZ, we were a long way behind our competitors. Everyone had sort of said, we're not going to, I thought the only way to go and raise the money is to go and ask for what Americans were getting. So I went into a couple of the big VCs in this country and said, right, I've got 4 world-class scientists, great idea, I'm pretty sure I can get hold of the IP, and I need 50 million. And it was a 4-page pitch, and I thought I'll get left out. I didn't get left out, but in the end they said, actually, we're much better investing in IP, and we think we can build the team. So it's a no from us. And then spoke to Claire at LifeArc, who was building, and her first response, this is exactly the sort of thing that LifeArc should be investing in. And that wasn't by the way, that wasn't 50 million. It was 500,000, but that was the first time I'd ever met Claire. In fact, it was online. We hadn't met in person. And through that, we were able to get hold of some licenses. We were able to bring some compounds in. We were able to do the testing. And then when we did the deal with AZ, I remember, hopefully I won't get told off for saying it, but I remember we were having drinks, first time most of us met in person on the top of LifeArc, and we had this thing called the Pretty Woman moment, which is in the film, if you've seen it, there is a moment where Julia Roberts tries to go and buy some fancy clothes and she gets sent out because she isn't the type of person that they wanted to sell to. And then after Richard Gere has spent an offensive amount of money, she goes back in with the bags and she goes, big mistake, massive. And there was that moment where after all the no's, you get that success. You just have that sense of actually, do you know what, it was worth being resilient. And, to those that didn't invest, they had their reasons, they probably made the right choice for them. But there was that one moment where we're just like, big mistake.
Look, I think that's a wonderful point to conclude the the panel on. And I'd like to say, as an investor, one of the most sort of motivating reasons to do what I do is to get to work with people like yourselves who are talented, gracious with your time, and really trying to do amazing things. And I think on behalf of LifeArc and everyone in the room, just like to thank you so much for being so candid and sharing your thoughts, and look forward to continuing the discussion outside. Thank you.
Thank you.
I run a company full time and I love learning so much that my extra 20 hours of free time a week I go and I have some really awesome applications and devices and projects. Basically all of my devices also are yeah, we love the
Hey, they had that deadline.
Yeah, we get one and yeah, exactly. So one of those partners, they run a company with partners and non-profit. We're still working our way through what we're doing as a company and exactly the focus we've got to. It's focused around trying to say, I mean, really big picture, it's saying, like fundamentally as people mentioned it, the way we're proposing outside of my area just doesn't work. It's not broken. What kind of technical solutions can we bring to solving that economic problems above and beyond just the...
I've been here about a year and a half.
Developing intentional protein antibiotics with really targeted value propositions to different nation groups.
So where are you going to launch that?
Where are you going to launch that? Well, that is sort of 10-year...
Yeah, but you know, it's got to be on your mind, right?
Exactly. At the moment, fundamentally, as far as I can see, our research is every company is about making drugs for Americans. And you have so many drug systems that Americans will have. And then well, and if you can't do it as your company, maybe it's a point of next time. Somebody else could do it there.
I say that because I found the exact same thing.
But you develop it outside the US, so it's very cheap. This analogy is now breaking down. It's good sort of 3 years ago, but we're thinking of the kind of ways that SpaceX is trying to colony on Mars. They said first we'll make reusable rockets, and use reusable rockets to make a satellite system. That kind of
live when you can't see anyone.
Good afternoon, everyone. Thanks so much for joining us for the Life Sciences Accelerators What Do Companies Need session. Really pleased to see you all today. My name is Jason Slingsby. I'm Chief Business Officer at LifeArc. It's a real pleasure to have you all with us from different walks of life, from different sectors, different interests. And I'm really thrilled to have this panel discussion with Barbara, Jason, and Catherine today. So LifeArc is really keen to see how we can support translation from discovery through to clinical trials and patient impact. And a lot of that happens in companies. And companies, clearly, I'm sure a lot of you are either running companies or funding companies or working with companies. It's often the first steps in a company's life or a proto-company's life that are really complex, really difficult, and the support and the mentorship and the financial stimulus that some companies can get can be all important, and if it's sometimes lacking, it can really be a drag on achieving the goals and aims of founders and companies. So I'm hoping this afternoon we are going to hear from three gurus in the field who are all active in the sector for accelerators. And after the discussion, we'll have 4 to 5 minutes from each of our colleagues to explain who they are, where they come from, and then we'll have a discussion around life science accelerators. Towards the end, I'd really hope that we can have 10 minutes to hear from you guys, because the title is, What Do Companies Need? So we're actually also in listening mode. Please think about what we say, whether you agree with us, whether you don't, or what we've forgotten that support and help that you would appreciate in journey to becoming a more established entity. So, with no further ado, I'm just going to briefly introduce Barbara Domain-Hayman, who's entrepreneur in residence at the Francis Crick Institute and also chair of KQ Labs. Then we have Jason Malad, we're sometimes known as the Jason Double Act, co-founder and chief executive of Start Codon in Cambridge, and Catherine Chapman, director of science and entrepreneurship at the Babraham Research Campus. Without further ado, Barbara, please tell us a little bit about yourself and your various accelerator interests.
Well, first of all, thank you very much indeed, Jason, for the invitation to participate. So my name is Barbara Domain-Hayman. Just going back a long way, by background I'm a chemist. But I went over to the dark side as soon as I finished my doctorate. I spent the first sort of 10 years of my career actually, I went into marketing in a big organisation, and then which was actually ICI, as it then was agrochemicals. But after nearly 11 years, I had an early midlife crisis, and I went to London Business School and actually stumbled on the world of entrepreneurship, and actually discovered that what I really wanted to do was to go back to my roots, use my science background, use my commercial experience, and build businesses, because that was the part of the commercial world that I'd really enjoyed doing. So it was a real light bulb moment to actually go into biotech. So that was 23 years ago, and I've spent the time since then basically working in biotech in a variety of companies, building them, and really enjoying that journey. I've done a whole range of different roles and a lot of business development, sort of CEO, chair, a couple of exits along the way. And for the last 10 years or so, I've been doing a portfolio of activities. So I'm still on the operational side. I'm still Chief Business Officer of a biotech company, or Tiffany Therapeutics. And I'm also have a foot in the investment camp now. So I'm actually on LifeArc's Early Ventures on their investment committee, which is great. I'm also involved with Cambridge Enterprise Seed Fund, their ventures investment committee. So I see from the investment perspective, and I'm also a Cambridge Angel. But I think for the purposes of this discussion, I think my role as entrepreneur in residence at the Crick is the most relevant. So at the Crick, we do a number of entrepreneurial training activities, which we think are very important. And because we get money from a variety of different sources, but including governmental sources, we do things for the whole of the UK. So it's not just about the Crick. We do the Pulse programme jointly with the BIA, which I'm sure some of you know about, which is a 3-day boot camp for first-time founders coming out of academia, basically taking their first steps into the commercial world. That's become a very established thing. We've done it 7 times now. I think perhaps most relevant for here, I'd like to talk about the KQ Labs programme, which is what we started 6.5 years ago. So when I arrived at the Crick, there was a lot of discussion about the interface between data and biomedical science and what a huge impact this convergence was going to have. And so we were thinking, well, what should we be doing about it? And actually looking around at the very early stage companies, we saw that early stage companies were really struggling to get investment. And we thought this was really because investors tended to fall into two camps. They were either coming from a biotech perspective, in which case they understood risk, they understood how science takes a long time, they understood the regulatory world, but they don't understand the business models in this new emerging area. And the business models are indeed still quite fluid. And that's quite scary because people don't understand how they're going to make money. Tech investors, on the other hand, less uncomfortable with that side of things, but uncomfortable with the regulatory constraints around life sciences and not really understanding how the life sciences world works, the pharma world. So these two groups of investors really sitting on their hands and not investing. And we thought, well, actually, if we don't support the very early stages, we're not going to have an ecosystem in this country. So we were thinking, what can we do to support those very early stages? And we were sitting in the Crick in King's Cross and looking around. And of course, we saw that there were the beginnings of a really interesting cluster there because there were the big tech companies were there, but also big biomedical institutions were there as well. And we thought, well, actually, there are no startups. What can we do to catalyze this to really become a kind of Kendall Square for data-driven health? And, but we'll talk about place, I think, later on, probably. But we really thought, what can we do? So we thought, OK, well, let's run an accelerator. We do experiments at the Crick. Let's do an experiment. We'll see what happens. So we got our first funding from Innovate UK, and we were inundated with demand. And it went very well. So we thought, OK, we'll do it again. And we've actually now done 6 cohorts of companies. So basically, we select 10 companies at a time. We put them through a 5-month program. And really the mission of the program is threefold. It's about helping the companies to validate their ideas, to introduce them to networks of people, investors, pharma companies, and so on. It's giving them that first network. Secondly, it's about investment. So, as I said, basically these companies are struggling for investment. So we help to get them investor ready. And then at the same time, we bring investors to meet them and encourage investors to perhaps go a little bit earlier than they might otherwise do so. And then finally, it's about the people, and it's about the ecosystem. So we're trying to build an ecosystem here of data-driven health startups. But we're also very conscious that the people who are running these companies, these are not going to be their last companies. They're going to go on and found other companies in the future. And we're really trying to train that next generation of health tech, or whatever you want to call it, leaders. So that's really what it's about. We've now done 6 cohorts, as I said. So 60 companies are out there that have been through our program. So we're beginning to have a critical mass. They've collectively raised about $120 million pounds since being on the program. These are very early stage companies. I mean, when they come to us on the program, they've typically raised anything between 0 and £1 million. Average raise about 300K. So these are very early stage companies. And they typically fall into 3 buckets. We define data-driven health very broadly. We're basically the only accelerator that's completely focused on data-driven health. But we define it broadly. About a third are related to drug discovery and development. So they're tools of AI in drug discovery or target discovery. The second group are mainly around diagnostics, imaging, biomarkers. And then the third group are more patient-facing, maybe apps for remote monitoring, specific diseases, et cetera, et cetera. So those are the sorts of companies that we have on the program. As I said, 60 have been through the program, and we are immensely grateful to LifeArc because LifeArc has funded the program for the last 4 years. We could not have done it without LifeArc.
Thank you.
So,
subliminal plug for us. Not intentional. Thanks so much, Barbara. Jason.
Thanks, Barbara. Thanks, Jason. Again, thank you for having me. We are not funded by LifeArc, but we're open. So if I can make a very, a plug for us as well. I have been in the UK since 2004, came over to do my degree, made my way through tech transfer and consultancy and biotech to set up Start Code On with my co-founder Dan back in 2019, and it's been a wild ride ever since. There was an unmet need that was recognized both in Cambridge for across the UK, very similar to what Barbara was stating, for bridging the gap between those first-time founders in particular who are going out and speaking to investors, and the investor says, if you accomplish X, Y, and Z, I'll invest in you. And the founder says, well, give me the money to do X, Y, and Z, and the investor says, go get money somewhere else and come back to me. So constantly, there's this disconnect between the early stage innovator who needs that extra hand-holding support, who's never done this before, who needs a team building, and has that understanding of their field, but not necessarily the commercial experience to give investors comfort. They come to a program like Start Code On, or indeed the others from my colleagues on stage, to really ensure that we help them through that difficult valley of death. I always like to say that the valley of death is the best place to be in, because when you have nothing, you've got nothing to lose. And you can get so much support for free. Once you've raised capital, consultants start charging money. When you're in the valley of death, they go, oh, I'll give you an hour of my time. And I say, leverage that. And so Start Code On is founded on the idea that we can use the community and all of this expertise, not only in Cambridge, but across the UK, to help British businesses get up and running and make them venture backable. We were very fortunately backed in the earliest days by the Baberham Research Campus, by Cambridge Innovation Capital, Jonathan Milner as a philanthropic gift of sorts. He's an investor in the Topco. Ian Tomlinson, who we adore, who unfortunately is not in the room with us today, but for any of you who know Ian, he is phenomenal, still is phenomenal, and we're expecting him back someday. And together with Genentech supporting, Novartis is one of the LPs in our fund, we brought all of this expertise, successful entrepreneur, very serious venture capital, a world-leading research campus, pharma, and we said, these are the elements that you need to make sure that you choose the right companies, you build them in the correct way for them to get investment, and you kind of ease them on the path towards success. We started off at the very beginning of COVID. So imagine incorporating in 2019, first close in Jan 2020, first cohort, some of whom I think are in this room, starting off, I'm going to say February of 2020, and then we all went home for 2 years. So it was quite challenging because we had a very set model of being in the Milner Therapeutics Institute, stay with us for 6 months, office and lab space, $250K check. We quite quickly turned into completely virtual, having to invest in people we hadn't met before, because part of our thesis is you have to deploy capital or else we don't pay our salaries. So it's been quite the journey from where we thought we were as a traditional accelerator, office lab space, set period of time and cohorts, to what we've evolved into over the time, which is bespoke programs, a rolling program that's throughout the year as a base, and then kind of bespoke interventions for each company. Looking across the UK, it expanded our horizons. Not everybody wanted or could move to Cambridge. It allowed us to branch out to Loughborough and Portsmouth and Birmingham and across the UK to get the best talent. And importantly, we feel that we've built up now a community of over 27 companies. They've raised £60 million over the last couple of years in total. They're still raising, so if any of them are in the audience and any investors are interested, come talk to me because we're always here to support them. And we originally thought it would be a kind of one and done as well, that they would join us for 6 months and then off they go. Turns out we've built a community. Whether we're taking board seats or just having a friendly WhatsApp chat or always being available to support, it doesn't stop. It's not as hands-on as it is in the beginning, but definitely the place of accelerators and incubators who really want their companies to succeed is building up the critical mass portfolio, making sure you have that kind of wraparound support. And that's one of the things that Start Code On is really enthusiastic about doing. In collaboration with so many others, we've had companies who've worked with us and also gone to KQ. We've had plenty at the Baberham and many other support networks. So really excited to be on the panel.
Thanks so much. Catherine.
Thank you. Thank you very much for inviting me as well today. So I'll say a little bit about myself and then a little bit about the Babraham. So I am a scientist by training. I have done postdocs in the UK and the US, and then I moved to GSK. So I've worked in pharma and in the academic arena as well. I then moved to UKRI for 10 years, where I helped to think about where government funding could be invested in startup companies and challenge-led innovation. And this was about really my passion, which is about trying to make sure that science that is funded in academic labs can have real-world application, and how to join those dots and make sure it's having impact in the real world. So that's where I sit. So for the last 9 years, I've been in the Cambridge ecosystem. So I moved there to set up the Milner Therapeutics Institute, and that was set up. You'll hear about that later, so I'm not going to say much about it. Kathy and Nicola are going to come and present on that. But again, it was fulfilling a gap in Cambridge at the university, that there was not a translational research institute where startups could sit with academics in residence, could sit with pharma companies, and that community could work together and support each other in that translational journey. And as I say, you'll hear a little bit more about the Milner later. So then I moved on from the Milner to the Babraham Research Campus, because that was a bigger playground to play with, in that there are 60 biotech companies on the Babraham Research Campus. It's got a long history of supporting biotech companies from very, very early stage, so from just single founders right the way up to companies of about 200 people. And so that's what my job is now. So I look after the innovation strategy at Babraham Research Campus, and that involves the institute and the science that goes on at the institute. There is a healthy aging institute there, and they do epigenetics, cell signaling, immunology, and it's about mechanisms of disease, mechanisms of healthy aging. And we work with them from a facilities perspective. So it's a government-funded campus, so most of our investment is from BBSRC. So there are flow cytometry facilities, sequencing facilities, imaging facilities that all of the startups and companies on campus can access. And so we start at that level, and then we have an accelerator program, which is probably the most relevant thing today, which has had 26 companies come through it, and they get £10,000. And they get 12 months free lab space in the Babraham. So we're very focused, and I can talk a little bit about how the programme and why the programme has evolved in a minute, but we're very focused on that lab support and the experimental support and the key no-go decisions to fail fast, so those companies can come in the labs, do those experiments. If they work, brilliant, they can get funding. If they don't work, okay, next idea. But we then have a pipeline so that those companies can move into incubator space. So we have really flexible lab space that companies can start using just for an hour, a couple of hours a week, and they can terminate, within 3 or 4 weeks' notice, right the way up to bigger space. So they can scale up, as I say, to about 200 people. So that's the campus, and the role brings together all that critical mass. So we have CEO networks, we have pitch days, we have CSO networks, we have everything to make sure that those peers can learn from each other, support each other, and have a really fantastic campus community. And I think maybe just talking about the accelerator program, one of the things and one of the reasons it exists like it does at the minute is because there are so many accelerator programs around the UK. And when we started in 2019, there weren't so many. I mean, we were working with Startcode and others. There was a gap. But actually, as we've seen the accelerator sector grow, we have also had to pivot and move and think about, okay, what can we as an accelerator provide that is distinct from some of the others that on the UK landscape? So we're now very, very focused on the lab offering that we have and the critical mass and on the Babraham campus of support. So CEOs, co-founders, serial entrepreneurs that have done it multiple times. That's really, really what we focus on.
Fascinating. Well, thank you all so much. So it sounds like there's a bit of a question in the sector. What's an incubator? What's an accelerator? Where does one end? Where does the other finish? And where does the sort of real estate industry itself begin? So if you think of the sort of 3 value elements, it sounds like from what you've been saying, there's an element of newcos needing labs and offices, needing funding. Although I do really appreciate your comments, Jason, I've previously been an entrepreneur with no money, and that's a powerful position. But it only lasts so long. And then there's the mentoring and networking. So maybe if you could start as Jason,
if you had to prioritise one of those three, which would you prioritise, or is it a magic of cooking needing a perfect combination of ingredients?
I think you need the chef, the recipe, and the ingredients. I think that you've got to have a combo of all three. It's a question of sequencing and also, let's say, quality versus quantity. So I think that in all those three verticals, you can imagine if you have too much funding, but you don't have the team that knows how to deploy that capital, it's going to fail. You can have numerous mentors, and we've had a very early experience, early doors. We thought anybody who wants to help can come in, but then we quite quickly learned that not everybody who says they're a mentor is truly a mentor and has been prepared. So now we do more triaging, we spend more time coaching mentors on how to be good mentors and how to be good advisors. So those elements are important. Quality matters. I've seen great companies, we write 250K checks. It's a small amount, and they're very, very lean pre-seed companies. But if you can leverage a bit of non-dilutive capital, a little of venture kind of syndication, you can get a lot done on relatively little. It forces you to focus. So I think quality always has to come in. Same thing with lab space. We were providing lab space via our time at the Milner, and we stopped because of post-COVID, we changed. But I find that it's important, but is it serviced in the right way? Are you in an environment where there are other young companies and other first-time founders that you can work and network with? So that environmental piece is important too.
Yeah. So Barbara, if you look back to your experience as both an entrepreneur, company founder, and KQ Labs, presumably infrastructure is not such an issue for your KQ Labs cohort, or?
That's true. I mean, basically, out of our typical cohort 10 companies, maybe one or two need lab space. The others don't. They just have office. They're in silico. So from that point of view, lab space is less important for them. Place is still important. And I think bringing people together and enabling that peer-to-peer learning, and just that place is definitely still important. And King's Cross, I think, has become an amazing place to do all this stuff. I think the other elements, I mean, money is important. I mean, basically, we give 40K to each of our companies. And, I mean, that's a very small amount of money. For a company that's already raised, maybe 800, it's not that meaningful. It maybe means that they can do one more thing, do one more project, hire one more person. But it's not that meaningful. For a company that's raised nothing, 40K is meaningful because it often means that the founders can give up their day jobs, for instance. So that's helpful. Money is important because without money you don't survive. So I would say the money is important, but what we provide is the catalyst towards the money. We're providing the introductions to the money. So I think that's important. And I mean, that links us to people, which I think is the most important thing of all, which is basically the mentoring that we provide. And we have a network of mentors, just as Jason does, and the networking. But I think the warm introductions is very, very important because that's how you get people's, if you send an email to an investor and they don't know you, you're probably not going to get a response. But if it's from somebody that they know there's going to be some kind of a quality bar, threshold associated with it, they're much more likely to actually respond. So warm introductions definitely very important. And people, of course. I mean, as I said before, we are training that next generation of leaders.
So, Catherine, when you think about Babraham's value proposition, how do you think about the larger commercial real estate investors that are ever more coming into increasing lab capacity, not only in London, but there's a lot of development going on in Cambridge. There's even a lot of development going on in Oxford, where I live. So how does Babraham see that sort of larger wave of real estate investment?
Very positively. Lab space in Cambridge there's been a shortage of lab space in Cambridge, so we need it. So that's that. I don't think it's not a bad thing at all. I think that Babraham still has the position, given that it has government funding, to be able to mix that, to work with companies that are very, very early, that can't actually get leases with those other real estate developers, but they will be a pipeline for them, and it means that they've got somewhere to grow. So it's the system works really well. And I think that's what's the most important thing here, is to have this high-functioning ecosystem where everybody works well together and is collaborative and sees where the companies can go. And in fact, the most successful companies in our ecosystem have been the ones that have been through Start Codon, been to the Milner, and now at the Babraham. They've had CIC funding. You know, it's basically a whole ecosystem supporting those companies that are most successful. So I think that's really important. So I guess a word to the developers of that, if the developers come in, it's to how do they fit into that ecosystem. And for us, we have a joint venture with Biomed. Actually, it's not just Biomed building on the biomedical campus, it's a joint venture, and Biomed fund the accelerator program. So it's a win-win. So it can work really well.
But you've got to be an authentic participant in the ecosystem and really not just pay lip service. Great. Well, one thing that I think LifeArc's really interested in is equity, diversity and inclusion across the board, but also in startups and the startup scene. And we're all kind of golden triangle sort of nodes here. I live in Oxford, London, Cambridge. So when you're thinking about your own accelerator strategies, how do you think about trying to stimulate, support those networks and spreading that energy, that capability across the UK? Barbara.
Yeah, I mean, great point. I mean, yes, so for the KQ Labs program, 63% of our companies come from London. I mean, London is a huge hub for data-driven health. Then next up is Cambridge, I think it's 16%, and then Oxford. So yes, it is still quite dominated by the Golden Triangle, but 16% is beyond the Golden Triangle. And we've had 3 from Bristol, we've had companies from Scotland, from the Midlands, from Leeds. We go to a lot of trouble, actually, to try and promote that. We really want to have companies coming from all over. It's quite hard to get that message across. And I mean, I think they've got a lot of stuff going on locally. And I mean, we're not trying to poach them. We're not trying to say, come and relocate here. We're trying to kind of leverage our network to help the companies. And I think, I'm not sure that that's necessarily the message always gets across as loudly as it should, because we would really like to have more people coming from beyond the Golden Triangle, but, you know, not sure what we need to do differently to achieve that. So it's a work in progress, I would say.
Jason, I think you're pretty proactive.
Yes, we do. We engage across the UK. We have roughly about a third of our companies in portfolio are from Cambridge area, 50% are from the Golden Triangle, the other 50% are from across the UK. And it's interesting because when we started off, we were very Cambridge-centric, as I said. That was before the pandemic hit. But as we lowered those hurdles for people to get access to our support and to our capital, we discovered that there's so much incredible deal flow across the country that it's actually been a boon for us, particularly because a lot of VCs, early stage VCs, aren't hunting in those grounds. So we swoop in and we say, this is fantastic. How come nobody's coming here? Wait, we won't tell them just yet. We'll grab the best deals for us. And then they can see in our portfolio, it's like, yes, you didn't realize that was happening in Portsmouth, or you didn't know there was something amazing happening up north. And the kind of follow-on from that is now we've been engaging with a lot of different ecosystems about how they can identify what their core pillars are and maybe even have their own version of a Start Codon. Whether it's a Start Codon franchise or they set up something bespoke for themselves is something that we're very keen on supporting. We have a philosophy that the more we can lift up the ecosystem with really strong and successful and well-run programs, the better. To Catherine's point before, there's a plethora of accelerators popping up. I call a lot of them stagnators. There was you're not really accelerating, you're just kind of sitting there and festering a bit. But it says accelerator on the cover. It's like, oh, this is great, it says accelerator. Not all accelerators are created the same. And what I think is a challenge for us is as we try to level up the entirety of the UK and beyond, we need to think about if you are a company that is in a vertical that maybe being in the Golden Triangle is particularly good for you in terms of talent, capital, etc., then we shouldn't have hurdles saying that you can't move there. But vice versa, if there are some amazing companies in London, Cambridge, or Oxford that might be better off moving up to a Newcastle or out to Norwich or out to Bristol, we should encourage that porosity as well. So I would advocate for eliminating the stagnators, making sure there are proper accelerator programs, and also having that porosity so people move back and forth.
Yeah, I in a previous life I was founder CEO of 2 companies in Austria, one in the south of the country, no biotechs there at all. So we got beautiful lab space, incredibly cheaply, so chic. Investors could just like never believe that the facilities were there, but no networks, difficult to hire people. But we had lab space was never ever a real aspect on our finance plan. So and once we recruited people, no one ever left because we were the only biotech in town. So I think there's a really captive.
Wait, hold on. You mean the best biotech in town?
Indeed, the best and the only. So I do think that there's some real distinct advantages to being in a cluster, because there's lots of people like you around and lots of skills and people to have coffee with, but then there's lots of other opportunities that come your way. So I think there's pros and cons. So How do you feel at Babraham about the sort of more national level agenda? Have you been able to attract lots of companies from outside of the sort of Cambridge area, or do you quite naturally see that most of your tenants do come from the local ecosystem?
Most of the tenants are from Cambridge. It's not quite the same on the accelerator programme. We do get a bit broader representation on the accelerator programme, but it's something I'm really, really passionate about. And I think there are a couple of aspects to this. So one is that if you look at all of the data and all of the reports, companies that are spinning out of an academic institution or starting up will generally stay with their local support system, because it's very difficult to move down to Cambridge from Manchester for a whole number of reasons that are not related to the company. So it's really hard to do that. So if we're thinking about the hurdles, then we need to think about, okay, how can we offer stuff which means people don't need to move, so that they can access our Cambridge network and all our Cambridge expertise without even moving. That's your online bit. And then there is the bit of how do we help people move just for a short period of time and have a sabbatical on the campus, and then help them to move back. So they're not coming to Cambridge to stay, they're coming to Cambridge to pick up all of this information like a sponge, and then we're saying, okay, but after a year you have to have your plan to move back. How do you do that? And give them funding to move back. So I think it's really, really, really important to do that, and I'm hoping that we can find ways to be able to do that together as the UK as a whole.
Yeah. I mean, if I could just add, I mean, I think it's really important, the points have come out, that, you know, the accelerators kind of play well together, and I think we do. And so basically we just need to signpost to people, you know, which are the strengths of all the accelerators. And I mean, you know, we're the only data-driven, health-focused one, but we know that, you know, you guys, and we've had a lot of traffic between us, both ways, actually, which is really nice. And I mean, we've had, same with Babraham. And I think it's really important that we do that. I think companies need to be a bit careful not to do too many accelerators. And we certainly have a bit of a
surfing was mentioned.
Yeah. We have a bit of a red light flashing if a company's done like more than 3 or something.
Really?
But I think you know, but as long as they can really justify why they've done each one, and there's a good reason for it.
Straw poll. Put your hands up if you've been through an accelerator program in the UK. Keep your hands up if you've done 2.
Oh, interesting.
Keep your hands up if you've done 3. No, we've only had one person.
Yeah, nobody dares. Exactly. It's okay. You can talk to us later. We have a support group. It's fine. Stay anonymous. But if maybe, Jason, I could come in as well, too, as I think that there's such an emphasis on the Golden Triangle. And I feel that I made the mistake when I went to Leeds the other day. The first time last year I went to Leeds, and I thought, oh, I've been to the north before, I've been to Peterborough. And they told me that Peterborough is not the north. And I started my educational journey to understand that there's a lot to be learned both ways. So we tend to focus on, we have a lot to teach you, come to us and we'll teach you. But there's so much to be learned from going to these other ecosystems as well. Because I can tell you they've got some incredible things happening up north and east of England, southwest, etc. So we've got to make sure that we're also traveling and learning and vice versa. It's not just that we've got the monopoly in the Golden Triangle on how to do things best. We are very fortunate to have access to capital and experience, but we have a lot to learn as well. So we've got to make sure that we've got that mutual exchange.
Well, that's true, and of course they've got Northern Gritstone
as
well now. So the capital's there as well.
Yeah. Times are changing.
So how do you see as a group of experienced colleagues about the role of the private sector versus the public sector in designing and setting up accelerators? I know it's probably public-private partnerships are often the best way to go, but should accelerators ever be conceived to try to be profitable, or is this something we're doing for the public good? So that's a really quiet, isn't it?
Well, I mean, I can speak because in some ways it's quite easy for us because we're BBSRC funded and we're measured on impact. We're not measured on return on investment. Right. So actually we are set up to offer non-dilutive capital and actually not to make money. And our key performance indicators are different. They are about impact to patients. They're about actually a company going to start Codon or KQ Labs or Stevenage or whatever. They're about that next step. So, I think the public funding enables you to take a few more risks. Mm-hmm. It enables you to invest in people when the idea is not necessarily going to make a lot of investment, but that person will be successful, but maybe in their second or third company. Yeah. So So I think we use some of our profits and we use some BBSRC money to do that. So there's definitely a role for the public sector.
Great. Jason?
I think you should definitely can and have both. I think it's very important because, just from the example on the stage, we have all worked with either public sources of funding like Innovate UK or LifeArc as a charity working with private organizations as well. I think it's so powerful when the two sectors combine and you get the best of both worlds. I am reluctant to say that one can do it on their own. There's a study, I know we were discussing previously, a study by Bohurst, which I hope I'm not misciting, but there's one figure in particular that I like to bring up. They did a study of about 180 different accelerators. This is probably mid, 2015, 2016. Over 180 accelerators across the UK. And they looked at performance of companies who'd been through them on just two metrics: the time it took to raise capital, venture capital, and how much they raised. And also the third one was at what valuation. And they found that at the top were accelerator programs that had venture backing or partners. Then the next tier of companies were those who had no accelerator help whatsoever. They just did it on their own. And the lowest tier were companies that had gone to an accelerator that did not have those kind of partnerships. So you were actually worse off going to a underperforming accelerator than if you had just done it yourself. Simple math, I'm sure there's no nuance to it, but it does highlight the importance of having the right partnerships and right people with the experience, domain expertise. It's not enough just to say, here's some space, here's some mentors. You need guidance of people who've done it to really help you navigate through.
Yeah.
Yeah, I think that's very interesting, actually. So KQ Lab's first year of funding was Innovate UK. Then we had actually the Wellcome Trust fund us for a year, and then LifeArc have funded us for the next 4 years. I think there is a real role for the third sector, actually, for the charities in this, as well as public and private. Don't know how you define that, but third sector, I think we call it. Yeah. Yes. So I think that's right. That's really interesting. I think a mixed model is probably the best. But I think, you don't necessarily have to have venture backing to have a venture backing mentality. I think if you have the right connections and the right contacts and the right way of running it, I don't think it necessarily has to be money coming from them. Yeah. But I agree. I think you have to have that leaning.
I agree. I think it's the mindset. But even if you don't have financial backing from them, you need to have partners, in my opinion, in the mix who can bring that other perspective. Because you could imagine a world where it's completely backed by a charity or by public funding, but at the demo day, for the Mentors Network, for the building, make sure you've got mentality. Yeah, exactly. You've got to have the mentality. Yeah.
And I should say that 30% of our funding does come from AstraZeneca, ASI, LifeArc, and Kidney Research UK. So it's a really nice mix of funders. Yeah.
Marvellous. So we've done our hard work. I'd like to throw it out to the audience. So I think the real question is, what do companies need? If you're in the audience, you'd like our colleagues here to consider ways of doing things that you'd like to see improved, or new offerings, or any questions you have. We have a colleague down here at the front in the second row.
Thank you very much. Is this on?
No. Number 2. Shall we try again?
Hello.
Or just speak
just
loud.
shout.
Just shout.
Can we try number 1?
So my name is Sally for Science. I'm Chief Executive of Stevenage Bioscience Catalyst. I don't shout in this one. Okay.
Brilliant.
And just to say,
great to hear the fantastic support that we've got from our accelerators here in the UK. And I think we all agree that the UK is fantastic for its science, and it's great with its startups and growing them on. I was looking at some of the information from the Centre for Entrepreneurship in Cambridge, and the stats showed that we're great at those earlier stages, and that the companies, 90% get to prototype, but then after that, only 10% get to full commercialization. Does the panel see that more support is required in that scale-up stage? And if so, what kind of things, answering the question there, is what do the companies need? And the reason I ask is because we're looking to set up an accelerator program that takes from prototype to commercialization.
Great question.
Well, I think it is a great question, actually. I mean, I think that is they do have a different set of needs, the companies that are going through scale up. And I mean, we follow our companies. As I think you said, it's not that they come in and they're done. We follow them and we support them, and some of our companies have now just done their Series A, for instance. So yes, it's a different skill set and it's a different mindset. I think going through that, especially for a more sort of a tech-based company, going through that very rapid expansion process is a completely different experience to actually building the first steps of a company. So I think yes, I think actually making sure that they speak to people who've been there and done it is absolutely crucially important, and to have support from those. And of course, you do need the money. But yes, yeah. So I think support from experienced people and money, basically.
Yeah, I mean, I was going to say funding, peer network and space. And I think one of the things that we've been trying to do, and I'm pretty sure Stephen is involved in this as well, is, when CEOs come off these accelerator programs, what do they do? Where do they go? Where's their network after that, apart from that one they've met? Where's their peer network? So we kind of created this founders growth community for people to move into. There's Life Science Org as well, which is another good organisation. It's the same thing of just making sure it's almost like you've still got that cohort feel, and there's enough of them to be able to say, I'm struggling with this particular problem, can I find someone else who's been through it? So I don't think we have enough people in our own clusters to provide that support. I think that is where we need to work together to give the support to those companies that are scaling up, because there's not enough people that have done it. And I think space is a key one, that growth, that flexibility, the flexibility to get bigger and then a bit smaller and then actually have people off site. And it's really, really, really important at that stage.
Yeah, I would say two quick things. I'll give a plug for our friend Uday Patki, who's got the Scale Up Academy with Health Innovation East, and it's about medtech companies trying to scale up. And so it's a really great framework to teach them that next level, but we need more of that. So it's great that you're setting that up, Sally Ann. I would also say, just to build on what Catherine and Barbara said, I think it's so important to people. We need to make sure, whether they are new to the UK or they're expats who've been abroad or have done this, that they're coming back and that there is a path for us to leverage their expertise. And I think there's a huge disconnect between all the amazing people who have grown and scaled companies, who'd happily support these companies to get across that chasm, but there's just not really the joining up the dots. And we need to make sure that we facilitate that. So whatever you set up, or anybody who's listening, make sure, to Barbara's point earlier as well, talent and people is everything, and creating a smooth path for them to be on those boards or operational roles in those companies.
Yeah, and I just think maybe the triple chasm model is something that lays out different skill sets, different challenges as you go from invention through to sort of global commercialization. So I think at LifeArc we're really aware that a lot of the barriers to patient impact can be on the commercial side. It can be on the pull mechanisms and market failures that we're aware of, from antimicrobial resistance to people not seeing the commercial incentives to develop novel antibiotics, or rare diseases, or childhood cancer, where there's very small patient numbers. So absolutely, the skill set moves to pricing, reimbursement, health technology assessments, regulatory affairs, and they're just as real barriers. And how to crack the NHS, I think that's been referred to earlier on, people wanting to sell into the world's biggest healthcare system that's right on our doorstep. How do you do that? So if SBC is able to further support and invest in this commercialization set of skills, then I think that will be a real magnet for many people, hopefully to stay in the UK and build scalable businesses. Thanks so much, really.
I mean, actually,
using the triple chasm model, and all the accelerator programs that we've
on and others runs tend to go to what's called CHASM 1, which
Yeah.
is prototype stage. So we're working with your guys. Yes, we are indeed working on the triple CHASM
Yeah.
model.
Yeah.
CHASM 1 being prototype stage, which
So where we
traditionally accelerator programs run to.
Yeah.
So it's a delight to work with you guys because then the stage 1 accelerator programs feed into our program, and we're built around a commercial core, which is very much our focus.
Yeah. And just
So
to
it's good
clarify,
to see that
none of us get commission from Uday.
You should talk to
I'm missing out. Okay, I need to get on that, but it is a great model.
Yeah, I mean, just to pick up on the point that you made, Jason, about commercialization, I mean, I think that's really important. I mean, I think one of the things that I think we do is we try and encourage our companies to look globally and actually not to just focus on the NHS, to be honest. I mean, I think the NHS is great in many ways, but it can be the death of companies if they just keep banging their heads against a brick wall and can't get adoption. So we really encourage them to actually look at We're not saying move to the US. You can be very successful in the US without actually relocating there, but it is a huge market, and we really encourage companies to look at that in some depth very early, which will help them scale.
Can I make a quick NHS comment? So, Barbara, I completely agree with you. I think what we need, though, is a new deal with the NHS when it comes to innovation in the country, because I find that you're right. If you're trying to push in and sell in, God, it's a hill. But if you find a way to be collaborative, maybe even get access to the data on an individual trust level, some of our companies have had a lot of success of saying, we're not trying to sell you anything. What we want is to validate our platform. We want access to your data. We want access to your expertise. And if we can start to think more about that might be the better way to get into the NHS to start off, because if you're going in with a hard sell, the barriers come up. And if you put that in a pitch deck, I guarantee you investors go, NHS is your go-to-market strategy? Really? But if you say, actually, we're collaborating, we're getting access to markets, we're doing investigator-led clinical trials so we can validate our approach, then it's a different conversation. So it feels like we need a kind of a reset of how we all think about the NHS, not as a grumpy thing we have to convince to buy our product, but maybe as a collaborator who can help us penetrate other markets and maybe be more of a partner.
I mean, I think you're right. And I mean, certainly a lot of our companies have been quite successful in doing feasibility studies, pilots with the NHS. That's great. They get fantastic data, but then they can go and use that data somewhere else.
Exactly.
And that breaks my heart in many ways.
I know, it is sad,
because actually it shouldn't be like that. But, the reality of it is that, unfortunately, that's actually quite a good path for them. So true.
Awesome. Any more questions for the panel? We have 3 whole minutes. Hi, Richard.
Hi, thank you. Is that working? Yes. Richard Hebden, Director of Health and Life Sciences, Innovate UK. Fascinating panel discussion. I can identify with a lot of the issues and themes you've raised. Innovate UK, of course, has been very much involved in accelerators or accelerator-like things for a few years now, working with Crick on KQ Labs, for example, the Audley Park Oncology Development Programme, and more recently our Biomedical Catalyst Accelerators Programme as well. Although I think we probably need a new name other than accelerators, because even within Innovate UK, there are other things called accelerators which are not the kinds of things we're talking about today. So if I may, two questions. One, in terms of definitions, when is an accelerator not an accelerator? And perhaps might be something else, say like an incubator. And secondly, given the emphasis on domain expertise and the importance of bringing in that right domain expertise, is it the case that actually accelerators tend to work best when they're thematic, i.e., an oncology accelerator or dementia therapeutics accelerator and so forth? But interested in your views on those questions. Thank you.
Who'd like to take that?
I mean, I can do the first bit if you want first, because I think an accelerator is something that has a proactive intervention to do something, and that is measurable. So it's something that shows it's either going from commercialization readiness level 1 to 3 if it's the triple chasm, or it's technology readiness, whatever, but you can measure it, and it's a really proactive intervention. That's what I would say an accelerator is, but it doesn't really matter what stage the company is, but it's doing something that accelerates. And an incubator is literally you go in there, you rent the space. That's my view on that.
Yeah, no, I would completely agree. I think you've summarised it perfectly. Yes, I mean, I think on the other point, which is the thematic point, yes, I mean, I think that's a very interesting point. And I mean, obviously, we do believe it to some extent, but data-driven health is it's specialised, but it's still quite broad. But even within those 10 companies, we find that there are very different areas. So I think we have to customize within that. And that's what we tend to do is we have a broad, well, actually, yeah, by most people's standards, we have quite a focused program. But even within that, we customize within that. Because I think that's where the value comes. It's actually when you get into the detail. The question when you choose themes for accelerators is how broad, how focused. Very, very difficult to gauge that exactly right, I think. Yeah. But yeah.
And I think if there's a physical aspect to it, it's really, really important to have some kind of theme that the companies that are based there are actually developing something similar, because it's the same kit you then need, whether it be flow cytometry or whatever. So for us, it's therapeutics, which is so broad, because it could be neuro-oncology or whatever, but it's still therapeutics, so antibody development or small molecule development, for instance.
Brilliant. Well, thanks to our colleague who's shown us that the next panel is coming. So I'm very much looking forward to handing over to my colleague Chris Baker for the next session. But let me just thank Catherine, Barbara, Jason, and a round of applause. Very interesting discussion. Thank you.
I suddenly
Great.
turned up with a
Okay.
very weird Scottish accent.
Really?
That was so bizarre. I was like,
Really?
It's-
Ladies and gentlemen, save your words. So, welcome to the final panel of the Biotech Ventures session, the next generation of university spin-outs. Unfortunately, we have run out of all our star quality chairs for the sessions, so I'm guessing the people in charge decided to have a bit of fun with this one. With the title of What is the Recipe for Success, they've chosen me, Chris Baker, to run the session. Now, fortunately, I'm investment principal within the Ventures team, and I have 10 years knowledge and work within the university tech transfer sector before I joined the venture trade. And I've had the pleasure of working with the majority of the people here or their organizations over that time. And I would like to just do a brief introduction for each of them, and then we'll get cracking, and hopefully, time allowing, because I know we have a coffee break at the end of this, we'll have about 5 minutes of questions towards the end. So, starting, representing Oxford, we have Katja, who's a senior partner and leader of the life science team at OSE, or Oxford Science Enterprises. She used to be at Alta Partners and Capricorn Partners, and is the past vice chair of Invest Europe. Now, we have a dark side or dark blue, you have to have a light blue. So representing Cambridge, we have Anne, who is a partner at Cambridge Innovation Capital, having previously been at Advent France Biotech, now known as AdBio, and also Sopranova Partners. Prior to that, she had a business development role at Cancer Research Technology. I did like that place. And now called Cancer Research Horizons, and trained as a medicinal chemist. And as we've found, the Golden Triangle is very important. So where there is Oxford, where there is Cambridge, you must have London. So we have Simon, who is a partner at Albion and leads the UCL Technology Fund's life science team. Prior to that, he was at Goldman Sachs in Melbourne and is a neuroscientist by training. And then finally, as you'll notice, we had one extra chair delivered for this session, and that's because we're going north, not to Peterborough, not to Leeds. We're going right up there, and someone who has the largest remit, I would say, of everyone here for the whole of Scotland. Anne, who is Director of Portfolio Management at Seed Stage Impact Investor EOS Advisory. Prior to that, she was Deputy Director of the Tech Transfer Office at the University of Dundee. Originally, she was a biochemist. Now, what I'd like to start in this session is start on something positive here that we've gone for. So I'd like to hear from the panel. What's changed in the last 5 to 10 years, which has led to actually quite a large amount of success? There have been much larger, there's been IPO'd companies coming out from the UK. What things have happened in the ecosystem that have led to that? And I'll start with you, Katja.
Thank you. I'm glad we're not talking about rowing, because when you started just making those parallels, I think the level of sophistication increased, and it's great on all levels. And as the industry matured, we have people now who have been through a cycle, and positive or negative, but you learn from each cycle. You have successful entrepreneurs who are more active in the ecosystem, and you have people coming back. So as Jason on the previous panel mentioned, talent. I mean, talent is everything. So you're a baker, so you know more about recipes. But I think that without talent, you cannot gel. So having more talented people with more experience in the ecosystem, I think that it's a great move forward.
Now, is this an Oxford-centric thing, or is this similar, or has there been something different that has happened in the Cambridge ecosystem?
Feels like we are pitted up against each other here. So let me carry on with the baking theme rather than... Sorry, Chris. I think it's quite important to recognize that a lot of these ecosystems have matured, as Katja was mentioning, and it starts right at the beginning. I think it starts with researchers, with great PIs, with their tech transfer, and then with the rest of the ecosystem. So bringing the talent, bringing the financing capability, bringing also the advisory capability and the services around that, and making sure, as you were saying, we've got a way of growing and scaling up. So I don't think it's necessarily Oxford or Cambridge or London, as much as you'd like to have some point scoring here, or Scotland. We've seen great spin-outs coming from all of these different places, but what matters, I think, in each of them is the critical mass. You do need a particular ecosystem, and as part of the ecosystem, you need a critical mass for people to learn from each other, to intertwine, to bring particular talent, opportunity, value, capital. And I think that's what we've seen over the last 5 years. It's a maturing of the ecosystem as a whole.
Fantastic. Now I'm going to try an and-to-and transition here within this. Scotland, is there the critical mass?
It's very different up north. Is that working okay? Can you hear? So is there critical mass? I mean, yes and no. So, I mean, Scotland's pretty high performing in terms of spin-outs. And, you know, that's against the background of, you know, really excellent research. So to some extent, we do. You know, it's probably the highest performing region outside the Golden Triangle. Normally in all the reports, it's, you know, it's kind of the fourth. But is it enough? Probably not. You know, if you look at what, in terms of number of transactions, then Scotland performs very high. But if you look at the amount raised per company, then it's lower than Golden Triangle. And we do lose companies. Like Catherine mentioned, you know, it was quite hard to get spin-outs to move from their originating university. We actually find it quite easy. And I don't think it's just the weather. I think the cluster has got something to do with it. So I think, you know, it's a high-performing region, and, you know, we are being positive, don't want to be negative, very high-performing. We've had some amazing results, you know, there's things like Excientia from Dundee, you know, one of the biggest exits from a spin-out in the UK, and it's really important to have those examples, but I think there's still a lot of improvement that needs to be done for us to be able to keep the companies in the different regions. So it's probably different in the central belt, you know, Glasgow, Edinburgh, but places like Dundee, you know, you can lose companies actually quite easily.
Fair enough.
Which makes it difficult then to, you know, keep new ones there. But, I mean, there's a lot of work going on to sort that. So it's kind of a yes or no question. Yes, no answer, I mean, sorry.
Fair enough. So Simon, do you agree? Is it all just been plain sailing, more critical mass, that's why we're doing so well? Has there been anything else?
I mean, I think critical mass and the various things we've been talking about in terms of talent, capital, you know, the things in the cake. I think we have to think about the oven as well, and you know, and actually taking a bit of a macro view that 2013 to 2019 was a period of enormous capital raising by biotech VCs. Vast amounts were raised, and what happened in 2020 was it turned into a bubble. You know, COVID came along, biotech will save us, and a lot of people in the public markets, generalist investors beyond the specialist healthcare investors, made a lot of investments. And I guess the big issue in the room is actually the volume on that oven, which I think of as interest rates. And ultimately, you look at interest rates and you look at biotech markets, and they are absolutely inversely correlated. Inflation expectations in the last few weeks have stayed high, and the biotech markets have come right back down again. And so I think there's been a big benefit for the UK ecosystem over the last 10 years in terms of that capital being raised and coming in, going through that first cycle that you mentioned, Katja, and people coming out of that first, whatever it was, 2013 to 2019 cycle, and now coming back in again, having kind of been there and done that. So I think that's been of great benefit. The flip side is now we're in an environment where interest rates are high, and that's been very problematic, not only for public markets, but obviously for private companies raising as well.
Great. Now, as mentioned in the last sector, and I want to last session, I want to make the same point here. People. So we've mentioned people being recycling around, for better or worse. Usually they've learned a bit or failed a bit and learned a bit more. But what about bringing through the next generation? Has there been any changes in your ecosystems rather than just hoping to get lucky, shall we say, that someone is super entrepreneurial? I'd like to take it first.
Happy to start first, and I perhaps can speak about Cambridge and the broader Cambridge region, which cheekily will include Stevenage and Norwich. But you'll forgive me, I'm French, so my geography is not great. Just thinking about the ecosystem in general, as you were saying, people are key, and that starts from the start. I have certainly seen a difference in terms of aspiration, entrepreneurial aspirations, of the much younger generation of scientists. You know, PhDs, postdocs, certainly are beginning to see companies doing very well. They realize that you don't have to be grey-haired to start a company. You may need some help and some mentoring and getting the right people around you, but that's not the barrier that it was 20 years ago or even 10 years ago. So I think there's been a lot of work done, certainly in our ecosystem within the Cambridge region, to provide at least some training. There's still a lot to be done, of course, but it's a start. there are various organizations coming together to provide either roundtables or formal training or other ways of explaining what the whole entrepreneurial journey looks like, and hopefully tease a little bit those who have got that in them to perhaps make the jump a bit earlier than they would have otherwise. And I think it goes all the way through, right? Having people who have been there, done that, and can become mentors, that will help elevate the next generation. So I think we have certainly seen that a lot more. And within Cambridge, life sciences, of course, is very strong, but so is deep tech. And they feed from each other. And of course, at the intersection, you've got what people may call tech bio, or you may have other names for it. But I think the two ecosystems where you had perhaps a larger entrepreneurial proportion of people going into the tech side are now feeding into the whole life science. And I think that's been very positive, that they learn from each other, of course. Some things are different, but there are also some similarities. So little by little, we see that more. And I think that's encouraged by tech transfer offices, by investors like we are. At CIC, we're very engaged within our community. So we take part in educational programs, in accelerators, and you've seen the panel before, so I won't reiterate, but having these particular accelerators and capabilities, which mean people don't go it alone, enables that next generation to come up and take that step.
I think, though, that there's a balance to be struck, and I think the universities themselves can and are, thankfully, doing more to kind of help those young people, because on the one hand, you want people, particularly early in their career, to see the possibilities of the impact that they can have, and recognising that government and philanthropy can't always fund those things right through to commercialisation, and so the private sector is going to be involved in those. At the same time, as universities, I think we want to keep our fantastic young people in that ecosystem as well, to be training the next generation afterwards. And so I think it's one step that people definitely have that greater aspiration now. But I think it's definitely incumbent on us within those universities to be helping those people think, okay, you could actually still be doing a bit of basic research, you could work in your spin out, we want to keep you within this ecosystem. And I think it's building those sorts of ecosystems. As we've talked about the Golden Triangle and other areas, but it is very fundamentally very attractive to be in a place where you've got lots of people around you. And I think we can still do more to kind of keep people in those kind of in those places, and to provide for a career path. Particularly, people talk about the post-docalypse, and that there's this great challenge for people in postdoc roles to be able to then go and get tenured positions. And I think that we can and are. Increasingly, but I think we can do more, creating an environment where people can kind of pursue those sorts of combined careers. And I think we should be encouraging that for sure.
Yeah, absolutely. It's no longer a stigma to leave academia for industry. Before a postdoc was not getting a tenure position someplace, he was probably considered to be a failure by some. It's no longer the case. One of the companies we have co-invested with LifeArc was DGS Antibodies. It was started by 2 postdocs. I mean, the guys had such a limited budget that they were sharing one bench 24/7, taking turns, and they didn't have enough money to buy lab-grade ice, so they were buying a green peas from Tesco. And a couple of years ago, they sold their company for a very substantial amount of money to AbbVie. And we can see now that, in the Oxford ecosystem, David and Joe, DGS, David and Joe, they were, they're considered to be role models. People are. It's not about so much about even making money, about starting something that becomes substantial. Now we have an AbbVie site in Oxford with a representative of AbbVie Ventures being there full time, etc. So people are inspired by example, and you can see you can take something from the lab, start a company, and it can fill the pipeline of a pharma company and hopefully bring a drug on the market.
I think things like iCare have been great as well for training of postdocs. So, we've, it's not public yet, but we've just invested in a company that, the CEO came out of the iCare program, and, the amount of industry engagement they got through that, was convincing enough for us to come in. It was really good. We've got other training programmes in Scotland. There's one called Converge, which is Scotland's national business plan competition, but it has a load of training along with it, which I think has really, really helped. And we've got a good non-dilutive programme called Scottish Enterprise Programme, or Economic Development Agency, called High Growth Spin-Out Programme, that again trains people through the process. So I think there's been a lot that has helped.
Now, having gone from the positives, actually no, and this could still be positive. First of all, for the audience, could you raise your hand if you've heard of the Use It guide, which came out last March, I think, March, April time? Okay, now of those people with their hands, could you keep your hand up or put your hand back up if you've used that guide in earnest at any point? So that's about 8%, I reckon. So for the panel, judging by that, not everything was useful within the Use It guide and being used on a regular basis. But was there anything which, when you saw it, you went, actually, yes, that is something that we would like to see done more, that could improve the ecosystem a bit further?
I'm happy to start. One thing I really like about it is that it talks about, the deal as a whole, because I think that's really important. So it's not, I don't think it's been that helpful to just fixate on the equity stake. You have to look at the deal as a whole. And I like that about it. It's really got to balance out, and it's got to work for the business plan of the company. I quite like the fact it's not too prescriptive. So I think it's that part. It's look at the deal as a whole. Don't fixate on one part, because
I couldn't agree more. And in fact, I think when as investors, we're trying to solve a problem for founders, for the university, for our LPs in the fund, and ultimately you will end up coming up with something that is, I guess, minimally dissatisfactory to everybody. And if you come up with that, you've come up with something good. But that includes the university and the TTO, and you've got to look at that in the round. A TTO, typically, certainly for pure biotech, it's different for tech bio and for software, but for pure biotech, a TTO's equity share, by the time those companies have raised hundreds of millions, billions, is going to be so diluted out. There is a fair position to be had that they should have milestones or royalties. And so I agree with you absolutely. You've got to look at it in the round. I also think, because each technology is different and each deal is different and each set of investors is different, I think we do tend to fixate a little bit on what the seed round looks like. And I guarantee you, once you get to Series A, again, I'm talking particularly in biotechnology, once you get to Series A, they do generally look relatively similar despite what happened at the seed round. And so I think we should be solving what happens after a proper institutional Series A, rather than thinking about, the tech transfer office got this, or the founders got that, or somebody took too big a stake. I think we should focus on the long term and where this company's going to in the long term.
Yeah, there's a particular part I really liked about it, is the fact that these 10 universities got together, but they also involved investors in that process. We were one of those who were, I think you were as well, Katja. And what I really liked was the philosophy of it. The idea being that we have something that can help TTOs feel that they're on the right track. We know every deal is different. We know every case is different. However, there are some common themes here. And if we can shortcut in some ways the negotiation time to spin out some really good technology from a university, from a research institute, into a spin-out company, then I think we've achieved something really important. We have seen several cases where certain TTOs are blessed with having a larger volume of startups, so they don't reinvent the wheel every time. And there's some learnings, if the team's stable, that they can capitalize on, and hopefully negotiations are not too long. But we have also other TTOs where you may not do that many spin-outs. I think we heard from Jason, during his travels, that he's found some great IP in various places, but sometimes this great IP just remains within the university because nobody takes it out and decides that this is a commercial proposition once it's been thought through and there really is a business there. And for these TTOs, it is really hard because you may do one every 2 years, 3 years. So you may not have the corporate knowledge, you may not have the benchmarks. And I think these guys provide exactly that. It's am I on the right ballpark? Is this thing taking too long? Am I negotiating something which, in the grand scheme of things, really just doesn't matter? And I think that part of spinning out the technologies out of universities and research institutes can go really, really well, but at the same time, it can end up being quite painful at either end, because people are negotiating things which, again, take a lot of time, effort, and then you end up with something a bit dissatisfactory where everyone has worked so hard on getting something out, instead of having the excitement of, hey, new company, let's get started. It's like, that was a hard slog. And now the harder work even to make it work. So if we can have a guide, it's not the answer to everything, but if we can have a guide that helps shortcut that, make it a bit more homogeneous, and for some TTOs it's helpful, right? Negotiating with founders, knowing they're on the right tracks. It's done by a third party, so there's no emotions involved. I think that's a great thing.
And I guess getting a slight segue here, do you think venture firms understand the difficulties sometimes of pulling together all the IP? Because you said the slog, it's something I hear a lot, the time, I'll never go back there again. What has been done about that so there are fewer grumbles? There's always going to be grumbles. We grumble a lot in venture. But fundamentally, what is being done or is hopefully going to be done to improve the efficiency beyond maybe just telling them, look, it's just not so simple?
Well, luckily, I think that in Cambridge and in Oxford, we are very often at the origin of the creation of the companies. Not all of them, but a very large part. And there is a certain level of patience within Oxford Science Enterprises, and we know that it takes a long time, and we know we have to be patient. And by the way, some of the academic founders are even more impatient than we are in some instances. And then I would say that probably 90% of the companies we start are too early for more traditional investors. So that's why we like to co-invest with people who have a longer horizon, like LifeArc, occasionally some corporate investors. And hopefully this painful step of company creation with university IP is less painful for traditional VCs, I would like to think. But some of them are very skilled as well, and I know some are in the room and participated in the creation of companies. But I think that a traditional venture fund cannot do only company creation from the universities. It will be just too time-consuming.
And then maybe changing gears a bit here, of quality versus quantity. Something I used to get is how many patents you've filed this year, how many spins you get. Those kind of metrics versus actually true quality. Where, or is every opportunity a spin-out? Let's keep it as simple as that. And what needs to be done to get the quality of the spin-outs coming out of universities to the next level, do you think? I'm hinting at funding is what I'm going to go for here. I'm going to allow you to go down that route.
Okay, so I don't think everything should be a spin-out. Definitely not. I mean, a lot of the universities in Scotland have got like a stage gate mechanism so that, there's a quality threshold or suitability threshold they have to pass through about whether it's a spin-out or a license. So I think that's really important. I think, somebody talked earlier, I think it was maybe Jason, about that gap, the translational funding. I think that's missing a lot. That's the thing that could make certain technologies a lot more investable. It's that go away and do X, Y, and Z, but there isn't anything to fund that X, Y, and Z. So I think that's key. Forgotten the question now, Chris. Sorry, what was it?
Well, on that, I'll give you a chance now. I've not allowed you to plug because I did your intros and everything.
Yeah.
What sort of translational funding do you have for those people who go, like it, but not ready yet?
Yeah.
You need to do X. What sort of scale are we looking at here? Because I think we had 250K, a bit of lab space, some people run around and tell you what to do.
Well, we put in place a program called Uncover, and it's a convertible loan of up to £1 million that we can give to a PI in a lab, and to perform some key experiments. And if they succeed, the loan converts into equity. Well, if it fails, it's a write-off. But what's very important here is to really guide the academics. So we come up with a plan of what has to be achieved, what experiments should be performed, very focused on potential translation. So we put several of those already in place in Oxford, and we'll see how it plays out. But I think that academics are quite open to it. And maybe it's more acceptable for people who are not necessarily interested in getting involved into a company or starting a company, but are missing this funding or a piece to get to the next level, and they might view it as a supplement to their research funding, and so it's kind of a win-win situation.
So 1 million convertible.
Up to 1 million.
Sorry, up to 1 million.
Well, but sometimes you need. We don't want to put too much money when there is no need, right? So I think that we put as little as 300. So it all depends on the plan.
Simon?
Can I answer that question and your previous question at the same time?
By all means.
Because to me, one of the most important things I think that we do as university investors is engage with academics. And building trusting relationships is the fundamental underpinning of what it is that we do. Often investment money is seen as very different from grant money. It's not something that a lot of academics have experience with. It can be quite scary. We do end up having a lot of conversations with people saying, here's this group of patients you want to treat. Don't be scared of pharma. They are basically a piece of infrastructure that is going to globalise your therapy for this big patient group. They have a different incentive structure. They want to make a profit. You need to file a patent so that they can protect their market so that they'll then go and treat your patients. Please, please don't put that poster up at the conference next week before you've come and spoken to somebody and we've done something about that, because then your idea will not get to those patients, and that just fundamentally is the process. And so there's an education piece, and I think some of that can come with sort of translational funding as well. The UCL Technology Fund, and recognising kind of the discussion before, not everything needs to be a spin-out. There's a lot of things where, you could just do that experiment for 300K, 500K, and the TTO will get a license out of that. And it could be a perfectly acceptable license. And in fact, I would say upward, certainly more than 3/4 of, commercialisation monies that TTOs make are from licenses rather than from equity stakes, certainly in life sciences. And so we provide, for example, essentially a cash advance. It's basically, to all intents and purposes, looks, smells, tastes like a grant. But it's up to 2 or 3 million. So it's a decent-sized translational grant where the exit from a venture perspective is actually a share of upfronts, milestones, royalties, much in the same way that you would get a grant from Cancer Research UK or the British Heart Foundation or anybody else. And really there, I would frankly rather spend, no offence to anybody, 200K on the mice than I would on the lawyers to set a company up for something that's going to cost maybe 300 or 500K to get to a point where it's a licensable program, and then you're done. I think there are other alternative ways, kind of like a CLN, same sort of thing. If it fails, we all walk away. That's the end of it. And we haven't gone to all of that time and trouble. I think even with I think the UCITS guide is fantastic in terms of helping to speed some of these processes up. It is still a difficult process, and often for a lot of academics, if the first time you meet them, the first thing you give them is a cap table, it can be really quite scary. So I think having some of these earlier forms of translational funding helps to build those relationships. By the time you get to the spin out, you've actually been working with them for a year or 2 years, and you're all working together and you're all pulling in the same direction.
Perhaps sorry.
So there's probably less. There's similar mechanisms in Scotland, but the amount is less. So, as I mentioned before, we've got a thing called Scottish Enterprise High Growth Spin-Out Programme. It's about, I can't remember the exact amount, it's like 200 to 300K. It's very rigorous, which can be a total pain for the academics. But for us, we know it's been through a really rigorous process. They do kind of pseudo board meetings, and it's just geared up to get technologies ready for spin-out. And it pulls together a team as well, so it brings in a commercial champion. I think that's something probably that universities don't focus on enough. It's like there's too much focus on the technology rather than making a company, rather than bringing in people. So it helps with that kind of quality. So there's something that looks much more like a company at the end of it. But the volume of cash is obviously a lot less, which is a problem for life science companies.
Just picking up on your question, since you very subtly guided us towards the funding kind of question. I think there's also 2 different parts to consider in terms of timing, and that ties in with your first question about is everything worth spinning out. You have the translational funding, which will help you pre-startup, really figure out whether it is something that, from a technology perspective, can have some legs. It can also be, is that a product, or is it more of a technology that's great to apply, but you won't really have a good route through spin-out because there's not a product coming out of it. And that's really a pre-formation, if if you like, translational financing. And there's a lot of actors playing in that particular field. We both work for Cancer Research UK, so we know CI UK, we know LifeArc, we know others have got this particular type of financing which is available, and I think that's really helpful. And then on the other hand, if the company is already created, you may have different types of experiments you want to do to validate your technology or make a very clear, I wish it was so obvious, but sometimes you have really clear go, no-go experiments you need to finance. It's not very often the case, it's often grey, right? But sometimes you do. And if you have got these, having a little bit of translational funding that goes into the company rather than the project, and that's a very different route, that's really helpful. And we heard about accelerators, we heard about, there's also some charitable funding we've got, particular thematic ones, cystic fibrosis, for example, they will finance quite a lot of either projects or companies. So there are various routes to address that funding question. And I think after that, they may then be ready for more traditional VC financing, if you like.
But Chris, to come back to one of your points about quantity versus quality, and is everything a spin out? As the ecosystem matures, and right now in the OEC portfolio, we have over 120 companies. And obviously it changes somewhat the dynamic and how we had several instances in the last 2 years where we identified interesting IP in Oxford or potentially a potential source of IP where we already had a portfolio company that could take it, in license this IP or start a new program. While if this company wouldn't be around, we probably would have started a company. So I think that it's also about growing companies to a certain scale, utilize certain talent pool, because once again, huge question, talent. So if you have already a team in place, talented team in place that's performing, why start another company on the similar theme, let's say around, and just another target, but around the same way. It's much more efficient to bring into an existing company if it's run by a competent team.
Very true.
And to bring that talent that we've been talking about, to bear at those early translational phases. Having a proto CEO or governance, company-like governance for a project, for example, is hugely critical. It's not you're not just giving the money to the academic to say you go off and do it. And having fantastic people who've been there, done that, right at those very, very earliest stages, coming in, working directly with academics, building those relationships, I think is going to improve the quality of what comes out at the other end.
Fantastic. Now, happy days, our clock has completely died. So I'm assuming we're almost at time. We'd like to have a question and answer session, even if it is a very short one. So if anyone has any burning questions, please put up. Oh, there's one at the back. That's it. That's what I need. Quick and easy. Because I'm going off my watch now, which I don't know if I've set correctly or not.
Thank you. Hi, in the back over there. So when you're a co-founder of a university spin-out, especially when you're negotiating the licensing deal, you have an intrinsic conflict of interest because you're both the CEO of the company and an employee of the university. And sometimes when the university is being a bit greedy, that can be a bit uncomfortable. I'm asking for a friend. Do you have any advice on how to handle that situation?
Who would like to go on that one?
I would tell your friend that a good thing to do is if you have something like an interim CEO, a CEO, a designate, somebody else you've got in the company or somebody advising you, take them with you, just as a bit of backup and just even things out. I think that's quite normal. Yep.
I'd say being transparent in a negotiation is helpful. It's a negotiation. Different people have different incentive structures and they want to get different things out of it. But simple things like, what is the market that we're actually addressing here? And if people have wildly different expectations of what that market is, then clearly the value that they're going to place on those things are going to be very, very different. So I think at least, in that process, things, agree on the things that you can agree on. So at least the parameters are set, and then you're at least talking in the same ballpark. It's amazing to see when you give a couple of different people different information, the sorts of deals that they come up with are wildly different. And so at least be in the same ballpark. Ultimately, founders, a TTO, a university, the investors are all wanting this to be successful, and it's got to be something particularly for where we sit at seed stage. We're thinking, well, when big tier 1 biotech investors come in and put money into this, it's got to be a deal that actually makes sense and doesn't have to be renegotiated from scratch, you know, when we go out for the next funding round. So if you can at least all agree that you want to come up with something sensible, then at least that sets the playing field for you.
Maybe bring along the UCIT guide.
Fantastic. Any other questions? Internal question at the front. He's maybe got a bad shoulder after that session. He couldn't get his hand all the way up.
Fantastic panel. Thanks so much. What are we still not doing as well as our US cousins in the university spin-out space?
Well-funded, well-resourced TTOs, and there are well-funded, well-resourced TTOs in the UK, and some of them do fantastically well. Others are heavily under-resourced, and I would say even the ones that are the best resourced are still under-resourced. I think we need to do better by our TTOs to make them better. They are already very good, but they can be much better, and it requires funding and it requires fantastic people.
I would follow up on the talent part, having some flexibility for an academic founder, for example, to go out, exploit the idea within the spin-out, and be able to come back to academia. Having that porosity is something that we haven't yet cracked, I don't think, in the UK.
And just one thing to add, having gone to the autumn conference last year, certain TTOs, certain universities, which are 10 times bigger, have people just dropping $1 billion individually into their translational or pre-spin-out grants. That does make a big difference. You can make a lot more mistakes. You can hire a lot more people to grow and get that infrastructure. But one of the key things we did here is that people could go out from academia and come back in afterwards, which is starting, I would say, to appear in this country. And with that, any other last burning questions? Because I know we are coming up into a coffee break, and there's too much talk about baking at the beginning there. And everybody who knows me knows that I like to have it. So, with that, I'd like to wrap up to say the next session starts at 3:30. We've got some fantastic early stage companies who are going to be pitching for about 8 minutes each. But before you manage to leave the room, even if you got your back to us, please can I ask you to put your hands together, say thank you to the panel.
Thank you, Chris.
Good afternoon, everyone. Really happy to welcome you to the Biotech Spotlight. I think for me this is probably the most exciting session of the LifeArc Translational Sciences Summit. We've heard a lot of talks about what's going on in the industry and themes. However, today we've got 7 companies, 7 CEOs who are really doing it in real world, developing products, developing diagnostics tools, hiring people, meeting with regulators and partners. So really pleased to introduce Ola Władek from Constructive Bio.
Good afternoon, everyone. Oh, good afternoon, everyone. I was getting better. So at Constructive Bio, we write entire genomes from scratch in order to create fully programmable biomolecules of the future. And why I'm here, it's because LifeArc and MRC have been instrumental for the company creation. We are spin out from Jason Shin's lab, and Jason is an incredibly active academic working in Cambridge, UK. For the last 20 years, Jason has published 178 papers, of which 16 has been in high impact factor periodicals. Last 5 years were perhaps most attractive in his career when his research started moving from a very foundational one into something that could be applicable and what created Constructive. So I want to highlight here 3 things in terms of the scientific curriculum. In 2019, Jason's team was the first one in the world who created a fully synthetic genome with recoded genetic code. So they have designed the genome in silico, they've kicked it off in the test tube, and then created a living organism of industrial quality that we have taken into Constructive as our workhorse. In 2021, Jason's team have moved a step further, and they have shown that you can reprogram and add new chemistries to the natural toolkit of what's programmable and can be made in the cell. That was a moment when Jason's team was approached by venture capitalists and Constructive story started. It took a year to spin out that technology into Constructive Bio. But we have been working since with LifeArc, and Jason has been publishing since as an academic, providing some further fantastic papers. I just want to highlight the recent one from January when he has put in new, very exciting chemistries in a programmable fashion into a cell for a ribosome. These are very, very exotic building blocks. At LifeArc, I want to today, as we celebrate LifeArc at this event, to highlight 2 people who have been absolutely fantastic. Jason here, always very helpful to us with advice, and Laura Stennett, who has been on the partnership teams and has made our life incredibly easy, but also I really respect her respect to the science that has been made on taxpayer sterling. So she combines a fantastic attitude to the business with really big respect to how the science is funded by a charity. So where do we start as Constructive is the belief that 60% of physical inputs of economy can be made with biology. But to realize that opportunity, we need to be able to write and program biology in its natural language, so the language of a DNA. And we need to expand drastically the range of chemistries the cell can introduce in a programmable fashion on the ribosome into the biological output. So we believe that combining these advances enable us to program biology to make a very wide range of things. And at Constructive Bio, we respond to that need with our 2 pillars of technology. First, we are able to write entire genomes from scratch. We have in license from Jason Shin's lab the molecular biology toolkit and algorithms that build the intellectual infrastructure for writing really long DNA in silico and then making them in a test tube. And the second, we have a set of tools to expand the range of chemistries in a scalable manner. So to bring in the new exciting chemistries, we, for example, design again in silico what needs to be put into biologics, and then we create the green cell lab biofactories of the future, so the strains that contain this machinery. That sounds simple. In fact, it's a little bit more complex. We have to bring in a number of engineered functionalities into the cell. So we focus on design of new orthogonal tRNAs. We focus on design of the new orthogonal tRNA synthetases. And we also have some ambition for ribosome engineering in the future in order to make those things, even the most complicated things, in a scalable fashion in the cell. So one might ask, why are the non-canonical building blocks useful? First, they bring in functionalities to the proteins that are completely new to nature. So these can be things like a moiety that change Ozempic from a drug that has been administered daily and hasn't been that efficacious in the clinical stage into a blockbuster that Novo is now marketing, which is Ozempic or Wegovy. But there are also more than that. There are antibody-drug conjugates that benefits from a very specific drug-antibody ratio. This can be engineered into biologics with ultimate precision through genetic code expansion. So we develop that range of new chemistries that can be brought into the cells, and we also make products which include those type of chemistries. So what is exciting to me about Constructive that both pillars of technologies provide a very exciting commercial opportunity. We have to date managed to start partnerships with people who are interested in long DNA writing. We managed to start partnerships with people, including a big pharma, who are interested in our platform use, as well as we are in conversations with people who are interested in the very specific molecules with new chemistries. Obviously, the tailwinds of Wegovy and the tailwinds from ADCs are helping us as a company, but the products that we can make with new chemistries are really exciting. What are these products? As I've said, peptides, very exciting class of drugs, but have got the natural liabilities. We overcome them by changing the backbone of what the peptides can be made of, but we also produce them in a green fashion. This is very important to us as a company. So we are very excited about being able to make those things in a cell and not with the solid phase chemistry. I don't know if you guys know, but making 1 ton of Wegovy, if that was possible, solid phase chemistry will create 16 tons of waste, including DMF as a solvent. So we respond to that with proper bioengineering and green cell lab biofactories. ADCs are very attractive to us. We look very favorably at a short chain modalities here. But we are also very excited about very complex protein engineering with molecular assemblies. We call them modular assemblies, and these include immunocytokines and so on. There is also North Star. We are looking at fully programmable biopolymers of the future, which will be transforming industries beyond biopharma, but that's perhaps a long-term plan beyond our IPO that will happen in the future. So I want to just remember 3 things or 4 things about Constructive. We have the world's leading technology thanks to ChinLab discoveries and our further developments. We have fantastic expansive market in front of us that is going to drive our commercial introduction. We have very strong IP moat thanks to LifeArc TTO, fantastic work on protecting the spin-out, the technology that we spin out from the Jason's lab with amazing IP portfolio. And we have now in the company, after a seed round, a world-class expertise, both in the scientific and the commercial team. So those 3 things, as I've just mentioned, I want you to remember. We expand the repertoire of building blocks in the cells. We write entire genomes from scratch, and we are going to make really amazing biomolecules of the future. Thank you.
So we have 1 minute for a question because Ola was so on time. Any questions? No? Brilliant. Thanks so much. Thanks. Great. So now I'm really pleased to introduce Aman Bhatti, CEO of Cumulus Neuroscience. Aman, thank you.
Hi, everybody. Aman Bhatti, as was stated, I'm CEO of Cumulus Neuroscience. Oh, that is not my slide deck. I'll go back. I'll do it anyway. So we're a gene therapy company, healing from within. That's all I got. I'll let you know about Cumulus. I'll use a minute while I can. So Cumulus Neuroscience, we're developing precision diagnostics to enable precision medicine. And LifeArc and DDF came together back in 2020 to really solve the need for how do you go ahead and monitor patients in clinical trials, ensure that you're enrolling the right person in the right trial, stratify patients, and determine right dose, right target, and all. And so Cumulus was created back in 2020 through Dementia Discovery Fund and LifeArc to really answer that with technology. And we work with leading pharmaceutical companies, 5 of which are actually LPs in the DDF, and additional 5 that have joined us as pharma advisors. We work with individuals from the digital biomarker landscape, translational science landscape within these companies within CNS, to, in essence, create a platform that when you're in clinical trials, you do a battery test on people in clinic, and then they go home on drug, and you have no idea what's happening to them on drug. So can we use technology to solve that? So we worked with these 10 companies to create this platform. What it is basically gamified versions of very well-established tasks that can be done at home. We've gamified them so that they could be engaging and allow for people to do this on a bi-weekly basis if needed. You could do it more often or less often if needed. And we also developed an EEG technology because it's not just enough to do do cognitive behavioral tests. In animal studies, for example, you do EEG and then you go into human trials and you're giving them pen and paper tests. So EEG often is the only source of truth. So we had to develop a technology that allows for people to do EEGs on their own. And so we developed this EEG headset that you see here. We actually just got this FDA cleared last year, and it is UKCA marked as well. We've won 3 design awards for it, one of them that we won just a few months ago. And how this works is, in essence, you have a patient that we could ship this to in a kit. They could put on themselves. The app tells them that it's on correctly. And again, we layer on the EEG data while the person is engaging with the tasks. And that's the whole benefit is by creating tasks that not all of them are required, or not all of them require EEG, but we've developed about 14 different biomarkers. We do record voice as well within the platform, so we could do voice analytics. We do plug that into, today we do it with Winterlight, that is now Cambridge Cognition, but we could plug it into any technology that's out there. We've developed all these tasks, again, to really help determine drug target engagement. Is this the right person that you want to include in a study and the like? So we validated this in 2 different trials that the 10 pharmaceutical companies helped us design in Cambridge and Oxford. One trial was in Alzheimer's patients where they used this platform every other week for 6 months, and then followed by another 6 months where they did this monthly. And then we had another trial in ALS and FTD, and again, that was an 8-month trial. Basically just a battery of tests that we put them through to make sure that they can use this. We actually engaged with them when we built the platform, but can they use this, and do we see reliable data, and can we validate that these gamified versions perform as the paper-based tests? You can see here some quick validation data. That's an older healthy patient in one of our Alzheimer's trials. You can see what an Alzheimer's patient looks like, an ALS and an FTD patient look like. All look very similar, but there's some distinct differences, and you wouldn't be able to get these differences if you didn't use a multimodal approach in doing this. So today we actually are generating revenue. We sell our platform to biotechs that are using us in all sorts of phase 1 and phase 2 trials. These obviously contracts provide us with cash, but they also give us data, some validation data, and act as a great incubator for new innovation. And our sales pipeline, we were created for dementia, we were created for neurodegenerative diseases in general, but because we're multimodal, we're getting more and more involved in psychiatry and psychedelics as well, and precision psychiatry being the next frontier in a sense. We actually had a press release with Immune Bio, one of our customers, where they were able to identify a EEG biomarker for one of their Alzheimer's drugs, just based off of patients actually using our EEG technology at home for 6 weeks post-dose. And so they were taking EEGs themselves, and they were able to identify a nice EEG biomarker. We published some data with Sumitomo as well around EEG's use as a biomarker in neuropsychiatric trials. But what we do envision is this platform can grow, as we say, from bench to bedside, being used in early phase development all the way to one day being a companion diagnostic, some facet of it being as a companion diagnostic. But what we're doing in parallel is looking at, after all these years of working with pharma, what we realize is we're finding that we need to find people earlier. We need to find people before they're actually are too late to treat. And so we're using our know-how and our technology to move into healthcare with a project that we have ongoing that is really trying to be able to create a technology that you could use anywhere to diagnose people with Alzheimer's or screen for people with Alzheimer's years before they're symptomatic, in a very simple, easy-to-use platform. We know the current tests today are very laborious for physicians to engage with. Patients, and you have patients that oftentimes are maybe not understand the directions. It takes 15 minutes, maybe 20 minutes to take these paper-based tests with patients, and oftentimes it's only clinicians that can actually assess the patient, right? You can't have the nurse or the medical assistant do it. So they're not really conducive for rapid screening. What we're working on now is a technology where we're using our EEG technology we built for pharma. We're using the iPad that we've developed apps for pharma. That's actually George Stothart that's created this new test called Excel ADX, a simple, very rapid, passive test that will screen for memory impairment. Works very simply where you're just looking at images, we're scanning your EEG, we're looking for a hippocampal response when we repeat certain images, and that is the first thing to go in Alzheimer's is memory impairment, early memory that you see. So, market opportunity there, it could be used by millions to screen millions of patients in the UK and in the US. You could tell I'm not from here, I'm from the US. And we see tens of millions of patients in the US that can have access to this. And we're creating this product to be utilized anywhere, right? So initially we would start in health systems, but you could expand in order to get the TAM. You expand into, you could do it in laboratory services, you could do it even in women's health centers, ophthalmologist offices, and even at home if the patients can't make it there. Provides value and impact across all the 4 Ps of healthcare, but obviously with patients being the critical where they could actually be part of their decision making. And we do see our technology going one day, the entire platform being released where we use one test to screen and then another test to actually determine what type of dementia or what type of disease the person may have. So, just very quickly at a glance, we have product development experience, FDA clearance experience. We're generating revenue. We have moving into healthcare, and we have some recognition and awards, most recently selected as a top innovative company by Fast Company just last month. So if anybody would like to learn more, any pharma folks want to talk about partnerships or leveraging our technology for anything you have, as well as any investors, we're happy to have that conversation over coffee. Thank you.
We've got some time. So, do we have any questions for Aman? Yep, Stephen.
I wondered if your technology can discriminate between the Alzheimer's disease and does your does your technology distinguish between Alzheimer's disease and other dementias, vascular dementia, small vessel disease?
Yeah, so we right now have data that's really related to more of the Alzheimer's, ALS, and FTD. As far as the vascular dementias, we have not studied that or any other cause dementias. But in essence, we hope to get the data points to be able to discern those as well. But we know the majority, obviously, of dementia is from Alzheimer's. One other question?
Not necessarily true. I think statistics between vascular disease, the statistics between vascular disease and the association with Alzheimer's, there's
from that perspective, right?
The combination of those two, I think, is certainly worthwhile looking at.
Yeah, we do have Ruth McKernan, who's a co-founder of the company as well, who's here, who could probably answer the scientific questions. So.
I cannot answer that in 30 seconds. You've got 2 minutes.
Yeah, okay, thanks for that. So yes, there's a lot of things in common. What this technology doesn't do, it doesn't look at blood vessels, it doesn't look at vascular flow, it doesn't look at lymphatics or anything like that. There are some other companies we could talk about that are very interested in that, but the overlap between the vascular dementia and the more classic as we think of it, but not necessarily pathologically classic, there's a lot of overlap there. We're just scratching the surface with this technology, but the promise is huge. Is that good enough?
Better answer than mine. Any other questions?
No? Great. Thanks very much. Thanks, man.
Thank you, Ruth.
So can we just have the next slides up so I don't blindside any people relating? So it gives me real pleasure to introduce Professor Mamoon Azouz, CEO of Blackfin Bio.
Thanks, Jason. So I am the academic founder of Blackfin Bio. Peter Nolan is our interim CEO. So thank you, Jason, and thanks for the invitation. It's a pleasure to be here to give an overview about Blackfin Bio. So We heard quite a lot during this event about the ecosystem, talent, high-quality science infrastructure. So Sheffield is currently a gene therapy hub with world-class research infrastructure and we have been working on this ecosystem for a couple of years now to fill the gaps, and one of the you can see, for example, one of the gaps that we have been successful in achieving is to establish the Sheffield Gene Therapy Innovation and Manufacturing Center, supported by LifeArc and MRC. So we also at Sheffield, we have a large number of collaborative efforts with other academics and pharma companies. And a good example of that is RDAT. This is a program involving 34 partners, co-led by Sheffield and Pfizer. So Blackfin Bio is a University of Sheffield spin-out focused on gene therapy for rare diseases of the central nervous system, and it will leverage Sheffield expertise, infrastructure, and AAV, adeno-associated virus platform IP, and accelerate commercialization of these assets. So where we are, one of our objectives obviously is to incorporate the company, and this was done in December last year with initial seed round of 2.75 million, and we are currently working towards the second seed round of up to 5 million. This is to start the phase 1 clinical trial with our lead product, and also to progress the second, our product assets related to dopamine deficiency. So the team, you can see here that we assembled a leadership team with a variety of expertise, including biotech experience, Peter Nolan, who worked for many years at Oxford Biomedica, scientific, regulatory, and other expertise. We have also a medical team involved in the company. So Darius, who is a neurologist at Boston Children's Hospital, who will be leading on the IND and as the investigator for clinical trial for the lead program, and Professor Stefan Palfi, who is leading on the dopamine deficiency program. So the pipeline, the lead program is gene replacement for spastic paraplegia type 47. And I'm excited by the second program, actually, dopamine deficiency diseases. And we are currently bringing this program from Oxford Biomedica. And the third program is neurodevelopmental disorder linked to SANGAP1 mutations. So in terms of timelines, I won't go through the timelines in detail, but the key message here is that with the lead program, we are progressing towards the IND submission and to start the recruitment for phase 1/2 trial at the end of the year. So the first program for gene therapy for spastic paraplegia 47, this is a disease caused by mutation, recessive mutation. It's a loss of function. That's why gene replacement is very attractive for this condition. And it's caused by mutation in AP4E1, B1 gene. So this is just background information. You can see here that AP4B1 is one of the 4 units of AP4 complex, and mutation in one of these units cause 4 different diseases with similar symptoms. So the technology is based on AAV vectors, and we plan to deliver this lead product through cystina magna, basically in the cerebrospinal fluid in patients. Current status of the program, we generated proof of concept both in vitro and in vivo, and we completed a GLP tox in NHP, in addition to the long-term safety study in mice, plus manufacturing of clinical vector is being completed at the moment with the lot release to be finalized by summer. And the clinical study, we are currently working with Boston Children's Hospital for the clinical design and IND submission to FDA. So I mentioned the exciting program about dopamine deficiency, and the reason why it's exciting because you have one product for dopamine synthesis that can be applied across various conditions caused by dopamine deficiency. So this is quite exciting for the company. The third program is SYNGAP1. This is a neurodevelopmental disorder caused by mutations in SYNGAP1 gene, and it's autosomal recessive. So again, gene replacement is a very attractive approach to as a treatment. So we are currently working on generating the therapeutic vector, and around that generating IP as well. In terms of competitive position advantage, we already secured the orphan ODD from FDA and rare pediatric disease designation, and we are planning to sponsor proof of concept for the second asset at the University of Sheffield. Where we are at the moment, so we completed the first 4 milestones, and we are generating the GMP manufacturing clinical vector to start phase 1 trial, and we work towards second round seed funding to support clinical trial, but also to progress dopamine deficiency asset. Thank you.
Thanks very much, Mamoon. Everyone's kept to time so amazingly. We do have 3 minutes for questions. So any questions? No? No. Awesome. Thanks so much. So we're going to have a 2-minute break so that we get back on time because we're too early. It's never happened before, but we want to be on time so that people coming in don't miss out. So thanks very much, everyone. I think hopefully that was a useful micro break of 2 minutes. So let's jump straight back into it. If we could just bring up the slides. So once again, I do not miss that. So yeah, it's my pleasure to invite Fraser Murray, CEO of Pheno Therapeutics.
Thank you. Can you hear okay? Yeah? Okay.
Thanks very much to LifeArc for the invite to speak and tell you about phenotherapeutics. So pheno is focused on targeting one of the brain's natural repair cells, oligodendrocytes, which produce a protective sheath that wraps around neurons called myelin. That myelin allows for normal conduction, and in many diseases, the myelin's damaged and you lose function. So our aim is to drug the oligodendrocytes to help them repair. So why is this remyelination interesting to us? Well, we believe it's a huge unmet need. MS, multiple sclerosis, is probably the best known demyelinating disease, and it's still the leading cause of disability, non-traumatic disability in young people. But there are many other CNS disorders that are characterized by demyelination, and they range from rare diseases like leukodystrophies, Alzheimer's, ALS, and even psychiatric indications like schizophrenia, where you have deficits in myelin that lead to functional decline. We're focused on a target called GPR17, been around for a long time, very well-known target in demyelination, remyelination, I should say, but it's been very difficult to drug. So we're quite excited to have a molecule that's almost clinic-ready. Our plan is to initiate studies, phase 1 studies, in the 4th quarter of this year. And we think we can get a proof of concept within 3 years, phase 2 proof of concept. So the company was formed in 2019, seed funded by LifeArc and Advent Life Sciences. It's an Edinburgh University spin-out based on the work of Professors Siddharthan Chandran and Neil Carragher. They had a really unique screening platform, high-throughput screening platform, with a lot of phenotypic information that we thought would allow us to pick targets for remyelination with a greater probability of success. So the initial seed funding we used to validate the platform, do a lot of screening, shortlist a number of targets that we thought were highly attractive to pursue. GPR17 was top of that list. We then managed to in-license a program, GPR17 antagonist program, from UCB. UCB had worked on the program for a number of years, deprioritized it for strategic reasons, and we managed to in-license it at lead optimization stage. We then identified a candidate molecule, which we call PTD802, and having identified that, we took an internal round from Advent and LifeArc to get us to phase 1 ready. And we have just recently completed GLP safety and tox. We have a clean profile, wide safety margin, and we'd anticipate being able to dose, as I say, in the 4th quarter of this year, pending a Series A raise, which we're just kicking off. So again, if anybody would like to discuss, please let me know. Excitingly, we have generated preclinical data in other non-MS indications, so Parkinson's disease, Alzheimer's, and ALS. And it really fits in with some of the emerging literature on GPR17 that it's a broadly applicable neurodegenerative target. So what does it do? It's almost exclusively expressed in the brain. It's almost exclusively expressed on committed oligodendrocyte precursors. And it's a negative regulator of myelination. So if you have too much of it, you do not produce myelin. Your oligodendrocytes cannot myelinate and repair. So on the top here, this is nicely illustrated in some MS post-mortem brain. You can see here high dark areas or high levels of GPR17 expression, and that correlates beautifully with the demyelinating lesions. And what happens is the oligodendrocytes are stuck in this immature state. They can't mature, they can't produce myelin, they cannot repair. If you block the receptor or you knock it down, you take the brakes off, they come out of suspended animation, they mature, and they wrap the myelin and they repair. And importantly, they give functional recovery. We can see that. We've shown it from the point of view of demyelination, and also excitingly, CSF neurofilament light, a marker of neuroaxonal damage. You can see in these MS models that we get a decrease in the model demyelination, and we get a decrease in neurofilament light. But nobody goes to their doctor complaining that they've got a myelin deficit. They go because they've lost function, whether it be cognitive, motor, behavioral. And we've shown here, and I've deliberately picked a Parkinson's and ALS model,
we get a really nice recovery in cognitive performance and a really nice recovery in motor score in the PD and ALS models, respectively. So really exciting target. Looks like it does what we expect it to do. It does influence oligodendrocytes, it increases repair, and most importantly, it restores function. So in summary, FeNO, we believe we're addressing a really significant unmet need. There are no remyelination therapies. MS patients still have progressive disability despite 15 launched immunomodulators, which deal with the immune component, but they still have demyelination, they still lose function. We've got a really efficacious, we believe, remyelination drug. It seems safe, well tolerated in preclinical GLP tox. We think it's the first and best in class GPR17 antagonist. Importantly, the candidate's covered by granted patents in all the major territories. We've shown efficacy in multiple models. Data supports effects not just on remyelination, but also on function, as well as neuroaxonal loss. Pivotal tox is finished. Wide safety margin appears to be there. First in human pending the raise in Q4 of this year. And most importantly, a phase 2 proof of concept using highly informative advanced imaging endpoints is possible on a biotech funding envelope. So we're very excited to push that forward and hopefully within 3 years see that it works in patients. And that's all I'm going to say. Thank you very much.
5 minutes. 5 minutes. Questions? I'm going to start with one then. Phase 1 trial design, is it just single ascending dose, or are you going to have any biomarkers to indicate that there's kind of target pathway engagement?
Yeah, so we've got traditional SAD-MAD, but with target engagement. We seem to have a potential EEG biomarker. We also have some exploratory CSF biomarker work ongoing, which again, we think based on the receptor, its localization and what it does in the literature, certainly in knockout mice, etc., we could have some CSF target engagement biomarkers as well. We wouldn't expect to see myelination biomarkers in a phase 1, it's just not long enough to see that change.
Great. Any further questions? I've got one more. Stephen, please. Sorry, Stephen isn't a plant, but he does work for LifeArc. But this is not arranged.
Talk into the mic.
Talking to the microphone. Yeah. What I was interested in is it's only expressed by talk into the microphone. It's only expressed by oligodendrocytes, and yet we see these variety of diseases that can be treated. Have you any thoughts on the mechanisms that might be involved beyond simple myelin repair?
Yeah, so that's a great question. And I think all of those diseases do have literature supporting oligodendrocyte issues, let's say. But oligodendrocytes do a lot more than just produce myelin. They have trophic effects. There's also myelin plasticity, where they can change the distance between the junctions in the myelin, which alters synaptic transmission. They can unwrap and rewrap and bring circuits into play. So there's a whole host of things that can occur. I mean, there's the possibility there could be, downstream changes post-signaling, but we believe that you could really pin a decent story on oligodendrocytes for all of these effects.
Brilliant. So thank you so much. Fascinating. Great. So with the next slide. Perfect. So I'm happy to introduce Alex Marshall, who's Head of Business Development, Licensing and Alliance Management at RQ Bio.
Thank you, Jason. Hello, everyone. Today I'm going to give you a bit of a background on RQ Bio and what we're doing to provide instant immunity to protect vulnerable patients. So RQ is, we're seeking to discover potent and broadly active and long-lasting monoclonal antibodies to prevent viral disease and hospitalizations and death in large vulnerable populations. RQ was founded in 2022 by experts previously involved in the BIA Antibody Task Force, which was a community set up to support the UK COVID response, looking at antibodies. And initial seed funding was provided by LifeArc. Now, in collaboration with the University of Oxford, we discovered and characterised a new mAb that is now called Sipavibart, which was licensed to AstraZeneca in 2002 for a $157 million deal, including upfronts and potential milestone payments plus royalties. Now, Sipavibart is now in phase 3 clinical trials for COVID-19 prophylaxis, and RQ is well funded to deliver multiple discovery programs. So who's involved in RQ? RQ is founded by an exceptional group of founders in Claire, Mike, Paul, Jane and Gavin, and further strengthened by 2 senior industry executives with deep sort of infectious disease vaccine experience in Rip and Jack. So what's the RQ team been brought together to do? So we know that vaccination doesn't adequately protect vulnerable groups against disease. So if you look on the left here, this was work published in The Lancet, where they compared the neutralization immunity response to ancestral strain COVID vaccine in 2 groups, healthcare workers and immunodeficient patients, and showed after 2 rounds of vaccination by COVID vaccine that a much higher proportion of immunodeficient patients didn't respond to the vaccine. So the vaccine just didn't work in the immunocompromised patients. And this is something which is increasingly being recognised and discussed by Big Pharma. This is work published by Janssen at ID Week last year. And it looks at the influenza hospitalisation risk of 5 million patients between 2015 2019 influenza flu seasons. And they looked, the number of groups have a much, much higher risk than the overall group of vaccinated patients. And in fact, immunocompromised transplant patients have almost 14 times the risk of hospitalization compared with the all-vaccinated group. So vaccines don't work on vulnerable patients. So what can we do? So rather than giving a patient a vaccine, expecting them to produce an antibody themselves, we can give the antibody directly, and it works very well. So monoclonal antibodies offer robust clinical efficacy for vulnerable groups. So if you look on the left here, for COVID, 80% protection from infection was achieved with mAbs, and consequently protection from disease and death from COVID infection. For RSV, similar efficacy was achieved to the level of vaccine in adults. And with the use case of Beyfortus for children in their first year of life, it's likely we're going to see whole birth cohorts treated with Beyfortus. And mAbs are not limited to viral diseases alone. The CIS-43 was shown to have improved efficacy over malarial vaccines in a phase 2 study. So when you combine mAbs with half-life extending technologies, you can get 6 months efficacy, 6 months protection with a single dose. So this is where mAbs sort of fit into the prevention treatment paradigm. They sort of bridge prevention, treatment, and mAbs that are neutralizing, the kind of ones we're looking at, can be used for both treatment and prevention. And this is sort of changing the way healthcare systems are thinking about protecting vulnerable patients. And we're moving rapidly to discover unique mAbs that will be first in class, best in class in terms of potency, breadth, and resilience to pathogen genetic variation. So how are we doing this? Well, we're leveraging an evolutionary and computational guided approach to passive mAb discovery. We've developed a novel method involving rational immunization of naive human immune repertoire mice with an evolutionary guided selection of viral antigens. So the mice contain all the antibody of a repertoire of the humans. So immunization of the mice leads to novel mAb discovery. And by using properly folded, carefully selected antigens that are stably folded, we can induce a novel immune exposure event that's never been seen in humans before. And this allows us to target more conserved immune subdominant regions of viral proteins that are otherwise hidden to the human immune system. So on the left here, you've got the influenza hemagglutinin, and there's regions in the head region which are conserved across different influenza strains, but the human immune system doesn't see them very well, and our approach allows us to target those. So in this way, our method seeks to generate potent and broadly neutralizing antibodies that cannot be found in nature and that can neutralize known variants and block infection by new variants that evolve over time. So how are we using this method? So we're using it across a number of our discovery campaigns where we have funding and strategic partnerships in place to deliver on our active discovery programs. So in COVID, we're working with Oxford as our discovery partner. Well, I've mentioned already we have a mAb in phase 3 with AstraZeneca, and we have a number of leads that we're characterising here. In influenza A, we're working with AbCellera over in Toronto in Canada, and also LifeArc, where we're looking to discover mAbs that are resilient to future seasonal variations, antigenic drift of influenza. For RSV and HPMV, we're working with LifeArc, where we're looking to discover mAbs with broader activity than those currently on the market that can neutralize both RSV and HPMV. And in CMV, we have an earlier program collaborating with AbCellera and sick kids to discover novel antigens to immunize the mice with. So this is RQ. We're providing instant immunity to protect the vulnerable. We're based in Scale Space in the UK. This is some of the team, a lot of the team, not all of the team though. And if you are specifically interested about anything we're doing here in terms of instant immunity, then feel free to contact me on the email address above. Thank you very much.
We've got 4 minutes.
Oh, right. 3 days.
Any questions for Alex? No? Great. Well, we're going to have another micro break to refresh yourselves. Speak to the person next to you. Connect randomly. Thank you. Welcome back, everyone. Thanks so much for following instructions to talk to the person next to you. Pretty much everyone was talking then, which is ideal. Next, I'm very pleased to introduce Arndt Schotelius, CEO of Maxion Therapeutics. Thanks.
Thank you. Good afternoon. I'm really excited to be here sharing the Maxion story and how we developed not-body therapeutics to treat ion channel and GPCR driven diseases. So our vision is really render ion channel and GPCR driven diseases treatable by biologics to improve patients' lives. We believe selective targeting of ion channels and GPCR has really potential to address a wide range of untreated or poorly treated diseases. And what we'll briefly introduce to you in the next slides, our revolution in not-body technology, we think really can overcome these key challenges ion channel GPCR antibody drug discovery that has been existed for quite some decades. We are up to raise a Series A to accelerate transformation of Maxion into clinical stage biotech company starting soon. Let me see. So we have a really experienced, great leadership team, are backed by wonderful investors. Big thanks here. The organizers LifeArc, also BGF and Monograph. I've just taken the helm CEO a few weeks ago, March 1st. Very exciting after quite a journey, 25 years in leading and building quite some antibody leaders over the years. I've enjoyed building research and development organizations, bringing many molecules to clinical proof of concept, including autoimmune disease. Anish, CSO, and John, now CTO, having led the company as CEO until recently, of course, are the co-inventors and co-founders of Maxion. They have also co-founded Iontas and made it a very successful company with 6 clinical antibodies in development. In addition, Anish brings really deep experience in antibody drug development as well as not in engineering. And John, of course, being the co-inventor antibody phage display, Nobel Prize winning work, and the co-founder of Cambridge antibody technology, so being around for quite a while. We see a huge untapped opportunity for biologics with the not bodies. There is an unmet need. I mentioned they play a role in many multiple diseases. And when you look at the small molecules, current standard of care, as small molecules often do, suffer from some limitations, potency, selectivity, insufficient exposure that can lead to poor clinical performance. Now, the opportunity for this platform, engineered antibodies, can offer superior selectivity, exposure. They're well proven therapeutically. But interestingly, there is no antibody drugs targeting ion channels today in clinical development or even anywhere approved. And when you look at the challenge of generating good functional antibodies, there is a significant challenge in the discovery stage that with our tools we can adequately address. So the solution we are providing is our proprietary not-body technology. It unlocks the discovery development of these antibodies. We can believe we can deliver differentiated drugs. Now, what are NOTINs? I only have 8 minutes, so I'll keep it very short. And please, John and Anish are here, hopefully also in the reception to speak about that more. They're naturally occurring secreted, small peptides. Some of them actually are approved or in development as drugs, but they have severe drawbacks. They're not really drug-like. They have very short half-life, often lack specificity. They're difficult to engineer and difficult and complex, expensive to make. So what we have done in developing the not-body technology, really taken all the advantages an antibody have, of course, known long half-life, enhanced specificity, engineering using the display technologies that we offer, standard manufacturing, and the NOTINs, which are excellent ion channel modulators. They have access to the membrane proximal epitope, and in their modular scaffold, they can be used interchangeably with thousands of them available and make these not bodies that really we believe are now creating safe, efficacious, long-acting biologics, antibody-like drugs that can address these problems. Some examples here, just very high level, as we have explored the potency of these ion channels. One target, I'll go into a little bit more detail, Kv1.3, plays an important role in autoimmune disease. And you see with the knot body developed here, it goes down to low nanomolar potency. There's also an acid sensing ion channel that could be used for ischemia or possibly stroke. And there's NAF 1.7, probably known to some of you, quite a well-explored target, but with the wrong molecule so far that can be used in chronic or acute pain. So a broad therapeutic applicability of the platform. Now, are these antibodies developable? Are they drugs? Yes. When we compare, for example, to trastuzumab, they can be manufactured. We see good expression developability compared to a standard antibody. And also when we look at the PK profiles, clearly kind of antibody-like, and over a normal not an over 100-fold improvement of half-life, so very drug-like properties and good developability. Now, a few words to our lead program, MAX001. It has broad use in many autoimmune diseases. We get that. You're aware that they're currently targeting drugs, very successful, but they still have unifying limitations. Often it's a blanket immunosuppression with increased infections or even malignancies, and a lot of patients still are failing to respond. So a huge unmet medical need that we believe with the technology we can address. Speaking a little bit more about the biology, Kv1.3, when you think about the T cells, and I won't go into great detail, but the activated effector memory T cell, the culprit really in many of the T cell-driven autoimmune diseases. This cell is actually literally addicted to Kv1.3. It needs the ion channel that drives a lot of the activation, whereas other T cells like naive effector or central memories are driven by other ion channels and not Kv1.3. Quite a selective, important target for the main culprits of autoimmune disease. With that really has broad therapeutic potential. I've listed some of the diseases that we're looking at. So I have 1 minute left. I better hurry up. It's a well-explored target with compelling validation, genetic validation, clinical validation, commercial. But the peptides that I explored, those small molecules, have the known and discussed drawbacks. So that's where we feel there is a big possibility to differentiate and come up with a differentiated and successful drug. So in summary, we really feel this is a truly novel technology addressing significant unmet needs. The strong management team has delivered the objectives of the pre-Series A raise that has delivered and achieved very strong in vivo proof of concept. The lead program is going to clinical candidate selection. The platform has been successfully scaled with 2 new programs. We have granted patents, broad patent protection, fully functional headquarters just in the south of Cambridge. And we will, as I said, go out and fundraise this year to really transform Maxim into a clinical development organization and drive the lead program into phase 1. If you're interested, happy to talk here. Also, again, co-founders John and Anish are here. Just approach us to discuss over drinks. Thank you.
One minute for one question. No? Brilliant. Thank you so much, Arnt.
Thank you.
So I am now pleased to introduce our last speaker, Susan Hill, CEO of MESTAG Therapeutics. Susan.
Good afternoon, everybody. Thank you for the invitation to tell you a bit about MESTAG. Get it going the right way. Wrong way. Right way. There we go. Right. So we founded MESTAG 4 years ago now to focus on activated fibroblast populations. And many of you will have heard of fibroblasts in terms of cell culture, fibrosis, wound healing. Our interest in fibroblasts was a little different. we're specifically interested in the impact that activated fibroblast populations have on the immune system. So we tend to think of ourselves as an immunology company, where our programs and our technology is inspired by understanding how fibroblasts are influencing immune effector cells. And this was really driven by a very comprehensive set of single-cell data generated by our founder team across inflammatory disease and oncology. So as a company, we work across both of those therapy areas. So a summary of who we are today. So our focus is on fibroblast immune insight. We have a pipeline of first-in-class antibodies, and our lead program, M300, which I'll describe in a minute, has been specifically designed to induce the formation of tertiary lymphoid structures in tumours. This is a whole new approach for solid tumours, super exciting, and we are now thinking about how we take that into clinic. Our second program, which we call M402, is a next-generation stromal checkpoint agonist. So these are antibodies that we're developing for the treatment of autoimmune and inflammatory disease. We have a platform, which is a series of human tissue assays that involve activated fibroblasts, immune effector cells, and diseased tissue. And we interrogate this platform across oncology and inflammatory disease to look for new targets. And we're currently doing that in collaboration with J&J. And we have a really super collaboration with them. We've raised, since we founded the company, $45 million to date, and our investor syndicate you can see here. So SV Health Investors, JJDC, Forbion, Google Ventures, and North Pond. So these are our academic founders. So this group, over the last sort of 5 years or so, have really created a new understanding of activated fibroblasts in biology. So they are a team of rheumatologists, inflammatory disease experts, oncologists from the UK, so from the Kennedy Institute of Rheumatology at the University of Oxford, so Chris Buckley and Mark Coles, but also from the East Coast US, so David Tuveson there is the head of the Cancer Center at Cold Spring Harbor, pancreatic cancer specialist. Shoma Ray Chowdhury is a bioinformatician at Broad, and Michael Brenner was previously the head of the rheumatology division at Brigham and Women's. And this group, individually and collectively, had looked at fibroblast subpopulations on a single cell level and had uncovered some really interesting biology and actually, pointed towards activated fibroblasts having some of the roles of immune effector cells, and that was what really caught our interest. So around the framework that our academic founders had created, we've now built a team of drug developers, and we are now in the process of transitioning this organization from a discovery stage organization to a clinical stage organization. So our CSO, Ray Jupp, has 30 years experience in the industry in inflammatory disease, autoimmune, fibrosis, and oncology. As a company, we're based in Cambridge, so we're kind of in antibody central, so we have access to some amazing antibody technology. And we're now building out our team with clinical and also manufacturing. And our lead program actually is just starting pre-IND development. So this team has sort of built on that foundation of biology. And we now have a pipeline of first-in-class antibodies. So our lead program, M300, is a bispecific. So it's a fibroblast-targeted bispecific molecule, which we use to induce tertiary lymphoid structures. And we're expecting that program to be heading into clinic towards the end of next year and into the beginning of 2026. So what is a tertiary lymphoid structure? So this is a picture of a tertiary lymphoid structure, and here you're looking at a human solid tumor. And it's an aggregate of immune cells, so it's B cells and T cells. It looks very much like a germinal center in a lymph node, for those of you that are familiar with that biology. And it really functions as a site of entry and education for immune cells into solid tumors. And it turns out that activated fibroblast populations are really important in the formation and maintenance of these structures. So why are these so important? So we find them super exciting because this is a means by which naive lymphocytes can enter the tumor and can see tumor antigen in situ in the tumour in a way which is not done without TLSs being present. And actually it's become very clear, sorry, pointing this in the right place, there we go. So it's become very clear over the last 3 to 4 years how important tertiary lymphoid structures are. So this is some of the early data that was published back in 2020, which started to point towards TLSs having a really important role in terms of patient outcomes. And there are now more than 10,000 subjects worth of data in the literature that shows that patients that have TLSs in their solid tumours do better, regardless really of what they're treated with. And TLSs also enhance response to therapies, including checkpoint. So we've designed a bispecific that conditionally induces tertiary lymphoid structure formation in solid tumours. And because I'm going to have to mega speed up, but because we only have a short amount of time, I just have a little bit of data to show here. So this is in an orthotopic model of breast cancer, and we're using this is a very cold model with very few tumour antigens in it. We use a peptide here as a kind of surrogate of tumour antigen in general. And what you can see is a little bit of a response with just the peptide and an adjuvant. In the blue line there, you can see monotherapy with our bispecific. But what's really exciting is that green line at the bottom, where we're seeing flatlining of tumours and actually tumour regression. And you can see on the right-hand side there, tumour weight correlates very nicely with CD8 T-cell infiltration. This is exactly what we would expect to see based on the mechanism of action of our programme here. So super exciting. So we're now taking this as fast as we can with urgency to clinic. We've got a lot of important decisions to take about how do we explore this mechanism, because this mechanism, which really brings the immune system into the tumour, has a really good fit, in particular with other therapies that release or increase tumour antigen in tumours. So that can be things like radiotherapy, can be things like ADC. There's some very interesting synergies with things like mRNA vaccine. We're also looking at things like immunogenic chemo. So we've got quite a lot to do and important decisions to take in terms of how we explore this from a clinical perspective. And we're quite excited to have the opportunity. So just briefly, our remaining programs, I'm going to go over, I'm sorry. After everybody being on time, I'm just going to take an extra minute. So our checkpoint agonist program, M402, we're really taking the same approach of very thoughtful antibody engineering and an understanding of fibroblast biology to develop a checkpoint agonist program against a novel target. And we are incorporating two things really into this program. One of them is that this target has a very different expression across immune cells. So what you can see here on the bottom there is PD-1 and BTLA, which are targets of clinical stage checkpoint agonist programs, primarily T-cell and B-cell expressed, as you can see, and our target has a very different expression profile. We're particularly interested in exploring myeloid biology with this particular program. But we're also building upon learnings of the first generation in terms of how to agonize checkpoint molecules and how to specifically dampen down immune cell subsets. 1 minute left, I can see at the back. So we're very excited about this. This program's a little earlier. We're right in the middle of some quite complicated biology and antibody design at this point. But we're very excited by this because even though checkpoint agonists are a clinical class, and they're in phase 2 in indications like RA and lupus, there is a lot of biology there still left to explore and a lot still to optimize about these mechanisms. And that's really what we're trying to do here with our 402 program. So just to finish up, we also have a platform that we use for novel target discovery. So it's comprised of a number of human tissue assays. We're currently working with Janssen on this to explore inflammatory disease. We are talking to other partners with respect to further partnerships in this area. So these are human tissue assays. They're complicated, taken a long time for us to develop, but they are robust, high quality, pharma grade screening assays. And we've had a great collaboration with J&J. And we're interested if anybody would like to talk to us about the platform. We're very happy to discuss it. So with that, I'm over time, but it's been a pleasure talking to you. If you have any questions, please don't hesitate. Did I use up my question time?
Thanks so much. One minute. Any questions? Brilliant. Thank you so much, Susan. Okay. So before everyone disappears, I would just like to thank all of our speakers for 7 really inspiring talks. It's just really, really exciting. You have 10 minutes before the afternoon plenary. Professor Lord Robert Winston, Lord David Willits talking about turbocharging translational science. In the UK, it's the big hall just next door. And then after that, at 5:15, please join us for a network reception at the Gallery Hall. But thank you all so much for your time. Thanks.
Yeah, oh yeah, cool, fabulous. Yeah, that was really good work.
initially, I knew him well, so we had a good discussion, but he was sort of taken aback. My idea is cool science, not kill science. No, this is the way it is. And that's exactly what it is. I spent a lot of money developing the molecules. You can do those experiments. I wouldn't.
What's worth in interesting. Fantastic.
One of the five is working very well. That's what we focus on. I've got two others that are sort of the one is working well.
And you feel like you actually like how to get money to get the person who knows how to do that kind of research into. Yeah, I'm sure that your product can sell it, but it's that kind of. That's the problem. I did this sort of misunderstanding of August last year. Taxi to a person. So you can see I should own your research. There were 12 floors of cars. 12 floors of cars, 5 schoolrooms, 5 people, and 65 staff. Maybe he was going to use it. Who got trapped with that? 65 staff, maybe.
Yeah, right.
If you say not a flooring, that's for sure.
Yeah, about sort of 5 minutes time we had to stop around the farm. Okay.
What's the other guy that
Yes, please.
Tata?
Just copy one thing over to I've just finished. Thank you very much. No problem. Oh, you already got the. Yeah, what to do? Right, yeah, cool. That's what Is this as next time? So that is as next time. What I've done, I've already made the folders on this. I don't know if it's SSD, so it might be longer. Yes. Okay. No problem. Yeah. But at the moment, it's got the AM. So we have three things. Perfect. Yeah. But there's a folder for each room. So yes. Thank you.
Going between you.
Yes.
See you. Nice working with you.
Yes, nice working with you.